scientists, professors, delegates, research scholars from around the corners of India as well as abroad. It gives me immense pleasure to welcome you all to this international conference on Agriculture for Sustainable Future, Agri Vision 2022. Previously, we had planned to organize this conference from February 6th to 8th. However, due to this Omicron crisis, we were compelled to postpone it to March 6th to 8th, and it all became possible because of the able guidance, support for the collaboration of Department of Botany, Ravenshaw University. I would request everyone to give a big round of applause for Department of Botany, Ravenshaw University. They have been very kind and supportive in organizing this conference. I welcome you all over here, and I believe that you all are going to enjoy this 3-day long conferences. So a brief introduction about this first day. So we'll be having our sessions till 4 o'clock in the afternoon. After that, we'll be having our official inauguration as well as the inaugural meeting, which is going to be inaugurated by Honorable Director General of ICAR, Dr. Trilochan Mahapatra, sir. Due to some official exigency, he couldn't join us physically. However, he is joining virtually. In addition to him, we'll be having among us Dr. Jai Krishna Jaina, sir, who is Deputy Director General of Fisheries Science, ICAR, along with Dr. Padmini Swai, Director NRRI, Dr. Ashok Kumar, Director NPPGR, Dr. Saroj Kumar Swai, Director SIFA, Honorable Vice Chancellor of Ravenshaw University, Professor Sanjay Kumar Naik, sir, and Honorable Vice Chancellor of OUAT, Professor Pavan Kumar Agarwal, sir. This grand event has been possible with the kind support and generous, I must say it's a generous support from all our sponsors and supporters. So not in order, Larsen and Toubro, Central Bank of India, Department of Tourism, Government of Odisha, Bank of India, APEDA, NABARD, Tea Board of India, National Medicinal Plant Board, Department of Biotechnology, NALCO, TriFed, CT, CRI, COIR Board, Botanical Survey of India, Central Institute of Women in Agriculture, Central Institute of Brackish Aquaculture, Central Institute of Freshwater Aquaculture, Central Horticulture Experiment Station Bhubaneswar, Mahanadi Coalfield Limited, Canada Bank, Coffee Board, ICATA, CMFRI, Coconut Development Board, NRRI, Odisha State Warehousing Corporation, Bharat Petroleum, Odisha Agro Industry Corporation, NBPGR, National States Corporation, CRIPCO, PCRA, Ruchi Food Line Limited, OMFED, Paradeep Phosphates Limited, ORMAS, then Department of Culture, Odia Language Literature, Government of Odisha. So I'm really thankful to all our sponsors and supporters. So without taking much of my time, I would request to proceed this program ahead. Thank you so much.
Thank you so much, Mr. Dibyan. Next 3 days.
probably by the end of May 2022. This campus houses 25 departments and provides courses and programs in undergraduates, postgraduates, masters, MPhil, and PhD. The department botany in this campus started in 1906, already about 120 years old, and has a rich legacy of botanists who have been graduated from this institution and have gathered international reputations. I welcome you all to this campus to have a glimpse of the campus and enjoy your stay during this 3 days conference. Enjoy all the scientific deliberations and all the hospitality. A brief account of the conference, Agriculture for Sustainable Future. I just will make what we expect from this conference. Probably you also expect from this conference, that when we come to agriculture for sustainable future, the 3 words, agriculture, sustainable, and future, have their own meanings. Agriculture started sometimes around 10,000 years old, but it is not the agriculture that we are practicing today. Agriculture has gone a paradigm shift, especially during last 50 years, with regard to the attempt to produce more, diversifying the agriculture, diversifying production, and making agriculture an industry, not a hobby. Sustainability, when it comes to sustainability, we have the question of sustainability in production, so both in the qualitative and the quantitative terms. Therefore, now agriculture is different from what we had agriculture 20 years before. And with regard to future, what type of future we see for the agriculture? What type of crop we will be cultivating, whether it will be the same crop that we are cultivating now, because the crop we were cultivating or the man was cultivating 100 years before or 1,000 years before are quite different from what we are cultivating now, and we do not know what we will be cultivating in the future. And also the cropping systems, the cropping period in the field, and looking beyond the crop fields. Because we have now realized that probably the field available to us for agriculture is not enough to satisfy our demands. So we have to look beyond the crop field, look towards the ocean, look towards the water bodies, look towards surfaces which are not land, like straw beds, tissue culture laboratories, aeroponics, hydroponics. Probably they will be the future of agriculture. Probably time will come when we will not need apple, rather we will make the tissues of apple fruits and we will purchase the tissues of apple fruits from market and we will consume it directly. So probably that is the future of agriculture. Our learned scientists will have deliberations and probably they will throw light in this regard. And another major problem that we are facing now is the agro-ecosystem health. We have dumped thousands of tons of chemicals into it. 3,000 different types of insecticides we are using. We have dumped them into the field. Residues are everywhere. Pest is a problem. Pathogen is a problem. Natural control system is almost non-existent. So all these things and all these challenges are to be dealt in future when we are considering agriculture as a venture. And agriculture not as an engagement, rather agriculture is to be converted into a profitable business. In this session, we expect that there will be deliberations on stress tolerance, varietal improvements, integrative farming, pest management through our environmental approaches, and a number of success stories are expected to be presented. There are, as I see from the programs, there are 2 plenary lectures and 23 keynote addresses, 21 young scientists presentations. There will be about 20 presentations by young researchers through oral presentations, and there will be more than 40 posters to be presented. I hope all of them will be academically very rich, and all of you who have come to this conference will be benefited from the deliberations that will be presented to you during last 3 days. I expect that you will enjoy your stay here. Thank you very much. Welcome you all to this conference and hope that you will have good memories once you attend the conference and go home. Thank you very much. Jai Jagannath.
Thank you so much, sir, for sharing us briefly about what we are going to witness in the next 3 days. Now requesting Professor Vaishnav Charan Tripathi ji, former Vice Chancellor, Ravenshaw University, and Professor Jawaharlal Nehru University, New Delhi, to please join on this podium and address the gathering on the topic genetic manipulation of chlorophyll biosynthesis and degradation pathways increases photosynthesis and stress tolerance. So let's welcome him with a huge round of applause, please. Thank you.
Okay, good. All right. Good morning to all. I am very happy to be here in this very hall that inaugurated 8 years ago, more than 8 years ago now. Last time I came, I talked about why chlorophylls are green, why chlorophylls evolved green, why not black. Black would have been ideal for chlorophylls because they would have absorbed all the wavelengths of light, okay, and utilized in photosynthesis. But they evolved green. That means they do not absorb green light. We talked about it before, and this topic is completely different from that. So I'll not talk about it. I'll talk about today how you can utilize, because the biotechnology conference, I'll talk about how we can utilize chlorophylls, manipulate chlorophylls, okay, to increase photosynthesis and plant productivity. Okay. There are many views of thoughts on chlorophylls. Some say that if you decrease the chlorophyll content from plants, photosynthesis will increase. Yeah, it may increase in a crop canopy system when there is a lot of dense planting and mutual shedding of the leaves are taking place. And if chlorophyll content is less, then probably it will percolate better to the bottom of the leaves, and probably it can happen. So increase photosynthesis. But if you take individual tree, okay, or individual plant, then I have shown many times that it's better have plants have more chlorophylls. Although nobody has really shown the concept, proved the concept actually thoroughly. There are some reports here and there that actually decreasing chlorophyll content decreases or increases photosynthesis. If you grow a plant in low light, that chlorophyll content increases. Why does it increase? Because plants need to harness more chlorophyll, more light. Okay, therefore they increase their chlorophyll content. Okay, in low light, so that they harness more light. So therefore chlorophyll content is not a limiting. Okay, since very fact that if you grow a plant in low light, they increase their chlorophyll content to harness more solar energy. Okay, because so that they are going low light, therefore the photosynthesis will increase. Okay, therefore if this fact tells us that chlorophylls are limiting in plants. Okay, not if you decrease the chlorophyll content of plants, not necessarily increase the rate of photosynthesis. Therefore, I talk about chlorophylls, the great manipulator of photosynthesis plant productivity. Okay. I don't know whether you can see this slide or not. Okay. But there's the glutamic acid is a chlorophyll biosynthesis intermediate. That glutamic acid is the amino acid. It is an amino acid actually. Okay, which is substrate for chlorophyll biosynthesis. So glutamic acid becomes glutamic tRNA, and glutamic tRNA utilized to produce delta amino levulinic acid, which is written here as. Does it go here or not? Okay, it is going there. Okay. Delta amino levulinic acid. Now it goes, but now sometimes it goes sometimes doesn't. I think it's gone bad. Okay. All right. Delta amino levulinic acid, ALA, and then ALA metabolized to many different pathways and reaches protochlorophylite. Okay. And protochlorophylite, is a light dependent step in the chlorophyll biosynthesis pathway. So if you grow a plant in dark, it doesn't green. Okay. Why doesn't green? Because there is an enzyme called protochlorophyll oxidoreductase, which needs light as a substrate. Okay. There are two enzymes in nature which requires light as a substrate. One is protochlorophyll oxidoreductase. Other is a DNA photolase required for DNA repair. Okay. It also absorbs requires light as a substrate. And this protochlorophylite is converted to chlorophyllite, okay, and then to chlorophyll, okay. And this protochlorophylite is converted to chlorophyllite with the help of light, okay. So enzyme that converts protochlorophylite to chlorophyllite is called protochlorophylite oxidoreductase. Are you able to see this slide or not? Yeah, you can see. All right.
Check.
Okay, good. Okay, this is protochlorophylite. Okay, and this chlorophyllite becomes absorbed. So chlorophyllite is produced when protochlorophylite is converted to chlorophyllite by light enzyme called protochlorophyll oxidoreductase. And chlorophyllite essence after that comes to chlorophyll. So therefore, when you grow a plants in dark, okay, they do not green. They are yellow because they are carotenoids, but they cannot synthesize chlorophylls because this enzyme protochlorophyll oxidoreductase requires light as a substrate for chlorophyll synthesis. With these words, I will go ahead, and this chlorophyllite. Okay. And if you grow a plant in sunset, okay. At sunset, therefore, what will happen? Protochlorophyll accumulate, even a green plant. At sunset, they'll accumulate protochlorophylite, but they cannot convert to chlorophyllite. Okay, during night time, they can synthesize chlorophyll only during morning sunrise. So therefore, no chlorophyll synthesis, actual chlorophyll synthesis takes place. No actual synthesis of chlorophyll takes place in dark, in night. Okay, only takes place during daytime. So these are the two priori I talked about. Okay, hope you understand this. Then we go ahead. And this is chlorophyll degradation. Okay, during senescence, lot of chlorophylls degrade. Okay, this is one thing. Now, during senescence, lot of chlorophyll degrades. Okay. Not only that, if the plant experiences stress, okay, like salt stress or drought stress, or whatever the stress is, okay, then also chlorophyll degrade. Okay. And so degradation, chlorophyll degradation is also problematic for the plant. Okay. Okay. If there is increased chlorophyll degradation. So chlorophyll could degrade in many pathways, but I'll say that chlorophyll phytol could be removed, then it becomes chlorophyllite, head group and tail group. So phytol could remove becomes chlorophyllite, and chlorophyllite becomes magnesium removed called piophorbyte. Okay, and piophorbyte, and there's an enzyme called piophorbyte oxygenase, which will form RCC and all that. So piophorbyte actually is okay. So what I want to say that if in case lot of chlorophyll degrade and this piophorbyte that's produced, it doesn't efficiently degrade to catabolites, then also harmful for the plants. So if the plant accumulates excess protochlorophylite, okay, it is harmful to the plant in the chlorophyll biosynthesis pathway. If the plant, this light photosensitizer, it absorbs a lot of light and can go to excited state if the plants are more protochlorophyllite. So when they absorb excess light, they go to excited state. When they come back to ground state, they give the energy to oxygen. Okay, so oxygen becomes singlet oxygen. Singlet oxygen is a rearrangement of electrons inside oxygen molecule. So it's called singlet oxygen. There's no extra electron involved. but it is a singlet oxygen. Okay, and singlet oxygen is very harmful, and that could kill the plant. Okay, therefore plants regulate both protochlorophyllates and chlorophyllite synthesis. But during stress conditions, all the regulations are lost. Okay, therefore, this regulation does not work, and the singlet oxygen damage, induced damage takes place on the plants. I am talking about an enzyme called phytyl kinase, okay, or VT5. It also leads to synthesis of vitamin E, okay. And it produces what? Tocopherols. We'll talk about that. This is vitamin E. It's produced in plants, okay. Called tocopherols. Okay. And this tocopherols is vitamin E here. This is tocopherols. And these tocopherols are produced in plants. I'll not talk about the exact synthesis on all that later on, but it's very important that there may be different kinds of tocopherols: alpha tocopherol, beta tocopherol, gamma tocopherol, and delta tocopherol. But out of all those, alpha tocopherol is the most important tocopherol that produce in the plants. They produce vitamin E. And this vitamin E is a quencher of singlet oxygen. It can quench singlet oxygen. So if vitamin E, as you can see, is highly hydrophobic. Okay. So since hydrophobic, it can remain in the membrane, thylakoid membrane, bound to thylakoid membrane, bound to membrane. Okay. And chlorophylls are present in thylakoid membrane. Okay. So when chlorophylls and vitamin E are very close, then the singlet oxygen produced by the chlorophylls or chlorophyll bags in the intermediates can also be quenched by vitamin E, tocopherol. And therefore, singlet oxygen concentration in the plant will go down, okay, because they're quencher of singlet oxygen. And therefore, because why I'm saying the quenchers? Because we don't have any, just like superoxides, okay, hydroxyl radical OH minus, or either H2O2, okay, also produced in plants. But we have enzymes for that, catalase, superoxide dismutase, glutathione reductase, and so on and so forth. Okay, but there is no enzymatic defense against singlet oxygen in plants. No enzyme. Once it is present, once it produced, it will create a damage unless it is quenched, destroyed immediately by certain things. There are two. Another quencher is carotenoids. Carotenoids are also singlet oxygen quenchers. So plants have evolved carotenoids to quench singlet oxygen also. But in stress conditions, when there are a lot of there are a lot of deregulation taking place, okay, and a lot of singlet oxygen are produced, okay. Why is singlet oxygen produced? Because let's say the plant is experiencing water stress, let's say, water stress. So there is no water in the field, okay. So photosynthesis stops, will decline, because all the enzymes photosynthesis will be deregulated. But chlorophyll is there, they will absorb the light, go to excited state, that physical process. Absorption of light is a physical process. So they will absorb the light, go to excited state. When they come back to ground state, they'll give their energy oxygen and singlet oxygen. Because these chlorophyll molecules, once they absorb a good excited state, since the photosynthesis is stopped, since the reaction center is stopped, okay, all right. So they can, reaction center is closed, let's say, reaction center is closed. Because they cannot get the excited energy because photosynthesis is stopped, photosynthesis pH2 is destroyed, okay, so, or pH1 is destroyed, okay. Therefore, they cannot get the energy from the sunlight. They can utilize it for photosynthesis. So that energy, what happens in photosynthesis? Energy absorbed, chlorophyll molecule absorb energy, sunlight, and that is utilized in photosynthesis. Okay. And that's why energy utilized, produce carbon dioxide, carbohydrate. But if that doesn't happen, if photosynthesis stops during stress condition, Okay, therefore reaction center is closed. Okay, it is not in a possible for energy to be utilized in photosynthesis. What the energy will go? The chlorophyll go to excited state, the physical process. They come back to ground state anyway. That is also another physical process. And they give the energy to oxygen, which becomes single oxygen. And this single oxygen becomes highly reactive. So under stress condition, single oxygen is overproduced because reaction centers are closed. Okay. All right. So it is therefore very important to limit the single oxygen concentration in plants. Okay. In light. Okay. Because there's very limit, no or minimal single oxygen produced during dark, very minimal. Okay. Most of the single oxygen produced are in light, daytime. Okay. So with these introductions, I will not go much to talk about, but I said the chlorophyll pathway, and if you manipulate phytal biosynthesis, phytal kinase, you're producing a lot of alpha tocopherol. Not only that, you also produce a lot of chlorophyll. Okay. Here the chlorophyll is degraded, you can produce phytal, and phytal becomes phytal kinase, so phytal phosphate, and phytal phosphate could go back and synthesize chlorophyll, chlorophyllite to chlorophyll. Okay. So there are two pathways. One, that this phytal goes, once chlorophyll degraded, the phytal could be utilized to form vitamin E. Okay. Or phytal could go back, to attach with another chlorophyllite to form chlorophyll. Okay. So our idea is to manipulate a gene called phytal kinase, VT5 here, phytal kinase. So it becomes phytal phosphate, and phytal phosphate later on becomes phytal diphosphate, and phytal diphosphate reacts with homogenous acid to form tocopherol. Okay. All right. So we are channelizing phytal, okay, into tocopherol synthesis. Okay. So I'll not go details here. I will go again talk about this. So we are again talk about phytal kinase. So from phytal phosphate will become phytal diphosphate, and phytal diphosphate react homogeneous acid to form tocopherols. They also say the vitamin K and all that. Okay. So one aspect. So we increase, therefore, okay, let's just skip this slide. So therefore, we increase the phytal kinase content in plants. Okay, in Brassica johnsia. Okay, by overexpressing it in Brassica, so that it produces a lot of phytals. So this is our probe VT5 on 35S promoter. Okay, so we can see that the green is VT5, right? Okay, 35S promoter, and precambia is the vector, and we got a transgenic plants. Okay, you can see we can see a lot of, you don't see here better the wild type and transgenic plants. I don't know why you don't see it better. Are you able to see from there? I'm not able to see from here very well. Okay, okay, yeah, from here you can see that. All right, and it's a south number to see that in two lines we have, in one line you have two inserts of the transgenics, others have singlet inserts of transgenics. So we transgene, so we took those are single insertion and did a real-time PCR analysis, RT-PCR analysis, and it's a relative gene protein expression. You can see the wild type plant phytal kinase and transgenic phytal kinase. Their concentration is here. So there are around 70%, 60 to 70% increase in the enzymatic, the enzymes, phytal kinase, or 80, or even sometimes 7 to 100% increase actually. Yeah, and this is the chlorophyll content. Okay, you can say chlorophyll content also increase. The wild type is always green or always blue, and transgenics are always red. There are two transgenic lines. Okay, you can see wild type is blue and transgenics are blue. Okay, so you can see the transgenics have more chlorophyll, and Yeah, yeah. Okay, good.
We're just changing the mic with the caller mic, so just bear with us for a 1 minute.
Okay. Okay. So you can see the vital content increased. The vital content increased in the transgenics. So increase the total tocopherol increased by twofold. Okay. So one increased by 50% and that became more than 100%. Okay. And alpha tocopherol also increased. Okay. And this is a stress condition. We applied stress to the plants. Salt stress here. Okay. 150 millimolar of salt and this is ultrastructure of thylakoid membranes. Okay. So you can see these are wild type control, BT5X control, wild type and transgenic. And you can see even after 150 millimolar stress condition, wild type thylakoid membranes highly disorganized, whereas thylakoid membranes are quite intact in the salt treated plants. And why this happened? Okay, not only that, these are all fluorescence, photosynthesis. Yeah, okay, this electron transport rate. Okay, electron transport rate you can see. Okay, here you can see, wild type. Wild type is blue always, and transgenic is red always. There are two controls. You can see increased photosynthetic efficiency in the plants having overexpressed the BT5. And not only that, under stress condition, you can see a wild type decline much more than the transgenic. And this is the theory behind it. Why this happened? Tetrapyrals of tetrapyrals with 4 pyrrole rings, chlorophylls or protochlorophyllites all are 4 pyrrole rings. Okay, they are the photosensitizers. They absorb the light. They go to excited state. When they're coming back to ground state, they're giving their energy to oxygen, which is becoming singlet oxygen. Okay, and this singlet oxygen is highly reactive. This singlet oxygen is highly reactive. Okay, and it destroys the plant, kills the plant. Okay, and therefore, it destroys the thylakoid membranes. And if there's a lot of singlet oxygen produced, plant become white actually, burns to white within 24 hours. Okay, this is a singlet generation, different parts of the cell of the leaf. Okay, this is thylakoid membrane. Okay, we have put photosystem 2, photosystem 1 where they operate, and all the chlorophyll bars and then intermediates. And there are a lot of places where the single oxygen could be produced in plants. Okay, this is actual demonstration that the single oxygen is produced less in transgenics. Actual demonstration. So what we did here, the wild type control plants, these are protoplasts actually. We isolated protoplasts, okay, from the wild type plants and from the transgenic plant, okay. Wild type plant and transgenic plant, protoplasts. Then what we did, we give protoplasts the stress, sodium chloride, applied sodium chloride, and expose them to light for a few minutes, okay. As you can see, this is red fluorescence, chlorophyll fluorescence, chlorophyll autofluorescence. And the green fluorescence is due to singlet oxygen fluorescence. Okay. Okay. So you can see these protoplasts, this is the wild type control, no salt here. Okay, control, there is no salt treatment. So you can see only you see the chlorophyll chlorophyll fluorescence. Okay. And we don't hardly see any fluorescence due to singlet oxygen, SOSG. SOSG is green fluorescence. We don't see any green fluorescence, hardly any green fluorescence. And we merge them together, you can see the same single oxygen fluorescence, red fluorescence or chlorophyll fluorescence. And when applied 200 millimolar sodium chloride to the protoplasts, what you see here, this is a lot of increase in chlorophyll fluorescence. Why? Because 200 millimolar sodium chloride, okay, inhibited photosynthesis. So the energy could not utilize in photosynthesis. Therefore, the chlorophylls, all the chlorophylls were in the excited state, and a lot of them came back to ground state, and therefore they're becoming more fluorescent. Not only that, they also produced singlet oxygen when it came back to ground state. Okay, so the chlorophyll fluorescence increased, and the singlet oxygen fluorescence increased because the photosynthesis are destroyed. Okay, so those energy cannot utilize in photosynthesis in 200 millimolar NaCl. Therefore, the chlorophyll went to high excited state. When they came back to ground state, they produced singlet oxygen. The singlet oxygen increased. And then this is a BTX5 plant. BTX5X plants means transgenic plants overexpressing phytal kinase, which, as I showed you, they're producing 100% increase in the tocopherol content. This, in a transgenic, they have a lot more tocopherols than wild type. And these tocopherols present in thylakoid membranes, they could absorb the quench singlet oxygen. They absorb singlet oxygen, destroy singlet oxygen. Okay. So they send singlet oxygen quenched actually. Okay. We call quenching. Okay. Singlet quenched and utilized by the tocopherol, destroyed by the singlet oxygen, by the tocopherols. At that point, you can see under identical conditions, okay, this bottom, of course, this is BTX5X plants again, control, okay, no salt, transgenics, no salt. Therefore, you don't see much singlet oxygen anyway, but you can see in 200 millimolar NaCl, we see singlet oxygen production, but very minimal singlet oxygen production in the transgenics, okay, and we merge them also here. Okay, so what it really demonstrates to you visually here that we are successful in minimizing the singlet oxygen production in plants, okay, by by tocopherols that are produced in the plants, okay, in response to stress. Okay, okay, and this is actual quantitative measurement of singlet oxygen. Okay, we saw visual measurement, the actual quantitative measurement singlet oxygen. Okay, wild type and transgenics, as I told you, wild type, no singlet oxygen. Okay, and once you give salt, you can see it has increased. Okay, and but in BTX5 plants, okay, there are two wild type control. This is BTX, this is wild type plant. You can see in wild type is always blue. Wild type plant, lot of singlet oxygen production as it exposure to light. This is how many 20 minutes and 50 what 40 minutes, 30 minutes exposure, 40 minutes exposure to light, and you can see here that this is more and this is wild type is less, but transgenic is less. Okay, so in transgenics we are producing less singlet oxygen than the wild type in response to stress. And what does this example I'm telling you, these are MDF production, malondialdehyde production, membrane lipid peroxidation. Okay, and this membrane lipid peroxidation is less in transgenics than the wild type because singlet oxygen is reduced. Therefore, thylakoid membranes are protected. They are not degraded by the singlet oxygen, which is highly reactive. Therefore, you can see that 100 millimolar stress. You can see how much MDA is produced in the wild type and how less in the transgenics. Okay, this is forgot. Oh, these are actual plants exposed to wild type and 150 mol NaCl, 200 millimolar NaCl stress. You can see here that, yeah, these plants on the top, these are bit 5x plants. They are, of course, they're faster growing also. You can also compared to wild type, okay? And they accumulate a lot more biomass compared to wild type, okay? And you can see also, but under stress condition, you can see, these plants have not flowered yet, okay? Where flowering successfully arrested, okay? And these are transgenics. So flowering is arrested actually in 200 million SCF. And this electron transport rate, I will not talk this for, okay. And this is net carbon, this is important, CO2 assimilation rate. And CO2 assimilation rate, you can see, your wild type is, white, and BT5 is black. Okay, black lines are, all are black, but open circles are wild type, field circles are transgenics. Okay, so you can see here that transgenic plants are have better CO2 assimilation rate than the wild type because they got more chlorophylls also. Okay, and these better chlorophyll will be better utilized in photosynthesis. Okay, and not only that, the photosynthesis and they are resistant to stress conditions. And the quantum yield of photosynthesis also decline in the wild type. Transgenic decline was much less. And starch accumulation, since photosynthesis declined, starch accumulation was also less in the transgenics, in the wild type than transgenics. This wild type is black, transgenic in gray here. Okay, so you can see here. And plant dry weight, this is important. Plant dry weight, wild type, transgenic, both are controlled. You can see transgenics had around 20% increase in the dry weight. But much more better increase in the stress conditions. Very important. Okay. So under stress condition is able to protect the plants. Okay. So that dry weight is much more than the wild type. Okay. Under stress conditions. And then also this yield. Yield is much better. Okay, per plant. Yield per plant here. Okay. So you can see control, of course, yield is better in transgenics. Okay, but under stress condition, on top 200 millimeter stress on top, you can see seeds per number of weight per seeds per plant. You can see on 200 millimeter NaCl here, you can see the transgenics, which is gray, and wild type is black. You can always see that transgenics have much better yield in the wild type. So with this, I must say that there are many things I can talk, but I will not talk much because my time is up. So this again, one must I say this much that how the protochlorophylate or phototransfer to chlorophyllite here in plants. Okay, this is protochlorophylite. This is protochlorophylite. This is chlorophyllite. Okay, so protochlorophylate oxide reductase is the double bond reduction here to chlorophyllite, okay, in presence of light. And okay, with these words, I must say that if you manipulate chlorophyll biosynthesis and chloroplast development, okay, and also we can modulate photosynthesis, increase photosynthetic efficiency of the plants, and they will be more resistant to stress conditions. Okay. All right, with this, thank you very much for your kind attention.
Do we have any questions?
Yes.
Yes.
Yeah, yeah.
Microphone.
Hmm.
Nowadays the loss of water going on.
on conversion of C3 rice plants to C4. Okay, so sir, Japan, they started the work on that line. In ED Philippines, they also started the work on line. Sir, in your opinion, how much it is feasible and whether is it possible to convert the C3 plant to C4? Then regarding that point, again, another question. Sir, when I was in the Cotton Research Institute, I have tested that when the environment carbon dioxide increases, then in C3 plant, the photosynthetic rate goes up. Then why do we need C4 plants instead of C3 when there are lots of questions about the environmental pollution and increase of carbon dioxide concentration?
Right, a very good question. Okay, first question. Okay. Let us start from your last question first. Okay. Then we go up. Okay. Your last question is, when the CO2 content in the atmosphere increasing, yes, photosynthesis rate is increasing. There's no doubt about it. Okay. And it will increase if we give a proper fertilizers to the plants. In agroeconomic system, agronomic system, okay, agronomy, you can say agriculture ecosystem, when we are able to manipulate the fertilizer input to the plant, we have to give more fertilizers to the plant so that nitrogen content goes down, because what happens, high CO2, photosynthesis increases, okay, whereas, and it comes from carbon dioxide in atmosphere. CO2 is more in atmosphere now, so carbon dioxide will consume to carbohydrates, so photosynthesis rate will increase, dry matter will increase. All right, okay. Now nitrogen is coming from soil, nitrate, okay, and it is not increasing in the soil, okay. So nitrate has to come, becomes nitrite, then ammonium becomes amino acids, and amino acids become proteins. So nitrate in soil is not increasing, where CO2 in the atmosphere is increasing. So therefore, in a forest ecosystem, where we cannot manipulate fertilizers input, their carbohydrate content is going to increase. In all kinds of seeds, dry weight, we are compared to proteins. Because per dry matter, per gram dry matter, there are more carbohydrates and less proteins. But in agricultural ecosystem, we should be able to manipulate nitrogen in the soil or urea. We give actually a lot of fertilizers than the plant needs actually. Therefore, many, the Harian and all that, after giving lot of years of fertilizer application, their soil becomes sodic. Okay. So therefore, yes, if you increase the fertilizer input in the soil, there will be less, because why the protein content decreases, 50% of leaf protein is rubisco. Okay, remember that. In a leaf, 50% of leaf protein is rubisco. If protein content decreased by 10%, leaf protein, rubisco also decreased by 10%. Okay. Okay. If rubisco released by 10%, rate of photosynthesis will decrease. And number one. Number two, if CO2 concentration is more in the atmosphere, plant will say, hey, why should I say invest so much of my energy amino acids on one protein called rubisco? Because I have a high CO2 now. Okay. So there is no need for me to invest 50% amino acids on one protein called rubisco. Okay. Let me diversify. So rubisco will go down a little bit. But if we increase the nitrogen content, so photosynthesis will decrease. Actually. Not decrease, but photosynthesis not as much increase as you expect. But still there will increase photosynthesis in high CO2. Even if you do not manipulate, increase the nitrogen content. Okay. Now therefore, yes. But when you say that why we need C4 plants, okay, yes, high CO2 world, photosynthesis rate is going to increase, there's no doubt about it. Okay. But especially in agronomic ecosystem. Okay. But not only CO2 is going to increase, but temperature is also going to increase. Okay. That is the bottom point, is that temperature is also going to increase. And C4 plants are much better they can withstand high temperature because they evolved in low CO2 and high CO2 world, low CO2 world and high temperature world. When in 50 millions of years ago, C4 photosynthesis evolved and when temperature was very high, of Earth's temperature very high, and CO2 concentration very low. Okay. So in high and therefore C4 plant evolved in high temperature and low CO2 world. Okay. And therefore, and C3 plants evolved high CO2 world and low temperature world. Okay. So therefore, it is very important. Okay. So C4 plants can naturally withstand high temperature because they evolve in high temperature world. Okay. All right. So therefore, C3 plants, if you grow a plant, C3 plant at 10 degrees and a C4 plant at 10 degrees, the rate of photosynthesis of C3 plant is much higher than C4. At 10 degrees centigrade, C3 plants performs better than C4. Okay, plant. But at high temperature, 35 degrees centigrade, C4 plants will perform much better than C3 plants. Okay, because the temperature of the world is increasing, therefore it is important to have C4 rise. Number one. So therefore, I'm saying that C4 rise is important. Okay, because not only CO2 is increasing, but the temperature is also going to increase. Number one. Yes, so this was important. But whether we can, convert a C3 plant to C4, probably not. Because needs tinkering of hundreds of genes. And when tinkering hundreds of genes, means virtually you are creating a new species. Okay, and that is extremely difficult for man-made species. Okay, so probably we can increase the rate of photosynthesis by manipulating C4 genes to a certain extent. Manipulating C4 genes increase the photosynthesis rate little more, but probably we are not in a position to create a C4 plants, C4 rice by manipulating the gene concentration. We may get a little bit increased rate of photosynthesis, but that will never be a C4 plant. Thank you very much. You have to give me this. Huh?
Okay. So for the next sessions, please keep your questions noted. We have a specified time for the question and answer session. And for all of your information, Professor Vaishnav Charan Tripathi ji would be chairing the session pre-lunch. So I just got the information, sir, so I needed to say this. Anyways, moving ahead, now I would request Dr. Arup Kumar Mukherjee, Principal Scientist, ICAR National Rice Research Institute, Cuttack, India, to please join us on this podium and talk about the topic, doubling farmers' income, role of trichoderma in sustainable agriculture. Let's have a wonderful round of applause for the gentleman.
I don't think it's like that. It's okay. So, good evening, everybody. Respected Tripathi sir, Sodanda, and the a galaxy of scientists and also budding scientists, I am little bit afraid because, or you can say I'm little bit nervous because you know that after Sunil Gavaskar batting, no, if Dilip Doshi comes to bat, the situation is like that to me because after listening Tripathi sir, whom we have read during our studenthood, his work, and also along with Professor Prasanna Mohanty, who is no more, it is my turn now. Okay. I have tried to make my slides in a popular presentation way because I know that most of the audiences are students. So, my dear students and my respected professors as well as all the galaxy of scientists, today I initially thought that I will talk about host pathogen interaction. It's deciphering the mechanism. But then after seeing the major topic, I thought that let me change it a little bit and let it fit to the topic of the broad topic of the conference. And but first I should tell thanks to Dipanshu Babu because I know I am attached with this conference since beginning, and in single-handedly he has arranged just a grand symposium, and I wish a grand success to the symposium. Okay, now the question is that it's a very important question that nowadays, you know, that our honorable prime minister is telling every time that doubling the farmers' income. Okay, and many persons thought it is a myth, it is not a reality, and many persons are thinking that it is a joke. Okay, but my point of telling is that it is not neither a joke or not a myth, it is a reality. But question is that how we are approaching it is the main important question, that how we are approaching. Reason is that if you see, if you tell three blind persons to see the elephant, somebody will touch the trunk and will tell it is like a huge pipe, and somebody will touch the leg and tell it is like a pillar. No, it's not like that. It has to be integrated approach. You have to integrate all the approaches, and then definitely the farmer's income will be double. What we are doing, we are trying to break the yield barrier. Okay, but that is not the only approach. Why I'm telling, the breaking the yield or yield is a particular character of a plant where number of genes are involved. It is a QTL, number of QTLs are involved. So to break the yield barrier, it is very difficult. Okay, we can enhance the productivity, but we cannot go just to double the productivity. It's not a magic, because if we would have done it, if you see, I am involved in the All India Coordinated Rice Research Improvement Trial since last 8 years, and every year when a new variety is released, we are seeing that 5 to 10% increase in the productivity. Just imagine, I am telling last 8 years I'm involved. So if each 8 year, if I calculate the 10% increase in the productivity, by now we would have got 80% more in yield. Did we get that? No. Okay, because I don't want to discuss in detail why not. Okay, but we have to definitely increase the productivity. Then how it can be done and how we can enhance the productivity, or we can enhance the farmer's income. We can prevent the crop loss. You'll be surprised that at present, every year, the crop is lost in such a rate that 30 crore of people can be fed if we can prevent the loss. Okay. Sorry. Then we can reduce the input cost. That can be done because you know that now the direct seeded rice is there, where we are able to reduce the water input, and we can also nitrogen use efficient rice crop is there, where we can reduce the input of nitrogen. Then there are a number of microbes involved, where we can go for solubilization of soil phosphorus. We can enhance the uptake of nitrogen by that microorganisms. Then we have to adopt the latest technologies. Okay, there are many technologies now developed now present, and this technology does not mean weeder, or you can say that a direct seeder like that, and happy seeder is not like that. There are many technologies, and one of them is biotechnology. And I'm happy to see that my younger brother, as well as the brightest scientist, Dr. Kutub, is here, who can tell that what biotechnology role can play, the role of biotechnology can play to enhance the productivity as well as the quality of product produced. Then we can go for value addition. If we can add the value of the crop, then definitely the price of the crop will go high and the farmers will get higher income. Then you see that whatever waste is produced from the agriculture produce, it is a mess. Because if you think, as Tripathi sir is here, he might have witnessed, sir, in Delhi during October, November, December, most of the colleges and schools are closed. Why? You know, because the rice straw is burned in such a way that that entire Delhi becomes totally, they don't receive the sunlight. Okay, it is so polluted, and it is becomes horrible for the young kids as well as the aged persons. Okay, so that thing, if we can prevent, and if we can make that waste to wealth, that is another way. How it will be done, I will show one by one. Then proper marketing. You know that in West Bengal, I recently visited West Bengal. It is my native place. So, and I am a village born. I am my family. I go to field regularly. Whenever I go to my village, I spend almost all the day in my field to see what is the condition of the crop. You won't believe the farmers, what they are doing. They are feeding their produce to the cattle because they're not getting market. Just cabbage, I don't know how much price you're paying in Bhubaneswar market, maybe 20 rupees or 30 rupees cost per kg. But there, in 10 rupees, you will get 15 cabbages. Just imagine the precarious condition of the farmers. Okay, so proper marketing is another important. And if you can combine or integrate all those things, then you are definitely you're enhancing the income of the farmers as well as the agriculture will be sustainable. Okay, because when you are reducing the input cost, means somewhere you are modifying or you are not wasting your water, nitrogen and fertilizer, everything. So, okay, in that case, your environment becomes safe. Okay, and then the farmers will be happy. Okay, then let me talk about start with my topic. I'm not going to tell about sustainable agriculture, what it is. Then my topic, I am coming directly that how trichoderma can be helpful for enhancing farmers' income and sustainable agriculture. Trichoderma is a wonderful fungus. It can reduce the load of pesticides, and also it can reduce the water use, then reduce the use of chemical fertilizer. Proper management, it can manage properly the agricultural wastage. Okay, and it can maintain the soil health by adding organic carbon to soil, and it can enhance the yield even up to 25%. Okay, and as a result of all, you can see that agriculture becomes sustainable. Then trichoderma is a very good biopesticide. I'm not going to tell about details about that. I'll show in subsequent slides and how trichoderma works as a biofertilizer or biopesticides. This is the schematic diagram. Okay. Then how it works as a biocontrol agent? It works in biocontrol agent as a mycoparasitism. That is, it penetrates the pathogen's hypha directly, and it uptakes the nutrients from the pathogen inside the pathogen mycelium. Then it can secrete some number of antibiotics which prevents the pathogen growth. Then it competes for the nutrition or space, that it induces resistance, inactivation of the pathogen's enzymes. And you can see what I was telling here. It is seen here. Is it visible? I don't know. I'm not able to see. You can see here. This is done. This work has been done by one of my student. Again, she is from this university, Santipurva. She's work still continuing. You can see here. This is the mycelia of Helminthospora oryzae, and this trichoderma is supercoiling this mycelia, Helminthospora oryzae, and it penetrates its inside. And you can see these are the penetration point, and it uptakes the nutrient from your mycelia of Helminthospora oryzae. Okay, then this is one work I initiated in Nagpur when I was there. We have identified this pathogen was identified for the first time by me. So we wanted to search a solution for that, how to manage it. Then you can see here what we did. This Sclerotium delphini, this pathogen could not allow many seeds to germinate. Okay, and only you can see 2 good seedlings are there. That also not good are there. And our candidate one, the THG, you can see 90% seeds have been germinated here in spite of it being a sick soil. It is having the same pathogen. Okay, so that we have made a formulation that is known as trichocas, and then that we have tested in consecutive 3 times in coordinated trial, and it was successful, and it also enhanced the yield. So now it is going to be sold as a product. Then when I was transferred to rice, then I started my work on rice itself, and the then director, Dr. Tiroshan Mahapatra sir, who is now DG, he told me that Arup, you have to file a patent and you have to show that how we can control the Rhizoctonia solani, a menace for the crop plant, rice crop. So I took up the challenge. Then I tried to isolate trichoderma from soil, not soil. My beauty is that whatever trichoderma formulation is available in the market, you know that at present 250 industries are running on trichoderma in India itself, and crores of business are going on. Okay, and all the trichoderma have been isolated from soil, but none of my trichoderma is from soil. They are from either naturally mycoparasite that they are growing on bracket fungi, and then or they are growing on tree bark. Okay, so I have isolated them. I have tested them for their efficacy, and of course I have characterized them by taking molecular markers for identification. And then I have tested that efficacy. They have identified in as different species. You can see in the table. And then we have tested for their efficacy, and you can see here they are preventing the growth of the pathogen. This is dual culture assay. This is called, and you can see this growth inhibition is presented here as in a tabular form, as well as I think it is not working properly. Yeah, okay, and also in the graphical form. Then we wanted to test that whether this trichoderma is having any adverse effect on the germination or not. If there is no adverse effect, and then we tested in the pot, and we got excellent result. Then this is the first testing against seed blight pathogen. This is the control. You can see the growth of the seed blight disease. But here, when we treated these seeds with trichoderma, we did not get or very less development of the disease is there. Okay. Sir? I'm not able to see anything from here. You can see here, so this is the disease development. These are little length here in the control. Okay. But you see here, it is treated. You can see that this is only this much. And here, almost it is not there. This is the disease symptom. Supply. Okay, then you can see that graphically represented how much disease progress is there when we treated with trichoderma. So this is control. Sorry. So you can see this is the reason or disease development in the control. And when we treated with trichoderma, this one and that one, you can see the competitive development. Then this is one of my student, again he is from Robinsa University. Hare Krishna is there. He will present in detail what we did, sir. In conidia, when we make formulation, that what happens, it remains active for 6 months. So if we can induce chlamydospore, then chlamydospore remains active even more than 1 year. So, but problem with chlamydospore is that when we apply into the plant, it takes little bit time to get activated. But even then, we have tested, it is working excellently in competition of the with the conidia. And so this is the disease plan or control, the disease development, and you can see here when we use the chlamydospore, conidia, and other. And this detail will be presented by Hare Krishna. He is here. Then, sir, one problem is with that rice plant is that we have 2 severe diseases. One is brown spot, one is seed blight. You can see the development. We have taken 2 varieties. One is corona, one is sawarida. You can see here the disease development here. in control. Whereas when we tested with our trichoderma treated, you can see here that this is the growth. Then, sir, we have this is about the control of the pathogen. So that means if we can give seed treatment with trichoderma, we can reduce the pesticide load. And so number of farmers have tested, and there is a huge demand of it. I'll come later on. Now I'm coming to the how it can promote the growth of the plant, and we can realize higher yield. You can see here, sir, germination time is reduced when we test with we treat with trichoderma. This is control, and these are different trichoderma species. And sir, finally we have to field in the test in the field, and you can see, sir, this is the control, and these are treated with trichoderma. You see the plant canopy. These are the density of the plant canopy. You can see. Sir, this one is the control. There's no trichoderma, and these are treated with trichoderma. This is another view again. This is the control, and these are treated with trichoderma. Then, sir, we have taken the fresh weight and dry weight. You can see the root growth in control one in comparison with the treated one. Our two candidates trichoderma species which we have made the formulation, and we have also applied for patent, and patent has been granted recently. You can see further, and this is the seedling vigor index. The seedling vigor index in T2 and T5 is much better in comparison with control and other species. And so this is that different agronomical traits we have tested, and you can see here these are our two candidates. They are having much better agronomical indicators than in comparison with control and other species. This is an NPK content of the rice plants. When we treated with trichoderma, you can see here NPK content is higher when in the root and shoot in comparison with the control. Then, sir, this one we tested in the farmers' field in Ganeshpur. One village is there in Kendrapada district, and I don't know whether it is visible or not. So this side, we do not treat with this side, we do not treat with trichoderma, and this side we treated with trichoderma. You can see the fluorescence and everything. What is it?
Where is cordless?
Cordless one. So, this one, Dr. Himanshu Pathak, who was the director that time, he visited the field himself, and he has seen, and he has seen in the field, and you can see here, sir himself is testing in the checking it whether I am telling the truth or not in the field, and he was highly impressed. And this one farmer from Jajpur, and so this is treated with trichoderma, and this is control. You see the difference in the root mass as well as the total biomass of the plant. Then, sir, our another problem was there. Again, I'm telling that it has to be sustainable. So, if we increase the load of a particular microorganism in the soil, then that may hamper the native microorganism population. So we tested that also, and we did not get any differences in native microorganism when we applied trichoderma external. Then, sir, you can see the yield component. Yield component, this is control, and these are our two trichoderma. You can see here in two different varieties, it is completely much higher yield we are getting. Then this is when we compare with the different trichoderma species. And, sir, this one again, the farmers from different farmers from Kendrapada. This person is known as Sukhdev Lenka. He is a regular user of our trichoderma. This has been used in black gram. Sir, you can see here, this is treated with trichoderma, and that backside is not treated. When I just taken a focus on photograph, you can see here the canopy density is much less here because this black gram is having one serious disease that is known as seedling wilt. Okay, and the seedling wilt, when even they eat also seed, and when it germinates, it kills completely the seedling. That's why you can see here the canopy density here when it is controlled, and you see just canopy density here. And that person, he regularly uses my formulation for rice and other crops also. And sir, this one is in Niger, and this has been conducted by a farmer in Koraput district. Okay, and you can see here, sir, this is control, and this is the treated. Then, sir, this is a small millet. You can see here, this is control at vegetative stage, and this is treated at vegetative stage at the same age. And you can see in the reproductive stage also, this is control and this is treated. You see the differences. Sir? Yes, sir. And this is maize, and this test is going on in Uttar Bengal Krishi Vishwavidyalaya by a farmer in Kuch Bihar. You can see here the 55 days old plant maize. He has sent the photogram recently, and this is zero tillage. Sir, just imagine this is zero tillage. Again, I am telling that cost is reduced. How cost is reduced? I am not going for tilling, so it is zero tillage. And you see, this is control and this is treated with trichoderma. See the plant condition. And sir, that part is plant growth promotion I told. Now I am telling for induction of drought. And this experiment, I got this result accidentally, because there are 7 days continuous holidays. So nobody was there to water the plants. And when the institute reopened, we saw that in control, the plants are totally wilted. And this is, again, I'm telling, this is not my area. One physiologist is involved, Dr. Kosik Chakraborty and Dr. Sarkar, as long as he was here, he was involved in this experiment. This control was completely wilted, and you can see the condition of this plant, this spot, and this spot. Okay. Then you can see the condition of the soil. Soil is completely cracked. You can see here the plants remain green. And after that, what we did, we make the micro plot. And you can see here, this is treated with trichoderma, this is control. Now, brown spot is a disease which is associated with drought. Means if drought is there, brown spot will be very severe. Okay. And you can see here, this incidence in the control and incidence when we treated with trichoderma. Okay. And then yield also per meter square. This is absolute control. Absolute control means we have supplied everything in normal way. And this is the control, means exposed with drought, then rewatered. And you see it's just compare, and these are treated with trichoderma. Consider the yield loss here. And then with the help of Madam Padmini Soy, we tested it again in the field condition that I show the photograph. And sir, you can see there, the reason why we got drought tolerance, probably the enhancement of the root growth. You see, this is the root of the treated plant. Okay. And you can see here, this is a drought tolerant variety. This was identified by Madam Padmini Sai again. And this is a drought susceptible variety. Just imagine, this is the control root and this is a treated root. I have never seen in my life the rice root so long. Okay. And you can see here also. And you can see here the root biomass. Then, sir, we went to the field in Madam. This experiment was done by Madam. And you can see here, so this is the control, no trichoderma, and this is with trichoderma. Okay. And when we rewatered, we got only 15% yield loss when we treated with trichoderma, whereas almost 90% yield loss when we did not treat with trichoderma. Okay, sir. Now, this is about drought induction. That work is going on. We have already done in detail, and regarding biotechnology work, already Dr. Kutubi is here. He is associated with that work also. We are going for transcriptome assay. Then other works also will be doing. Now I am going to rice straw management. You should know that rice straw, as I told, it has become menace. Madam, I have time. How much time I have? Okay. So rice straw is become menace in the cropping season, and I told that almost entire Delhi becomes standstill. Okay. So we use our trichoderma. We identified. We characterize from different straw degrading enzymes. Okay. Then after that, What we did, we treated our trichoderma, rice straw with trichoderma. This is the control. These are treated with different trichoderma. We got 55 to 60 days, it takes time, but the farmers need within 30 days it should be. But we are not able to get that. Let me confess, we are getting around 55 to 60 days to decompose. Then we studied the enzymatic index, and then we went for the effect of that trichoderma assisted decomposed in the rice, and we have seen that it is again enhancing the plant growth. It is increasing the plant growth. That means it is applying some extra nutrient. And I just keep that slide that Rupela, who was a scientist in ICRISAT, he has shown that we are losing some $1 million or rupees by burning the rice straw. Because the rice straw, it only does not have the cellulose content, but it is having number of nutrients content in it. So if, and as well as organic carbon. So if we can add that thing to the soil, we are preventing the environment to be polluted, as well as we are adding nutrient to the soil. So sir, there are a little bit of physiological works I wanted to do, but let me tell you sir, whatever work I presented, none are my works. It is the students, and fortunately I have got 3 excellent students from Robinson University who have realized my idea to the reality. And this work was done by Sosmita. She will be presenting in detail also sir in subsequent presentation. Then little bit of science we tried, and then sir, what we did, we have identified a new trichoderma as a biocontrol agent. We went for whole genome sequencing, and we got some beautiful information. I'm not going to tell in detail. Some new genes also we have got. Then this is the different enzymatic very expression classes, how much percentage they are there inside it, the number of genes are present in this trichoderma species. And this is based on RNA polymerase, how I identified my trichoderma species as a new one. Then transcriptomic assay, we have got some novel genes that we are working on it. Again, as I told sir, I am not an expert, so I need help from Dr. Kutub and Dr. Kaushik and others are there. They are helping me in this work. So chlorophyll content, again, as I told, it varies when we treated with trichoderma, that Sosmita will tell in detail. And sir, this is different enzymes expression when we use real-time PCR. Using real-time PCR, how they are varying different enzyme expression. And we have seen that it is much higher in treated with trichoderma than in the control. So these are some works we are doing in the farmers field. We are training the farmers, because our director is there, Dr. Dean Director, Dr. Himanshu Pathak, he was there. And then we conducted some training also here. And sir, this is the patent have been awarded recently. And sir, I should tell that what I was telling, that if trichoderma being a single species, you can see it can reduce the pesticide load. It can reduce the number application of nitrogen fertilizer. Okay, it can reduce the water input. Okay, and then we can use the waste in a proper manner. Okay, so if we use all this integrated approach, I'm talking about only one trichoderma, one microorganism, but there are a number of microorganisms which can help in other ways. Okay, so thank you, sir. Thank you for giving me the opportunity. Thank you very much. But let me tell you, sir, these works are, again, they are not, these are the few papers we have published in different high impact journals. So these are not my works. These are works of my students who are here. So thank you very much, sir. Thank you for giving me the opportunity.
So we'll be giving you the time for question and answer post the keynote session, which is going to commence after 20 minutes of a tea break. So now you may please have your tea, snacks, and then come back again sharp at 12, because we'll start the keynote session at 12 sharp. Thank you, Sach. check. 12টা, sharp 12. बाकी लोग को घरे खाइबे अब. So if you are done with your tea and snacks, please join us inside the hall. We'll start the session in about 5 minutes more. Please join us inside the hall. We are about to begin the session. These session, okay, this session pre-lunch, it is chaired by Professor Vaishnav Charan Tripathi. As we have already mentioned that earlier. Each session for now would be concluded within 20 minutes. This is a humble request from our side to all the professors.
Hello. Se kଣ?
One minute. Yeah. So it's a humble request that we'll confine our sessions to 20 minutes and not 25 minutes further. The first session, the first keynote session for now would be on the topic valorization of leaf meal for replacement of DORB, de-oiled rice bran, a forwarding approach for sustainable aquaculture by Dr. Narottam P. Sahu, Joint Director, ICAR CIFE, Mumbai, India. He'll be joining us in a short while.
So good afternoon, everybody. Am I audible? Hello. Am I audible to everybody? Hello. 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 Am I audible? Hello. 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 कौन हो जाता है? कई जाने पड़ते हैं responding. Hello. Hello. Am I audible? Hello. अरे केही रेस्पॉन्ड करू नांती मो ऑडिबल को नाही केही कहू नांती मो स्क्रीन शेयर तो करिछि ना केही रेस्पॉन्ड करू नांती मो वॉइस जाउत ना नाही but there is no more responding. देखो स्क्रीन आस्ते ना स्क्रीन ताल they should respond ना हेलो अरे केही रेस्पॉन्ड करू नांती मो ऑडिबल को नाही केही कहू नांती मो स्क्रीन शेयर तो करिछि हां सो shall I start? Okay केही रेस्पॉन्ड करू नांती मो
Good afternoon, sir. Hello.
yes good
Good
afternoon.
afternoon, sir. This is actually, I'm also one of the participant. I'm Dr. Yashasvi Naik from
Okay.
Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us.
Okay. Technical
But I am able to hear you. I have also joined online, sir.
Screen is visible. Screen.
Yes.
I don't know. So shall I go ahead?
Good afternoon, sir. Hello.
Yeah. Good afternoon.
Good afternoon, sir. This is actually, I'm also one of the participant. I'm Dr. Yashasvi Naik from
Okay.
Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us.
Okay.
But I am able to hear you. I have also joined online, sir.
But I think everything is echo.
Yes.
I don't know. So shall I go ahead?
Good afternoon, sir. Hello.
Yeah. Good afternoon.
Good afternoon, sir. This is actually, I'm also one of the participant. I'm Dr. Yashasvi Naik from
Okay.
Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us.
Some technical problem.
But I am able to hear you. I have also joined online, sir.
Everything is echo.
Yes.
I don't know. So shall I go ahead?
Good afternoon, sir. Hello.
Yeah.
Good afternoon, sir. This is actually, I'm also one of the participants. I'm Dr. Yashasvi Naik from
Okay.
Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us.
Some technical problem.
But I am able to hear you. I have also joined online, sir.
Everything is echo.
Yes.
I don't know. So shall I go ahead?
Good afternoon, sir. Hello.
Yeah.
Good afternoon, sir. This is actually, I'm also one of the participants. I'm Dr. Yashasvi Naik from
Okay.
Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us.
Some technical problem.
But I am able to hear you. I have also joined online, sir.
Everything is echo.
Yes.
I don't know. So shall I go ahead?
Good afternoon, sir. Hello.
Yeah.
Good afternoon, sir. This is actually, I'm also one of the participants. I'm Dr. Yashasvi Naik from Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us.
Some technical problem.
But I am able to hear you. I have also joined online, sir.
Everything is echo.
Yes.
I don't know. So shall I go ahead?
Good afternoon, sir. Hello.
Yeah.
Good afternoon, sir. This is actually, I'm also one of the participants. I'm Dr. Yashasvi Naik from Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us.
Some technical problem.
But I am able to hear you. I have also joined online, sir.
Everything is echo.
Yes.
I don't know. So shall I go ahead?
Good afternoon, sir. Hello.
Yeah.
Good afternoon, sir. This is actually, I'm also one of the participants. I'm Dr. Yashasvi Naik from Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us.
Some technical problem.
But I am able to hear you. I have also joined online, sir.
Everything is echo.
Yes.
I don't know. So shall I go ahead?
Good afternoon, sir. This is actually, I'm also one of the participants. I'm Dr. Yashasvi Naik from Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us. But I am able to hear you. I have also joined online, sir.
Everything is echo.
Yes.
I don't know. So shall I go ahead?
Good afternoon, sir. I'm also one of the participants. I'm Dr. Yashasvi Naik from Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us. But I am able to hear you. I have also joined online, sir.
Everything is echo.
Yes.
I don't know. So shall I go ahead?
Good afternoon, sir. I'm also one of the participants. I'm Dr. Yashasvi Naik from Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us. But I am able to hear you. I have also joined online, sir.
Everything is echo.
Yes.
I don't know. So shall I go ahead?
Good afternoon, sir. I'm also one of the participants. I'm Dr. Yashasvi Naik from Centurion University. I have also joined online. I can hear you, sir, but I don't know what has happened with the main stage. I think they are not able to hear us.
Next speaker, rating presentation.
But I am able to hear you. I have also joined online.
এটা কার
या पूर्व
I I'm going to when I was getting ready to I'm going one like I think but I don't know if I don't do they are
ల్యాప్టాప్ లాగిపోయా పోరస్ వస్తుంది
I'm going to when I was getting ready to I'm going one like I think but I don't know if I don't do
ల్యాప్టాప్ లాగిపోయా పోరస్ వస్తుంది
I'm going to when I was getting ready to I'm going one like I think but I don't know if I don't do
ల్యాప్టాప్ లాగిపోయా పోరస్
I'm going to when I was getting ready to I'm going one like I think but I don't know if I don't do
ల్యాప్టాప్ లాగిపోయా పోరస్
I'm going to when I was getting ready to I'm going one like I think but I don't know if I don't do
ల్యాప్టాప్ లాగిపోయా పోరస్
I'm going to when I was getting ready to I'm going one like I think but I don't know if I don't do
లాగిపోయా పోరస్ లాగిపోయా
Now I am audible.
Yes, yes, sir, you are audible.
But there's too much background sound is coming. Please allow me to share my slide. I'm audible. Yeah,
Yes.
audible. But that background sound still is coming.
So you're audible?
Yeah, too much background sound is coming. Please allow me to share my slide. But again, what I'm telling, the same thing is echoing.
All the other speakers should be on the mute. Only the speaker should be with the open mic. We will
I'm
now.
audible. Yeah,
Yes.
audible. But request all the audience to mute so that the background sound will stop. Background sound is coming. Please allow me to share my slide. But again, what I'm telling, the same thing is echoing.
All the other speakers should be on the mute. Only the speaker should be. We will
I'm
now.
audible. Yeah, audible. But the audience to mute so that the background sound will stop. Background sound is coming. Please allow me to share my slide. But again, what I'm telling, the same thing is echoing.
Sir, if you can please shut the door where you're in the room where you're sitting. If you can please shut the door.
I think somebody they are having joining with the two system. That's why too much echo is there. So now this screen is visible. Screen is visible. Hello. Am I audible now?
Yes, sir, you're audible.
Screen is also visible. Screen. I
Yes.
have shared the screen.
Yeah.
Is visible now?
Yes, sir, it's visible.
Okay. So shall I go ahead?
Yes, sir.
Okay. So good afternoon to everybody, respected chair of the sessions, the rapporteurs, and all the delegates present here. I'm sorry, there is some technical hitch was there for which that took some time. But anyway, 20 minutes given, I will finish. So now the screen is visible. Again, the same thing. What I have told, it is again echoing. Screen is visible. Hello. Am I audible?
Yes, sir, you're audible.
I'm same thing. What I have told, the same thing is going here. Whether you are comfortable, is visible now.
Yes, sir, it's visible.
Okay, so shall I go ahead?
Yes, sir.
Okay. So good afternoon to everybody, respected chair of the sessions, the rapporteurs, and all the delegates present here. I'm sorry, there is some technical hitch was there for which that took some time. But anyway, 20 minutes given, I will finish. So now the screen is visible. Again, the same thing. What I have told, it is again echoing. Screen is visible. Anyway. So
Hello.
Shall I start?
Yes, sir, you're audible.
I heard same thing. What I have told, the same thing is going here. Whether you are comfortable, is visible now.
Yes, sir, it's visible.
Okay, so shall I go ahead? Everybody, respected chair of the sessions, the rapporteurs, and all the delegates present here. I'm sorry, there is some technical hitch was there for which that took some time. But anyway, 20 minutes given, I will finish. So now the screen is visible. Anyway, so again, good afternoon, everybody. I still some problems persist. Visible. Anyway, I have to go ahead. The topic given to me, that's the valorization of leaf mill for replacing DORB, a forwarding approach for sustainable aquaculture. In short, I can say this is made for just like replacing the de-oiled rice bran, which is a major source used in the aquafeed, while leaf mill. The leaf mill is now not used in fish feed. So I will give why and how we can use the fish feed.
The chair of the sessions, the rapporteurs and all the delegates present there.
So before going, I'll just the audience want to inform the audience. The feed contains many ingredients. Maybe starting from at least 10, 12 to maybe 20, 25 based on the formulations, based on the different companies, they put some different ingredients. So the feed contains many ingredients. And if you see the feeding practice in aquaculture, and still the people used to, by traditional method of giving this rice brans by either, I can say, broadcasting in the pond, but only 3 to 4% the farmers, they are using the pelleted feed, because as you know that aquaculture is in demand and more fish is in demand. And so aquaculture is moving toward feed-based aquaculture, but still we are in India, we are using the rice bran. So there is major consumption of the rice bran is there. Before going, the peculiarity is that the rice bran, it is shared by all animal production sectors, including the steam, poultry, animals, including the fish. So when you are using these ingredients, it will be shared by all the sectors. Whenever we are planning or we are projecting to increase the fish production or aquaculture production to double, obviously there will be a basic demand of double requirement of all ingredients, including DORB. Why I am putting DORB here? Because there is maximum requirement of this DORB. Because as you know that aquaculture, this DORB, if it contains at least 50-60%, it costs the major expenditures. So now in future, if you see the production status of paddy, then you can know that why it needs to be replaced. So in our research of last 5 years, we want to replace this fish meal, the leaf, with the DORB. Why DORB used? If you see the Indian map, India is mostly the paddy-based countries. Every state they are producing paddy except one, the Rajasthan. So that's why every household or every farmers, the rice bran will be available with them. That is the reason why they are using this rice bran as a major ingredients for the livestock production, including fish. As I told, if you see the productions of this paddy, there is almost static production. Static productions. Means contains at least there is very little bit increase in paddy production every year. So if you are asking or if you are expecting that we want to double the aquaculture production, so we want double our research of DORB, rice bran. So if the paddy production is increasing, not increasing to the tune of double, so we cannot expect a double amount of rice bran will be available in near future, so as to support the sustainable aquaculture productions. So every household. So if you will compare the DORB, you see the DORB means de-oiled rice bran. That means after the extraction of the oil, the remaining mass is called the DORB, which contains 14% protein. Commercial carb feed, it contains 20% protein. The question comes whether we can develop a feed rather only based on the DORB, which can support 20% protein originally used as a commercial feed. So if we have done some of our research approach, how best we can use the DORB or de-oiled rice bran as rice bran will be available in near future. So this rice bran contains, though it contains 14% protein, but the quality of the protein is not that good. So it needs some treatments for which better nutrient utilization will be there from the rice bran. Instead of using rice bran as such, we are going to which contains treated rice bran. So these are some methods or the approaches I have given. One is the solid state fermentation. The solid state fermentation can be done either by the fungus or some selected bacteria are there, or you can also mix. Only beside this also we can go for addition of exogenous enzymes. Because you know the rice bran contains more fibers, it is not suitable for monogastric animal like fish. Beside this, some deficient nutrient like this essential amino acids like glycine, essential fatty acids, that can be supplemented, so that finally this rice bran, the nutrient utilization from rice bran will be better. I have given the after fermentation of DORB, after 96 hour fermentation with rice bran, instead of selected fungus and bacteria, the protein content increase almost to twofold after 72 hours of fermentation.
The approaches I have given, one is
14% proteins, it will become around 20 to 24% proteins. That is due to accumulation of the microbial proteins.
are there, or it will be also.
Interestingly, if you find the essential amino acid which is mostly lacking, that is lysine, the increases to at least 5 times more in case of fermented DORB. Now, after this, we have done made economical calculations
That can be supplemented.
that based on that 4-5 feed, we compare with the commercial feed, that is C is control first one, that is the commercial feed.
After fermentation of
T1 is the only feeding with DORB. T2 that is fed with fermented DORB. Then T3 that is DORB with enzyme supplementations. Then T4 it's supplemented with fermented DORB with enzymes, with amino acids, with fatty acids. Then the last one, the enzyme source is a liquid enzyme. So you will find always supplementation with enzyme, rather the liquid supplementation of enzyme was better compared to DORB. But DORB, when the farmers, mostly farmers, they are using this DORB because they don't know how to use it, or they don't know aware of this use of these enzymes. So simply use of these enzymes will manifold increase the utilization of DORB.
Control first one. That is
Now we have found that if you want to use a DORB based diet, if you are giving 26% protein with 7% lipid, that will be the best. Now coming to the leaf, as I told that now the DORB means the paddy will not be available to support the matching demand of doubling the aquaculture productions. So we have to think for some alternatives.
Last
We have
one.
seen that many leaves available around us are not used for any purpose other than search for some fuel purpose.
Enzyme.
We have selected some leaves which contain more than 20% proteins. That is, I have given some names, that is groundnut leaf, otherwise known as subabul. That is moong meal. Then Gokul Kanta is a weed. Then some menthas. Mentha, that is corn mint. It is plentifully available, or it is available only due to an industrial byproduct. It's truckload of these byproducts are available without any use. It contains 20% proteins. Ipamia, it is also weed. Then lemongrass, also it is an industrial byproduct. Then siam weed, barsim. Then cauliflower, that leaf. These are some of the weeds, but contains a very fair amount of proteins. Green pea, then arhar or soybean meal, soybean leaf meal. These are some leaf meal I have given, for example, which contains more than 20% proteins. That is, I have given some names as groundnut leaf. Now we have conducted some of the feeding trials of the 6 leaves, where we have found that rice bran can be replaced either 100% or partially it can be replaced. Example are, which is groundnut leaf, which can complete 100% replace this. The mentha, which only can replace 50%, because maximum 30% we are using in the feed, so 15% can be included, 50% replacing. But this black moong leaf, it can replace completely the DORB, but 20% inclusion is better than DORB. So similarly, this Ipamia weed, it can 10% we can include. Mentha also, partially we can use it. And then one more weed, that is Gokul Kanta. It's mostly available in the Odisha. 100% we can replace it. Now I will give some example of the leaf meal. That's a Gokul Kanta or Hygrophila spinosa. This is a weed. It's never been used for any other purpose. So we found that it contains fair amount of proteins. So when we are using in the fish feed, only after using this weed along with some enzymes, this result was at par with the commercial feed. So can you imagine this leaf, which has never been used for any other purpose, when you are using along with some of the enzymes, it can be completely replaced with URB. Similarly, the moong. We can use ground. One more black gram leaf. When we have tried 10, 20, 30% replacement, 30% is the maximum, or we can say 100% replacement. You see, I can replace 100%, but at a 20% inclusion level, it's better. Hygrophila spinosa. This is a weed. It's never similarly. We have introduced one more strategy, that if some weed or a leaf cannot be used daily, if it can alternately use, then at least the utilization will be more. That also we found it successful. when we are giving, you see in T4, it is 3A means the control diet, and 3B means this wheat-based diet, black gram-based leaf meal diet. Now, just one more. We did a control diet, 3B this leaf meal-based diet. It was better than, it was at par with the control. You see, now coming to the groundnut leaf meal, it is a very good leaf meal. It could successfully replace URB. Rather, this 30% replacement, the growth rate was much better than the control. We have introduced one more strategy, the groundnut leaf meal. It is far better than our. If you can alternately use, then I have given purposefully this leaf meal, that is a lemongrass, because the lemongrass oil, after extraction of this oil which has been commercial use, it is lots of this biomass available without any use. So it contains very much high fiber. When we have used this fiber, of course, along with the addition of some enzymes, you see, it is better than, it is at par with control. So this type of biomass which has never been used before, now along with these enzymes can be used. Rather, this now Ipomia, it is aquatic wheat, it is maintenance everywhere. Now this meal also it can be used. You see, at a 10% nitrogen level, the growth rate is better than this control group, DORB based diet or rice bran based diet. But when we are giving at 20%, it is as par with the control, but 30% nitrogen level, growth rate was reduced. Very much. Now, one more is a very good leaf meal that is called green pea leaf meals. Green pea leaf meal also it contains, you see, continuously whenever inclusion level increases, growth rate decreases. But whenever you are using some enzymes, again the growth rate was augmented. So this leaf meal, when we are giving some enzymes, the growth rate is compensated. It is another lab is Kajanas or leaf meal. It is also very good leaf meals. You see, at 30% inclusion level, the growth rate are better than the control. Growth rate is this leaf meal is better than the DORB. Another is soybean leaf meal. The soybean leaf meal, you know, that is when we are using as such without this enzyme, the growth rate was not comparable with control, but when we are giving along with this enzyme, the growth rate was comparable with the control. One more is a very good leaf meal that is called green pea leaf. Now, Sesbania leaf meal, it is you will find everywhere. This meal it contains some of the mycin or antinuclear factor, but it contains 28% protein. Again, you just can't overlook this type of huge biomass available, which contains high protein. We have tried, we have found that many saffron and phytic acid, these are the anti-nutrition factors present in this leaf meal. But when you are using this with solid state fermentation, after fermentation of this leaf meal, it will be able to better than the growth rate at par with the control. Otherwise, you know that when you are increasing the growth rate 15%, 30%, the growth rate was drastically reduced. Only after this treatment of solid state fermentations, it could be suitable for using the fish. Similarly, one is that is Bursim leaf meal. Bursim also contains good amount of proteins. You know, when we are giving it by replacing 50%, the growth rate was better than the rice. Can't overlook this type of huge biome. Another approach is that a mixed leaf meal. Then generally it is asked that one may not get a sufficient amount of leaf meal in one locality. So a mixed leaf will be an ideal approach because it will be plentifully available. That's why you have taken this approach by mixing 3 leaves, that is better than growth rate as par with the control. Otherwise, like increasing the growth rate, then Ipomia and one more weed. So 3 different type of this leaf we have used 1:1:1 ratio, and we found the mixed leaf also it is the growth rate of 20% inclusion level. It was even also 40% inclusion level, while addition of some enzymes and was better deficient amino acids, the growth rate was far better than the control. Another approach is from LM40 to LM40E. So there is a significant difference. So that's why we have developed some of this fermenter mixers at low cost by using a drum only. We have used this one. This approach by any leaf meal or rice bran can be put in 20 kg rice bran can put here, and one suitable culture will be provided. If it will go for fermentation for 96 hours, then the nutritional value of that leaf meal will be increased, and that can be used in any leaf. Growth rate. So I want to conclude from this thing that DORB should be mixed with exogenous enzyme. DORB SS should not be used.
Addition of some enzymes.
Fermented DORB gives a better result.
Amino acids.
Lysine and methionine supplementation with DORB improves the feed utilization and growth.
From LM40
Leaf feed can easily replace DORB in carb feed.
That's why
Which is suitable for our conditions. The fermentation of leaf meal reduces the anti-nutritional factors and improves nutrient utilization. Supplementation of enzymes and amino acids improves the growth of fish. The mixed leaf is better than to ensure the availability of the leaf throughout the year. So this is all of my presentation. Thank you very much for giving me a scope to share my some of the thoughts. But sorry, some technical problem was there. So it was created.
conclude from this thing.
Some disturbance there from my side because I am getting the echo all the time.
Addition of some enzymes.
Fermented DORB gives a better result.
Amino acids.
Lysine and methionine supplementation with DORB. So we can have some discussion.
From LM40
Leaf feed can easily replace DORB in carb feed.
That's why
Which is suitable for our conditions. The fermentation of leaf meal reduces the anti-nutritional improves nutrient utilization. Supplementation of enzymes and amino acids improves the growth of fish.
Check.
The mixed leaf is
So,
better than
Thank you so
ensure
much,
the
sir.
availability of the leaf.
Despite of the technical glitches, I hope the slides would be very beneficial for all of us.
Presentation.
And let's move ahead with the next keynote session by Dr. Kutubuddin Ali Molla. He'll be talking about precision plant breeding with CRISPR CAS tools. Let's have him. He's already here. So it would be a live session. I hope that would definitely benefit you a lot. Let's have a wonderful round of applause for the gentleman.
Thank you very much.
from LM40
Can easily replace DORB in carb feed. That's why which is suitable for our condition. The fermentation of leaf meal reduces the anti-nutritional.
Hello. Oh, am I audible from the back? Yes, thank you. Oh, thank you for your nice introduction, and thank you, the chair, Professor B.C. Tripathi, Dr. P.K. Mohapatra. Thank you, Agri Vision, for inviting me. And okay, get back to this physical environment. Okay, I know it was very problematic to our ear during the last session, but I hope you would enjoy every slide of my presentation if you are attentive. And I will be sharing few of my research stories with you, not a single story, but few stories. And to begin with each of the story, oh, thank you for your nice introduction, and thank you, the chair, Professor B.C. Tripathi, Dr. P.K. Mohapatra. Thank you, Agri Vision, for inviting me. And okay, get back to this physical environment. Okay, I know it was very problematic. Okay, great. To our ear. Okay. Good afternoon, everyone. So it's my real pleasure to be here and to talk about something very deep in the molecular biology and how you can edit the genetic letter or the letter in DNA. I know all of you, most of you are students, and you write something, project report or any kind of writing, and you give it to your teacher, and the teacher finds some mistake in your writing. So he suggests, or he or she suggests you to put a comma or maybe a full stop, or maybe delete one word and add another a good phrase to make your writing looks good. Same thing in case of our genetic letter, in DNA letter, you can do it with a tool, and I am going to talk about that tool today with you to improve the crop for our own benefit, to reduce the negative impact, and for many other aspects that you want to do. Okay, so moving forward, so let me go back to 9,000 years ago. Can you identify what is it? So it is called teosinte. It is the wild ancestor of the modern day maize or corn that we eat today. So in 9,000 years ago, it looked like this. Okay. Now after 9,000 years, what we got by crop domestication,
we got the modern image. And if you compare this one, it has only maybe 9 to 10 grains per cob. Now you can compare with the modern day one, and it is 1,000 grains. And it took by means of natural evolution and artificial selection, we could make our crop in such a way that it would feed our belly to secure the food security of the whole world. Okay, so this is because of natural mutation, evolution, and artificial selection. Now another type of manipulation that we do in our agriculture. Now suppose this is the wild teosinte that was found to grow in a very sparse manner, means here one and another one. So in this whole, if you consider this whole auditorium as a field, you can get maybe 15 or 20 plants, but that is not suitable for agriculture or farming, because you need to grow lot more plants in a small space, because you do not have a lot of agricultural land. So we modified this plant in such a way that they could grow or they could fit in a very small space, so that you can grow more crop from a small land. So this is how we modified the crop intentionally. Okay, and for many different reasons also that I mentioned here, that are suitable to human requirement, taste, yield, storage, and also cultivation practices. Now, let me give you some more examples here. Let me give you some more examples here. So if you see, this is Brassica oleracea, this is a wild mustard, and all the vegetables, we evolved those vegetables by deliberate selection. Suppose this one, kohlrabi, when you selected for stem, we developed this vegetable. When you selected for leaf, we developed kale. Kale is actually eaten in Western world. And then broccoli, stem and flower. When you select only for stem and flower, you develop broccoli. Similarly, flower cluster gives rise to cauliflower. Okay, so these are all our deliberate selection. They have not evolved naturally. Remember this. Similarly, here you can see in this photograph, this was a video, but I do not know, it is not being played here. So you have peach, banana, all these things, eggplant, where you modified their nutrition, you modified their taste, you modified their structure, even their size. Okay, so those are all deliberate selection. Now, in mid-1880s, when Gregor Johann Mendel outlined the principles of heredity, then we started scientific breeding. What is scientific breeding? So because we know or calculate how much offspring will contain this kind of character, how much offspring will contain this kind of character, okay? So that we can calculate after this discovery. So, but let me share this one. Here I got it from one of our students. So you have peach, banana, all these things, eggplant, where you modified their nutrition, you modified their taste, you modified their structure, even their size. Okay, so those are all deliberate selection. Now, in mid-1880s, when Gregor Johann Mendel outlined the principles of heredity, then we started scientific breeding. What is scientific breeding? So because we know or calculate how much offspring will contain this kind of character, how much offspring will contain this kind of character, okay? So that we can calculate after this discovery. So, but let me share this one. Here I got it from one of our students. So you have peach, banana, all these things, eggplant, where you modified the nutrition, you modified the taste, you modified the structure, even their size. Okay, so those are all deliberate selection. Now, in mid-1880s, when Gregor Johann Mendel outlined the principles of heredity, then we started scientific breeding. What is scientific breeding? So because we know or calculate how much offspring will contain this kind of character, how much offspring will contain this kind of character. Okay, so that we can calculate after this discovery. So, but let me share this one. Here I got it from one of our students. back. Hello. Okay, so I think I request because I'm going to get a little bit more time because I lost 5 minutes here. Okay, so let us back to the point where we are. So what do you think? Agriculture is natural or artificial? What do you think? Anyone? Agriculture is natural or artificial? Or man-made? Agriculture is man-made. Okay, now okay, so this was the last point I was talking about. So you can see this is the rice varieties and different kind of germplasm and lot of variation that you see in this phenotypic, in this picture. The grain size and also the color is different. And okay, so I think and why this kind of variation exists? Do you have any idea why this kind of variation exists? Because of the genetic variation and also their interaction with the environment. Okay, this kind of variation exists, and you can see anyone if you compare this rice with this rice, and if you compare them, you can see a lot of different genetic variation exists, and because of one of the genetic variation that caused the size smaller and also the color is different, and also it may also differ in taste. So genetic variation is the key to the phenotypic variation that you see. Okay, now up until 20th century, spontaneous mutation was the only kind of mutation. Mutation means the way DNA later changes from one type to another. That is called mutation. So until 20th century, we have the only way to harness the power of spontaneous mutation. What is spontaneous mutation? Mutation that happens in nature without any human interference. That is called spontaneous mutation. But remember, spontaneous mutation is very slow in nature. It's not as fast as you want to make. So mankind could exploit in selecting plants and animals for those phenotypic variation that have arisen due to spontaneous natural changes in the DNA. Have you got my point? Okay. Now we have a lot of problem arising, and then as we learn that the genetic variation is the key to crop improvement, because as I showed you, different grain shape, different color of the seeds and yield improvement all are dependent on genetic variation. So as it is key to crop improvement, then we started pondering how to really we can artificially generate genetic variation, because spontaneous is slow. If you want to get more phenotypic variation, you need to do some artificial way to make changes in the DNA, because spontaneous is slow. Now this is a photo that I showed you. This is a gamma garden. It is a radioactive garden where your plant is kept and the gamma radiation is given, and this is called mutation breeding. Okay. And now, as a result, in 1920s and 40s, we learned this procedure, and we treated our plants with X-rays, gamma rays, and some chemicals also to induce changes in the DNA artificially. That was not naturally available. And we got this kind of variation due to the treatment of this kind of gamma chamber treatment. Okay, so a lot of different variations. You know why they have arisen? Because of your treatment with radioactive, a very dangerous thing to that causes a lot of changes in the DNA, and that ultimately manifested this with this kind of different colors of a flower. This is an example. Now, but remember from this slide, what you have to remember, mutation breeding is random. So you cannot fix that you need to change this portion of DNA with mutation breeding. If you do mutation breeding, the whole genome is being modified, you do not know. And then you select those plants, you grow, suppose you have 1,000 plants out of a mutant population, and you select the plant that you like, that you want to develop, but that is by chance. You may get or may not get. That is also a by chance thing. Okay. Now, if I want to give you a statistics, Food and Agricultural Organization, they said that more than 2,500 plant species all over the world that are derived from mutation breeding, that we are eating those plants without any harmful effect. Remember, out of mutation breeding, that causes random changes. Now, you cannot, okay, no, it's okay. You need to change this. Now, what if we have tools or methods that allow us to modify DNA at a desired spot? Because we know we have improved a lot in genome sequencing. We know which gene is present and which gene is controlling what kind of trait. And do we have a tool that can allow you to modify DNA at the point where you desire? So now this is called precise genetic variation on demand. And here comes the CRISPR-Cas that I'm going to talk about a little bit more in my next slides. Okay, so this is the outline of my today's presentation. Is it visible? No. Eating those plants without any harmful effects. Remember, I don't know why. Again, it is gone. That causes random changes. Now, you cannot. Okay, no, it's okay. Now, what if we have tools or methods that allow us to modify DNA at its desired spot? Because we know we have improved a lot in genome sequencing. We know which gene is present and which gene is controlling what kind of trait. And do we no, sir, it is not working. I'm just moving it, but it's not working. To modify DNA at the point where you desire. So now this is called precise genetic variation on demand. Okay. So this is the outline. The first part I would be talking about what is genome editing, especially CRISPR-Cas9, focusing on, and how is genome editing helping in precision breeding. So breeding makes more precision. So I'd be talking about CRISPR-Cas9 in a perspective of a new breeding tools that helps breeders to go for precision breeding. In part two, more precise editing, that is called at a single base resolution. So you can change a single DNA letter in your gene as you desire. And in part three, I would be talking about few examples of plant breeding innovation out of these tools. So coming to the first part, let us go to the basis of this talk. So, human body, all the higher eukaryotes, we are made up of almost 37 trillion of cells, similar to a rice plant also. This is higher eukaryotes, and it has also trillion of cells. And this is an example of a plant cell, and that has this nucleus. That nucleus is the head of the cell. Why this is head of the cell? Because this nucleus has chromosome in it, which is the hereditary material. And what is there in chromosome? Chromosome is nothing but a long stretch of DNA that is coiled around histone protein. But no need to remember histone protein here. Only remember chromosome is DNA. Okay. Now the whole long stretch of DNA is not equally functional or is not equally important. And the portion which is important or functional unit of the DNA is called a gene. Now, what is in the gene and why gene is functional? Because when this portion, the green highlighted gene, is expressed, it produces some mRNA and then it produces a protein. Okay? And this protein directly or indirectly controls a character. Why this is head of the cell? Because directly or indirectly controls a character. And that is why gene is a functional unit of a DNA portion. Okay. Now, what is there in the gene? It is nothing but these 4 letters, A, G, C, and T. But suppose gene 1 and gene 2, what differences they have? Gene 1 is different from gene 2 by their sequences. only by their sequences. But the later are all same, A, G, C, T, but their sequences are different. Suppose there are a lot of girls here. So if you wear a necklace out of lot of beads, and different colors of beads, and if you see one beads is one A, another bead is G, another bead is C, another is T, and the combination of one student is different from the another student because they have different necklace, okay? And the pattern is different, and that is the difference from one gene to another gene. Now, in order to introduce, let me go to a plant pathogen interaction story where, bacterial blight of rice, it is caused by the bacteria Xanthomonas origi. And this bacteria is very, very intelligent because whenever, and let me just start with, this is a rice cell, and it has a gene that is called sweet gene, and this gene produces a protein. As I told you, gene produces a protein. This gene produces a protein which function to transport sucrose from inside to outside of a plant cell. Have you got my point? Sweet gene produces a protein that transports sucrose from inside a plant cell to outside of a plant cell. Now, this Jew is the pathogen Xanthomonas origi, which is very intelligent, as I told you. It secretes another protein. This bacteria secretes a protein, and this protein goes and binds with the promoter sequence of the gene. In order to produce a protein from the gene, this region is called promoter, which is very important. Now, the protein which is secreted from the bacteria, it comes and binds here, and it induces the expression of this gene. It activates the expression of this gene, so that this gene produces more number of protein. You can see here a lot of protein, okay? The colorful protein, violet color. And as a result, there are more protein, sweet protein, and it will pump more sucrose from inside to outside. So outside sucrose is available. Now bacteria is very happy. Why happy? Bacteria needs nutrition. Sucrose is their nutrition. So they are tricking a plant gene in order to feed themselves. Okay? So their food security here. Okay? Now, you see the result. When there are a lot of sucrose, they grow more. They have nutrition abundant. And this is how rice plant is being affected by this bacteria, which is very important. Bacterial blight disease, where Dr. Arup Mukherjee works in our institute. So he knows better than me in this aspect. Now, if I want to give you a zoom in view, here, sweet gene, and this affected secretes from the bacteria and it binds here. This is called EBE sequence, elicitor binding element. Elicitor means this is called elicitor. Okay. Now the question, and this binding is very specific. This effector from the bacteria cannot bind anywhere. It can bind only in a specific region of the promoter. Now, do we have this is the question to you. Can we specifically disrupt the EBE sequence in the promoter? If this promoter region EBE is disrupted, somehow the sequence is changed, then the effector would not be able to bind. Because I told you that this is very specific binding. It only binds to this specific sequence. So if you change the sequence, then this effector would not be able to bind here, and this gene would not be induced. So it would not transport sucrose outside, and bacteria will be deprived from nutrition, and your plant will be saved. The question, if I want to answer the question, the answer is genome editing. Yes, you can do that kind of manipulation in the sequence by genome editing. So in a way that change the sequence in such a way that it no longer works. Suppose this region is the what region? EBE sequence, and you just lock that sequence so that the tail effector cannot bind there. And this is how the sequence looks like. And if you change the sequence, disrupt the sequence, EBE would not be able to bind. Okay? So let us come to this CRISPR-Cas9. That is a way of genome editing that allow you to changes the DNA later, that AGCD sequence. It has two components. One is a customizable RNA. There's a small RNA that you can customize by yourself. Suppose you go to Pantaloons, you buy a dress, that is already made, and you see whether it is fitting you or not. But when you are going to marry, you would not go to pantaloons, you go to a tailor and tell them, okay, so make something which fits me. So this is my marriage coat. So this is also customized. Okay, so this is a customizable RNA. So you can also design this RNA. And another portion is DNA cutting protein. That's called Cas9. It is a nucleus. Nucleus means it degrades DNA. And if I want to give you a very simple analogy, then Cas9 is the postman. Is a postman which has two scissors in his pocket, and this is the address, genetic address that you have to customize or you have to give. So now, when you give the genetic address to Cas9, Cas9 takes the address, means postman takes the address, and it goes to the genome, and it finds for the address. It will look where is the address is present. What is address? Because RNA has letter. Similarly, DNA has a letter. So they will find for a complementary sequence similar, and whenever or wherever they find, they will bind there. And this binding, absolute binding, signals the postman that this is the address where he or she needs to make a cut. Okay, now what is the next step? Next step is, because the postman has got the signal and it has two scissors, one scissor would cut one strand, another scissor would cut another strand. And this is how you have the ability to give the address to the postman to make a specific cut to the gene that you want. Okay, now you can ask me, okay, so we know that it can make a double strand break in the DNA where I want, but how the editing is happening? Here is the answer. So, you know, when this kind of double strand break in the DNA happens, it is a serious event for any cell, because DNA is the hereditary material and DNA controls almost everything how an organism should behave. Okay, now any DNA damage is very, very deleterious. So that is why cell immediately want to repair it. And while repairing, it has two pathways. One is predominant pathway, almost more than 90% time this pathway works, NHEJ. And this pathway, when it repair the two broken end, it is, you know, it makes sometime mistake. And due to those mistake, it inserts some new DNA letter or delete some original DNA letter that I showed here in red color. And as I told you, a gene is defined by its sequence. So now when there is a new insertion and deletion happened, then the whole gene sequence is disrupted. And this is called open reading frame, and this open reading frame is disrupted. It means it would not produce the same protein as it was producing earlier in its original form. So you get your gene knocked out. What is knocked out? You disrupted the function of the gene because it is not now producing the same protein. Okay, so this is how you generate a gene, what is gene edited things. Now this video would, okay, so let me see. There was a video, I do not know, it is not getting played. Double letter is being deleted, that is random and we cannot control that. But if we have a big repair data, big data means you have thousands of experiments data, and what kind of experiment you have done, that means all data you have, and if you have those data and the genomic sequences, you can feed in machine learning things, means machine could learn that what kind of sequences would give rise to what kind of mutations. So, and those models could be developed. And let me give you a simple example. So this is the genetic address that I told you. And if the -4th position has a T, then if the postman cuts there, then you are most likely going to get another T inserted. If you have a double C here at -4 position, you are most likely going to get another one C is deleted. So this is how you can predict what kind of mutation would be generated from your genome editing experiment. Okay, so this is just example from one of our work. You can see this is the chromatogram. Chromatogram means this is how we read our sequence. So this is a sequence that we targeted, and you can see up. This is the portion. This is -1, -2, -3, -4. What is there in -4? A T. And you can see here how many T? Double T is there. So our prediction now works here. So this is a real example from the real experiment. You can see the prediction was another T inserted, and here we got another T inserted. Double peak. Red peak means T, black peak means G, green means A, and blue means C. So here double red you can see. Okay. So this is how we can predict, but I'm not going into details. Okay. So let us just give you a real life problem. What is this problem? This is a rice field. This is also a rice field. What difference you see here? What difference? Lodging. So if you have a high wind velocity, even cyclone, Odisha experience very frequently, you get this kind of situation, and where farmers are suffering a lot because of this lodging problem. And I think it was common maybe 60, 70 years ago, because we had rice plant this much of height, and any kind of wind velocity, it would lodge and it would fall down, and then farmers cannot harvest properly, and they suffer from yield losses. Okay. Now, what technological advancement helped us to overcome this problem recently? You know, green revolution. You know, green revolution. What is green revolution? Here is the story of green revolution on that photograph. You see, normal is a tall plant. Now you have to make it lodging resistance, and in green revolution we developed semi-dwarf rice. Semi-dwarf means a little bit, smaller. This is taller and smaller. Taller is bad because it has the proneness to lodging. Now we made it lodging resistant by developing a lot of semi-dwarf cultivars in wheat, in rice, whatever you eat today, even last dinner or last lunch you ate, all are from semi-dwarf rice. Why this happened? Because of a mutation in SD1 gene. If SD1 is functional, you are going to get this kind of tall plant. If SD1 is non-functional, you will get a smaller plant. Why? Because SD1 is a GA biosynthetic gene, gibberellic acid biosynthetic gene. Okay. So this is a very well-proved technology. You can see this is the long pita variety, and this is the DGO gene, very small one. And when they were crossed, it developed a semi-dwarf rice cultivar IR8, and that's the first green revolution variety that is also called as a miracle rice, and that gave us food security, especially in India. Now, what happened to that SD1? When people analyzed why it became semi-dwarf, IR8, why it became semi-dwarf, they have seen this gene SD1 has this kind of deletion. This 383 base pair deletion from exon 1 and exon 2. There is a long chunk of DNA. 383 DNA later is deleted. Where? In the semi-dwarf, when they compared with the longer one. Now you see this deletion helped you to achieve your desired character. Now what we are doing, But this kind of, if you want to cross them, sometimes normally people have been too much successful about this breeding techniques, but it takes long time to make a semi-dwarf cultivar. And also, when you transfer this SD1 mutated gene, you can also transfer some associated negatively impacting gene that is called linkage drag. Okay, but with the genome editing technology, we can make, without any cross, we can make the same variety. We can make this kind of deletion in precisely in that SD1 gene without any linkage drag effect and without taking too much of time. So our approach is the same gene is there. We have some economically important cultivars from Odisha, and we have selected those cultivars, and we selected SD1 gene, and we will make one cut here, another cut here, exactly similar to the one that has already demonstrated through traditional breeding. So while we make two cut with the postman Cas9, we can delete the intervening region. Okay, so this work is going on and we got some initial success, but I'm not going to go giving you a result here. Okay, so now coming to more precise breeding, this is very interesting. And now we are going to more precise breeding. So you can ask, wonder that what would be more precise? Because going to a specific gene and deleting that is precise, but what is more precise? Here is the, please conclude. But how I can conclude? I already wasted 10 minutes, but I will try. Okay, sure. So here you can see which movie is this? Oro and Oro's friend. Both are 12 years old. And do you know what is the difference among them? The difference is a single DNA letter. Oro had a wrong letter A, and she has a right G. So because of this single wrong letter, Oro was suffering from a childhood aging disease that's called progeria, which is a rare genetic disease. And we knew the reason, but we did not have any tool to correct it. And Oro died at 13, like all other progeria affected children. This is an example of from plant perspective. This plant is susceptible because of a wrong letter C. When the C is mutated to T, it becomes a disease resistant cultivar because of a single letter changes. Okay, now the question here I ask you, is there a way to rewrite genetic letters? Rewrite genetic letters precisely, not disrupting a gene. Here you have to replace the wrong letter with the right letter. The question, the answer is base editing is a technology that I'm working with that is kind of pencil and that can change in a single base resolution from A to G, G to A, and also T to C, C to T, and similarly C to G to G to C. And this is how it works. This Cas9 is fused with the D aminos. This is base editing technology and is developed by David Liu from MIT Harvard. And just remember this phase. He is going to be the next Nobel laureate in few years. A lot of people are, this is not my word, but a lot of people are expecting because he has contributed immensely in the therapeutic field of genome editing. So, okay, coming to my, just give you an example. What do you see here? The wrong letter. This is a sky that does not have any star. Why? Because you got some problem in the code. Now, if you have base editing, you correct it and you got twinkle, twinkle, little star, and you can't affect the right genetic codes effect is lot of star in your sky. So this is exactly base editing if you want to remember for a long time. Now, let me give you an example of base editing that is called adenine base editor, which can perform A to G editing or in the opposite strand T to C. So I'm not going into details in this. So just sharing my experience with this. So we targeted this A letter. Here is a control plant, means wild type plant, normal rice plant. The gene is WSL5. This green peak means what? A. Now we wanted to change A to G by applying base editing technology. Now, when we applied base editor, and we could achieve precisely A to G. Precisely A to G. And you can match the other peak, they are unchanged. They remain unchanged. only this targeted A has been changed to G, and this is the resultant of the treatment with base editor. And when you see the phenotype, why you targeted that specific A, because this is for demonstration purpose, and we knew that this A is very important from earlier literature. And when we modified this single A to G, we got this kind of mutant which have white stripe in the leaf instead of a green color. So yes, sir. So now this is the second one, A to G also, and we got same A to G changed, and we got this kind of phenotype. And you can also develop very quickly base editor pre-mutant, even in a single generation where you do not have any base editor incorporated. Okay, I do not have time to discuss. So this is another one. This is the last story I would want to share with you. This TAG is a signal to the protein synthesizing machinery that you should stop here. And this is a stop signal, okay? TAG. If it is present in DNA, that protein synthesis would not go beyond this. Okay. Now, because of this, in some genotypes, there are some natural mutation, and it should be present at the end of a gene. But now, because of the mutation, it is present at the beginning or at the mid of the gene. So you would not get a whole length of protein, and the plant became disease susceptible. Now, when we tried to mutate or to correct this kind of signal, TAG, to a TGG with the base editor, and now this TGG gives you green signal for protein synthesis. Now you get the whole length of protein, and now the plant becomes disease resistant. So this is already developed, and this is undergoing further experiments. So if you want to read more about this application, this precise editing tool, I would recommend to go through this recent review article that we wrote. And it could be used in herbicide tolerance development, nutrition alterations, and also rapid domestication. What is rapid domestication? Here, with this technology, you can target the domestication gene and you can reduce the time drastically. Similarly, yield improvement, also disease resistance that I showed you an example in my work, and input use efficiency. So nitrogen or other fertilizer use efficiency can be also improved. So this technology has already been applied to different crops. This is the snapshot only for different traits. And so this is for your question. So what I showed you, these are not only literature. So are there any crops that are commercially cultivated in the world out of genome editing? The answer is yes. So here is a soybean. This is a Calino soybean that has the oil quality like olive oil. This is already approved in USA and it is being marketed. And recently in 2021, a high GABA tomato has been approved in Japan, and it helps in lowering blood pressure. So these two crops, if you get a question in your CSN net or some other interview also, you should mention these two. One is soybean, another is tomato. Okay, if you want to know more, this is the thing, and I should acknowledge all my previous lab mates. And this is my recent group in NRRI, and I have also collaboration with Dr. Mukherjee and other labs also. Thank you very much for bearing with us, but sorry for taking more time. It's done? Awesome, just let me. Okay.
Thank you so much, sir. So this would be the last session for the pre-lunch sessions that we are running. Post this, we will have all the sessions after the lunch. So this one is by Dr. Yashashvi Nayak, HOD, Department of Zoology from CUTM, Bhubaneswar, India. An integrated system of fish and plant farming is her topic. Let's hear it from her. I guess this would be an virtual session as well. Yes.
Thank you so much. Just a minute, let me share my screen. I hope my screen is visible. Hello. Hello, madam. Am I audible? Hello, am I audible? And is my screen visible? Hello? 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 Hello, am I audible? Hello? Am I audible to all? Hello, am I audible?
Check. Yes, ma'am, you're audible.
Okay, thank you so much. So I thank the AgriVision to give me honor to participate in this conference. So thank you so much. So I'm here with my topic, aquaponics, that is growing fish and plants together. So it is simply the combination of fish and plants growing in harmony, that is water containing their waste products are processed by a biological filter and then treated, and then it is directed to the plants. So, madam, beg your pardon. Can I off my video because my bandwidth is very low? Hello?
Yes, ma'am, we can hear you. You can.
Okay. My bandwidth is low. Can I off my video?
Yes, you can.
Okay. Thank you so much. So this is what the aquaponics is. So it is basically a sustainable mode of organic agricultural practice that involves a symbiotic combination of aquaculture and hydroponics. Hello,
Yes, ma'am.
my slide is moving.
Yes, it's moving, ma'am. We can hear you too. You can continue with your presentation.
Okay, but I'm not able to see my slide moving. That is why I'm just, okay. So madam, it is not moving.
No, ma'am. Now it's not moving.
Just a moment. Because it is a problem if I do screen, if I do slideshow mode, that is why. Anyway, let me check again, please. Please bear with me, just a moment.
Ma'am, can you please share your screen?
Okay, I'm already sharing my screen, madam.
Yes, I guess she is sharing it.
Okay, let me just stop it and share it again otherwise. Now my screen is visible.
Yes, it is.
I don't know why it is not going, why it is not moving. Just a moment, madam, just a moment.
It's okay, ma'am. Ma'am, if you can open your PPT and then if you could move the same.
Yeah, just a moment. Yeah, that I'll do it.
Ma'am, you can try the rejoining.
Yeah, now it is moving, madam. Can you see?
No, we can't see, ma'am.
Okay, Just, yeah, I will just leave and rejoin otherwise. That will be better. Yeah, just a moment, madam.
Can we check if she rejoined? Now she's there.
Is my screen visible, madam?
It is visible, ma'am, and you're audible as well.
Okay, so let me not do the slide mode, otherwise it is not moving. So I hope it is comfortable for you.
Yes, it is comfortable for all of us here.
Okay, thank you so much. So let me start. So I am here with aquaponics today, which is the growing fish and plants together. And this is basically a combination of fish and plants growing in harmony. That is water containing their waste. Products are processed by biological filter and then directed to plants. So it is a sustainable mode of organic agricultural practice that involves a symbiotic combination of aquatics and hydroponics. Now, basically, it is also defined as the growing fish and plants together in a recirculating system. So it involves two systems, that is aquaculture as well as the growth of the plants, and the plants grow without the media. So how exactly the aquaponics works? So here the fish are raised in a tank. So can you see the fish tank here? So fish are raised in a tank and are provided with fish feed, and these fish, they produce ammonia as a waste material after having those feed. Now fish also create ammonia through their gills, and the bacteria which helps in transforming the toxic ammonia into less toxic nitrites and nitrates are introduced into the grow beds. And so the nitrosomonas bacteria which are present in the grow beds, they love to grow in oxygenated water, and they convert the ammonia, which is a waste product of the fish, the first waste product of the fish, and they convert into nitrites. But the nitrites are still toxic to the growing fish. So again, another bacteria, that is the nitrobacter, so that converts the nitrates into nitrites, and plants, and the nitrites filled with water enters into the plants, and the plants love the nutrition provided to them by the fish. Now let us consider it as why it is sustainable. So when a system is maintained in a steady state, aquaponics works as a simple ecosystem. So let me first tell you what is basically sustainable means. So basically sustainable means meeting the needs of the present generation without compromising the ability of future generation to meet their own needs. So in this system, why we consider aquaponics as sustainable? Because in this system, it is maintained in a steady state and it works as a simple ecosystem. As a result, it is considered as an environmental friendly and regarded as a sustainable agriculture practice. And moreover, it also provides continuous organic fertilizer. So why people should go for aquaponics? Many people, they say, why should we go for aquaponics? But the answer is that it is enjoying a resurgence in the popularity across the world. And the simple reason is the current number of people in the world, they are going without organic food. And an aquaponic system can produce fish and plants to eat while taking up considerably less space than traditional farming methods. And a refined version of this is known as the recirculating aquaculture system, or simply we also call it as RAS. RAS, which allows the farmers to stock the water with much larger quantity of fish than the traditional netted off areas. Unfortunately, in farms, a large amount of antibiotics is also needed to keep the fish healthy. And there is a significant amount of waste are also produced. And because of which, there is an increased cost of this type of breeding and potentially damages the environment of other living animals and human beings. So aquaponics offers a more environmentally friendly way of breeding fish with the added benefit of accelerated plant growth. So here we do not use no pesticides, no herbicides, only the waste of the fish, we call it as fish fertilizer, and which can also be grown in your indoors and can get the food all year around. And this also works in a drought areas which is having a poor soil quality. And water used in comparison to this is much less. In other farms and ponds, we use thousands and thousands of gallons per pound of production. Whereas in integrated aquaponics, wastewater is directed to greenhouse and there is no discharge. So that is why the less water consumption is there in this system. Now, in comparison to this, it is also that it is not just the better for productivity and reducing environmental issues, but also an effective method of growing organic plants without using large amount of water. So a properly closed loop aquaponic system needs only 10% of water without any discharge. Next, we'd like to see what are the components of aquaponics. So basically it requires a fish tank, a place the plants to grow, the water pump, the air pump, usually in the aquarium, whatever we need it for the survival of the fish, and the irrigation tube. The water heater is actually optional, the filtration is optional, but still we use it so that the bacteria can grow, and then fish and the plants. So these are some of the things. A small fish tank, which is also required. It can be in the form of aquarium. Just above the aquarium, also you can grow the plants. So these are some medium-sized fish tanks which can be used as an aquarium, and also on the top of that, the plants can grow by using the water which comes out from the fish. So these are some big fish tanks which can also be used. And recently we are trying to get it in a university. So we are just in a very infant stage. So we are just making the whole aquaponics unit to grow. So the safe materials for using, the safe materials to use in aquaponics in the tank system, basically, it is the propylene and it is the PVC pipes, and which are all the UV resistant and also which are fire retardant material. And this is a caution for everybody that we should never use any copper thing in this aquaponic system, which is basically a toxic to the fish. Next is aquaponics system design. So we have 5 different types of aquaponic design. The first one, let us say it is media grow based system. So this media grow based system, just a moment. So this is the media filled base. This is generally considered the easiest form of aquaponics, and the best one for the beginners, because it acts as a grow media and a biofilter, and the media filled bed is also called as grow bed. Generally, this grow beds, can you see this? So these are the grow beds, and generally these grow beds are used with larger plants, and they need big roots to keep them upright. But sometimes the depth of these beds is also 12 inches deep and are perfect for growing some herbaceous plants like tomatoes, coriander, lettuce, all these things. And there are two types of systems basically for the media bed. The first one, we call it as continuous flow, while the other one is known as flood drain or ebb and flow, which requires this ebb, this is the siphoning system. So continuous flow will keep the grow beds filled at a constant level, which means the media will be submerged all the time without letting the oxygen to access the roots, and which will be a problematic if we do not have a proper aeration to our system. So that is why usually we don't recommend using a continuous flow system. So we recommend flood and drain where there is a siphoning system, and the siphon method also helps to fill the whole media bed with water and then drain again using a siphon. So this way the growing media and the roots will be exposed to the air in every cycle, which also promotes bacteria to grow and roots to be exposed to oxygen. Next, I'm sorry. So these are some of the media based things, that is we can use the gravel, the hydroton, the lava rock, the packing foam, sponges, or perlite and vermiculite. So all these medias are basically, they are they have the capacity to retain the water and also helps to moisturize the roots every moment. So that is why we use these kinds of media based grow beds. So they are spread in this grow beds, and then where the plants can plants can grow properly by making use of the water which comes to them through fish waste, I should say. But there are some pros and cons. So the pros are that they are easily, they easily found in the markets, easy to build, and also grows lots of supports the growth of lots of different plants in one system. Whereas the cons are that they build up a lot of anaerobic zones sometimes, so, and which it is very difficult to clean it also. And sometimes it requires worms. We use the worms to clean it up. So that is basically the earthworms of Eudrilus eugeniae. So now these are the aquarium systems. So these are some of the systems which we have grown in our labs. So now we are making it into the open, like we are now having some big tanks to have this kind of system. So these are some small ones which we have grown it in the labs just above the aquarium. So we have some air filters and the connection between the plants as well as the fish. The fish waste is coming into the plants, and again from the plants it is going back. So it is just recirculating the things. So this is how we have developed in our labs. But now we are trying to put it in the open air. So we are just in just an infant stage. Now this is another one which is known as the barrel ponics, the barrel aquaponics system, which uses 55 gallons of drum, and which is cut in such a way that one half is made into the fish tank and the other half is used for growing plants in grow media. Water circulation and air are provided by water and air pumps. Now these are some of the grow beds. We are trying to build up this type of grow beds in our university, so which we are still trying, but we have not made it now. And these are another one is that was the media types, media filled bed, and this is another method of aquaponics which is known as raft method. Now in the raft method, here planting in a raft system of aquaponics, it involves suspending plants in a floating rafts with their roots hanging down in water. Now these floating rafts have hole drilled into the fit to net the cups used for supporting plants. Now, if you see, so these are some of the rafts. These are the raft system where you will find the holes, and in the holes you will find the some of the cups, and in these cups the plants are planted. So basically, after the plants grow it into 10 to 12 centimeters, then we put it in this raft to grow it further. So basically, we use some lettuce, some herbaceous plants, lettuce, spinach, and tomatoes, chilies, capsicums, and some of the big plants like big vines, I should say, like cucumbers, and then sometimes carrots also. So we can use this raft method to use this. Next is, now these are the small scale raft systems which supports the lightweight plants, and also the vegetables also grow in a very good manner where the environment moist environment can collaborate with the small root system. So next is another one, and the pros and cons of the raft method is that the pros is that it gives you very high yield of both fish and plants crop, and as well as the fish. The size of the fish also grows in a very small period of time. Whereas cons is that why we should not use this raft method is that it allows to grow only a specific type of crop, that is either lettuce or basil or sometimes cabbage, like that. Very short-heighted plants. Then this is another one which is known as nutrient film technique. Now this nutrient film technique. Hello, am I audible? Hello, am I audible to all of you?
Yes, audible.
Okay. Now this nutrient film technique is, the growing area is very much essentially a collection of small gutter pipes. So can you see these are the small gutter pipes over here? And each of the gutter pipes have holes. So I think the holes are also very much visible, holes in the picture. And these holes are cut out where a plant should be located. And the plant is placed in a cup within the hole and allowing its roots to go to the bottom of the gutter and access the nutrient-rich water. In this case of aquaponics, the waste water from the fish passes through the solid filter and biofilter first before flowing along the gutter pipes and then into the sump, and then again it is pumped back into the fish tank. So here, see here the fish tank with this, this is the fish tank, and then the water from the fish tank enters into the biofilter, and then it enters into the plants. The roots are washed with the nutrients of the fish, and again it comes to the sump tank, and from here the water is again flows back into the fish tank. So this is what is known as nutrient film technique. This is another one. The next part is the another one, and this is how even in the rooftop garden can also be constructed by using this technique. So this is a concept of NFT, that is nutrient film technique. So here the plants are just set up in a row wise, and which is then it is connected with a water connection, and also having the fish inside the water tank. Next is the vertical towers. So this is very much prevalent in foreign countries basically, but nowadays it has also come up in Bangalore and Hyderabad. So one of our faculty members have done this in their rooftop. So, but it has still not grown properly. So it is the method that grow plants without soil in the columns above a fish tank. So this is what is the vertical towers. It is in the form of towers, basically. So that is how, and these towers are connected with the water tanks below, and above the water tank, these vertical columns are made with the holes, and from the holes arises the plants. And then now here, in the vertical system also, the vegetables, the small herbaceous vegetables can also be grown. So these are the tomatoes, which here, these pictures, we have got it from a place near Hyderabad. So one of our student is working on this. So he has sent us the pictures. So that's how he is also, undergoing this kind of project. So very recently he has sent up this photograph with his vertigrow system. So which is a very simplified version of vertical aquaponics, where the grow bed is positioned on the top of the fish tank. And it is but only one layer. But a true vertical system will focus on growing as many plants as possible on the top of each other. Next, fish inputs. So basically the feed which the fish is taking from the water, it is the feed which we give it to the fish. So it is in the form of basically the fish feed that is available in the market. And the output is basically the water and the ammonia, which is a nitrogenous waste, the carbon dioxide and the feces and this uneaten feed. And that is why time to time it has to be always cleaned. The fish tank should be cleaned, the solids should be removed, and the biofiltration portion should be cleaned, and that helps in proper aeration and oxygenation. Then which also, and cleaning the tanks always makes the fish healthy. The pH is maintained. The pH always should be maintained between 6 to 8. The nitrates are fairly safe. Then fish needs oxygen. Otherwise, without the oxygen, the fish can die within around half an hour. So that is why the battery-based aerators are also used sometimes if there is some power failure and all. So this battery aerators, they can be used as a backup. And we should not give the fish the direct sunlight, and because there are some fish which are very sensitive to light. And these are the temperature and the growth rate, because the temperature, hello, the water temperature is one of the most critical parameters that an aquaponics grower must maintain consistently. And the water temperature must be kept in the range which is safe for the fish, plants, as well as bacteria. Now, there are many factors that can affect the water temperature, such as the capacity, the climate, the air temperature, the tank materials, the growing systems, the insulation, as well as other backup systems also. Now, the optimum temperature range for aquaponics is basically 22 to 32 degrees Celsius in the tropical areas, and the cold water fish can sustain by 10 to 18 degrees Celsius. And most of the vegetables that are used to grow in aquaponics are between 18 to 30 degrees C. So you can see in this graph, so here the fishes thrives well in 24 degrees Celsius with the growth almost, the specific rate is almost the maximum in this area. So, but when the starvation happens, so that always goes down. The plants are also affected by the water temperature, and also if the fishes are starving, the growth of the plants also goes down. So this is what about the temperature and growth rate. Now these are the fish which we can use it in the aquaponic system. That is some of the aquarium fish, small fish, guppy fish, angel fish and all, tilapia, yellow perch, trout, catfish, bass, all these things. And what we usually consider it in our the aquariums are basically goldfish. So some of the goldfish, koi, guppies are also used as an aquarium fish. And then tilapia. Tilapia, we are now just making this tilapia to use it in the big ponds, which we are just constructing it in the open areas. So, and this tilapia can survive in warm water around 20 to 30 degrees Celsius, which also can tolerate pH and shifts temperature changes. And also these are the omnivorous fish, which can have the pellet, which we provide in the form of the fish feed available in the market. Sometimes the duck feeds, veggies from the system also. And also it can grow within 6 to 9 months in ideal conditions, and which are ready for marketing. And these are the yellow perch, which can also tolerate a lower dissolved oxygen and adjust to some pH changes. And the next one is the trout, which also requires high oxygen levels and are very sensitive to pH changes, and which also eats pellet fish food. Then next is our bass, bluegill, catfish. So all these are the fish which can retain in the changing pH and temperature, but there are some fish which are very sensitive to temperature, pH and water quality. And we can harvest this vast within 15 to 18 months. The bluegill harvest can be done in 12 to 16 months, whereas catfish harvest can be done within 15 to 10 months. Next, some of the fish like koi fish, goldfish, carp, etc. So these are all omnivorous fish, and they can be also sold as pets if they are suppose if they are just maintained in the aquaponics. So we can always sell them as pets also. Whereas koi are a fancy expensive carp which needs around temperature of 65 to 75 Fahrenheit. And these are some of the fish health management we should consider because sometimes what happens because of high pH or because of some problem in the water. So sometimes the fish develops some fungal disease. So we have to quarantine those fish before stocking them in the system. We should monitor their health, and then which we should provide them very low stress environment so that the fish can maintain their health. Next is our fish maintenance. So we should observe the fish behavior and appearance, should be fed 2 to 3 times a day, and we should monitor their body weight. And we should also check after giving them the feed, the water quality should be checked. If there are some leftovers are found in the water, it has to be removed. Otherwise, the pH alters and it gives stress to the fish. So these are some commercial feeds provided to the fish, which are laden with protein, carbohydrate, and other vitamin requirements for the specific fish. And also there are some plant-based proteins that are provided, which includes soya meal, cornmeal, and wheat bran, rice bran, etc. And the commercial feeds are basically gives 10 to 35% of the proteins. And apart from this, we should also take care of the presence of the bacteria. And the 50% of fish waste is in the form of ammonia that is released in the form of urine and also the fecal matter and also through the gills. Now this bacteria consumes the fish waste, and then there are two bacteria, two different types. One is nitrosomonas and one is nitrobacter. So the nitrosomonas converts ammonia to nitrate, and then from nitrate to nitrite by nitrobacter. So nitrate is a common primary source of plant nutrition, which is a good stuff of nitrogen, safe for fish and for the growing plants. Now, these are some water testing things. So we should always take care of this water testing capacities. So what is this? Basically, water testing means the pH has to be monitored. So if the pH is not monitored, it may kill your fish, or it may take the toll of the death rate of the fish becomes high. So water testing is very much essential. So in this, the water testing, basically, it helps the fish to grow in a normal pH, and also maintains the pH in such a manner that it provides the good environment for the fish as well as the growing plants, because there are some plants which are also very much resistant to high pH. So these are some of the plants which we usually use in the aquaponics, that is some vegetables, herbs, fruits, and flowers. So we have tried with the lettuce, we have tried with the spinach, we have tried with the basil, and other fruits and herbs we have not tried. So we are yet to try over this. So why do plants basically like aquaponics? Because the roots are constantly in touch with water and they don't have to search for food. So they also, for them, it is less effort is needed for them to get the energy. And moreover, the weeds are not present. It does not encourage the weeds to grow. So here we can grow the lettuce, which the ideal temperature of the growth of the lettuce is 60 to 80 Fahrenheit, and also can tolerate up to 45 Fahrenheit. Temperature over lettuce bolts, if it is above 80 Fahrenheit, then it is difficult to grow for the lettuce. Even the tomatoes also, here the tomatoes are very popular in hydroponics, and the ideal temperature is around 20 to 30 degrees Celsius, and peat can survive in the pH of 5.8 to 6.8, and which can also tolerate the pH up to 7.2. Then these are some of the cucumbers, zucchini and squash, which can be grown because it can pollinate by themselves, and the ideal temperature is around 75 to 78 degrees Fahrenheit, and which can be harvested in 2 months. But if it is grown in soil, it takes around 3 to 4 months to get harvested. Here, these are the strawberries, which can also, these strawberries can be grown in vertical gardens also, vertical growers, which I've shown you just a few slides back. So, and it also does well in artificial light, and it is very much sensitive to temperature that affects the flowering and fruit sugar. And here the strawberries are very happy to grow because the plants do not have pesticides or fertilizers, and the ideal pH is 5.8 to 6.2. And these are the basils which is growing in this raft system. So this is a picture which is taken by one of our students. So this is a raft system which he has maintained in his backyard. So these are the basil which he has grown. And these are some watercress. And these are some of the vegetables which can be grown in this aquaponics system. And then, so how do we calculate that how much should be the volume? So here, just a very simple calculation, it has to be either 2:1. If the tank volume ratio is typically is 2:1, that is, or it can also be 3:1. If the depth is suppose 2 meters, then the grow beds can be around 1 meter. Or 1 centimeter, I'm sorry, it can be up to 1 meter, or if it is a smaller one, so it can be about 1 centimeters. So, and suppose if it is 1 cubic foot, then it can accommodate around 7.48 gallons of water. And these are some of the scenarios and examples which we have just calculated. So if the depth is 6 inches, then the grow bed can be around 2.8 inches. And then now this is the fish stocking densities. So if the total fish weight is 75 lbs, so number of fish can be kept only 50 and if it is a 20% capacity, then we can use only 10 fish. So basically it all depends upon how big the system is. And fish feeding, so it depends upon the average total body weight. So it can be fed either one time or it can be fed either 2 times by looking at the body weight. So we have to observe the fish eating and also determine the proper amount of feed which is given to the fish. Now we have to maintain the systems it is needed, that is the filters should be cleaned, filter tanks, tubings, the water pumps, the grow bed media. We have to check the cleanliness of things, and sometimes after harvesting, so we have to take out the cuttings so that it is not affected with the bugs and some pests. So we have to look after the pests also. So there are some handy tips and tricks which we usually use it in our grow medias, which we can use vitamin C to air pump to bubble out chlorine and chloramine from tap water. We can also use worms in media beds to break down the solids and reduce the anaerobic zones. And we should never use pesticides or fertilizers like substances in fish tanks or other grow beds. Now, these are some of the handy tricks which I've already said you. So we should go for a common outdoor plants that is that can survive in the shades and not from this, and we should not use that kind of plant which requires a lot of sunlight. And also we should have a power backup always available to work the pumps and the aerators non-stop. So these are some of the web resources which have got the informations, and we are working on this to install the things in the outside part of our university premises, because we already have this in our labs, but we are now trying to get it into our the outside part. So thank you so much. So this is all about aquaponics. Thank you. Hello. 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 Hello, madam. Am I audible? Hello, sir. Sir, ଆପଣ ଅଛନ୍ତି କି ସେଠି?
Hello. Good afternoon, everyone. In fact, first of all, I thank the organizers of Agri who has organized this wonderful seminar, international seminar, and they have invited me to speak on freshwater aquaculture technologies for sustainable livelihood development. In fact, I'm Saroj Kumar Swai, working as a director, SIFA, Bhubaneswar. So I'll just speak about the freshwater aquaculture technologies and for its sustainable livelihood development. And we all know that global aquaculture is on rise, and the capture fisheries is almost like parallel, but the aquaculture is growing. Throughout the world, we can have a look of the slide. Throughout the world, the scenario is different, and the aquaculture is a growing subject compared to the capture fisheries. So we know that we have plenty of resources, that is 2.4 million hectare ponds and tanks, and 1.707 million hectare delict waters, and so many water bodies we have in plenty of resource in India, and for which we can think of aquaculture in a very big way. And if you see the fish production in India, again, the marine fish production is 3.7 million metric ton, whereas the inland fish production again rising to 10.4 million tons. But the present, if you see the fish production system, fish production in India, it is 14.1 million tons. And we have the target of 20 million metric tons in 2025. That is the Pradhan Mantri Matsya Sampada Yojana target. And if you see the projected fish production, we have, of course, SIFA has made a actionable strategies for increasing the freshwater aquaculture production, and this report has been given to action report has been given to Government of India, Pradhan Mantri Matsya Sampada Yojana. And the projected fish production, if you see the present production is 88.9 lakh ton, and the target by 2025 it is 150 lakh ton. This what we have done by sitting together in a common platform with all the stakeholders. It has been found that the additional requirement is 61 lakh ton. Of course, the proposed additional resources has to be increased, and pond area, cage culture, pen culture, RAS, and biofloc. These are all the areas where we can increase the fish production by increasing horizontal land and all this. More number of water has to be increased through pond and reservoir and peri-urban area. Again, we think when you think about the pond, we have to think for gram panchayat pond, we have to think for the cultivable pond of the farmer, and reservoir and peri-urban. When we talk about the peri-urban, we talk about RAS, biofloc, and FIMTA and all this. Now, we can ask that how aquaculture is different from animal husbandry, because the animal husbandry, the terrestrial animal, they are not that complex, because fisheries and aquaculture, mostly fishes remain inside the water and the environment is totally different from the terrestrial animal. But if you see the production system, fish is the lowest, FCR 1.2 compared to the cattle, chicken, and pork. If you give 1.2 kilo of food, you will get 1 kilo of fish flesh. So this is where the economics, which is relatively profitable aquaculture, and there is a lot of market demand and very good consumer preference. Thereby, aquaculture is the one of the important aspect in present day research. If you see the fish availability in India, Indians mostly they prefer Indian major carbs at about 77%, whereas minor carbs, exotic carbs, morels, catfish, and others, they are in a minor group. So major carbs dominating the aquaculture. And if you see the increasing, when you think about the how to increase the aquaculture production, we have four areas where diversification is our prime aim. Number of species we have to add into diversified species, we have to add into the food basket. Then genetically improved breeds, this is again one of the important area where you talk about. Then cost effective feed formulation, this is also another area where you talk about the how best you can, because feed is the maximum expenditure and recurring expenditure, almost like 60% of the recurring expenditure feed cost. Then feed formulations, then proactive health management. These are all four areas where strategies where we are working on and for the increasing aquaculture production. And these are the species technology package available with us. We have about 30 species. We are working on 30 species, and in addition to Indian major carbs, exotic carbs, there are minor carbs, there are scampi, there are morels, there are other species which are also again pangas we talk about, then magur, singi, pabda. These are all technology packages available with us, and the farmers are ready to take all this, and farmers are doing also. So if you go to carp culture, when we talk about the carp culture, we think about rohu, mrigal, katla, and the demand goes for maximum for rohu. When you think about the carp culture, that's polyculture, we achieve 3 to 4.5 tons per hectare with a net profit of 1.4 to 1.8 lakhs per hectare. So this is what generally commonly people they do, that carp culture. When you little again in a additional way, if you carp culture zero stage stocking, if you think of, then again the rohu gets the highest 70% and katla 20% and 10%. With this stocking density, we can get 6 to 8 tons per hectare. Of course, this we can get for 2 crops. That second crop is very much possible. So here there is an opportunity again to stock zero size stocking with a stocking size will be a little bit higher with 100 to 150 gram size. When you think about freshwater prawn again carp polyculture can be added with a freshwater prawn, that is 0.5 to 0.7 per hectare, 0.7 ton per hectare we can achieve. In addition to fish, that's 4.5 tons per hectare. This is again the consumer preference. The scampi being a bottom dweller, it gives a 500 to 700 kg per hectare. So this is an additional income to the farmer. So this also we can think of the freshwater prawn with carp polyculture. Then pangas culture. In northern India, these pangas fetching a better price because there is a single bone. So flesh is more. So it's a high grown species. And we can achieve here highest production 20 to 25 tons per hectare. So this is what we talk about for northern India, especially Delhi, Himachal, Delhi, Rajasthan, and all those areas. Uttar Pradesh also, this pangas culture is very popular, and Government of India is looking ahead to take more entrepreneurs in this area. Then again, magur culture. This is a Indian magur. This is Clarius species, and this magur, it is a small species, but it fetches the highest good price, and we can achieve 2 to 2.5 tons per hectare. This is again the Indian desi magur. If you we here, I must say that there are another magur, that Garipinus species also has come into the some of the farmers' pond, but we do not advocate on this Garipinus species because it is highly carnivorous. But we think about this where there is a little water body, small water bodies with little depth, so we can go for this magur. Again, one of the another subject is the morel culture. This morel also again fetching better price in Telangana, Andhra Pradesh, and again the northern India. 2 to 2.5 tons per hectare production we can achieve by stocking these Chana striatus. This is again the profit we can get 2.5 lakhs per hectare. And again, the lastly, I can say that in addition to the fish, freshwater pearl culture also gaining popularity day by day. We can have, these lamellidens marginalis, these margil is available in freshwater, and those species can be used for the pearl production, image pearl production. You can see the images, how many images are coming up in the mantle tissue, and the so we can go for this freshwater pearl culture. Here I can say that Central Institute of Freshwater Aquaculture is a pioneer to produce, to have the technology of freshwater pearl culture, and now it's gaining again popularity after the Pradhan Mantri ji Man Ki Baat program. So many farmers are there coming. Of course, now we are not able to do the training programs. Nowadays, farmers are coming in virtual mode and they're learning the techniques in we have made a video mode technique. Then one more area is ornamental aquaculture. This is again another lucrative business where a farmer starting from in a very small scale in the rural area, women can be involved in ornamental fish farming with investment of very little investment they can start with, and getting income again. He or she has to increase the land area, and there are varieties are many. So there are domestic varieties. I mean, there are many exotic varieties. There are indigenous varieties, and those can be taken up for the ornamental aquaculture, for which SIFA has has made a action plan for this ornamental aquaculture, because again in Pradhan Mantri Matsya Sampada Yojana is around 600 crores is allocated for the production of ornamental fisheries and its allied subject. When you talk about, in addition to the polyculture and other things, when you talk about the advanced, say, intensive aquaculture system, and we think about aquaponics, we think about RAS, we think about biofloc. These are all again, energy intensive farming system where we require 24/7 energy, electric energy, and with a high stocking density and feed based aquaculture. Here we can do. We can get a better production, subject to you should have availability of 24/7 electricity, and with a limited space. Again, the high stocking density aquaculture can be possible. Nowadays is gaining popularity, and SIFA also we have started this research, and maybe at the end of the year we can have come out with some kind of technology package. Then we have certain FRP portable carp hatchery, where there is not having any construction or kind of space requirement is less here. We can have a solar powered feeder, mobile vending machines. All these gadgets are available nowadays. So those gadgets can be taken up for the production system. Then one of the important area, when we talk about the livelihood development, the aquaculture field school plays an important role. Here, what we do, we choose entrepreneur, those who are having knowledge as well as those who are having resources. And by taking the help of those farmers, we initiate any aquaculture field school. SIFA recognize there are about 20 numbers of aquaculture field school right now. And when we talk about the livelihood development and the production, Pradhan Mantri Matsya Sampada Yojana, again, they have a target to create 55 lakh beneficiaries and many more employment opportunities. Through livelihood program only, we can involve women, we can involve the rural women for ornamental fish farming, pearl culture, aquaculture, seed rearing, and many aspects. women can be involved in production system and livelihood development. Then our strength is genetically improved breeds. In my front, I am my HOD, Dr. Sundara is sitting. He will speak more about these species. And they have developed Jayanti Rahu. Now presently 9th and 10th generation is going on, which is giving 50 to 55% increase in overall growth. This is again one of the good achievement of SIFA, where this plays a very good role. Then we have genetically improved breed like Katla, where second generation is going. We are getting a lot of feedback that Katla is growing very faster than the normal Katla, what it has been come from. And this is again fetching a better price. So people are interested to have genetically improved breeds. Again, genetically improved breeds like Scampi, it's also again plays an important role in aquaculture. Now we are in 10th generation, 20 to 25% increase in overall growth. This improved freshwater prawn again fetching a lot of demand in the market, and many multiplier units are coming. So we are all signing the MOU with multiplier units, and we are providing them brooder seed to the multiplier units so that they can produce more and distribute to the farmers, and the income can be generated and the production can go up. Again, one more subject is that signing barb. This is again an ornamental fish. We have developed a variety, and this is called a golden glittering signing barb. It fetches better price than the normal rosy barb. It has been developed from Pethia conconeus, and now this developed variety again fetching a better price because of glittering gold color, and since the coloration is more, the price is also more. So there is an opportunity for development of different types of brood. Then cost-effective feed formulation. This is our SIFA brood. It fetches a better price, and also again second time re-commercialization we have done it. And farm-made feed. This is again, if the farmer is not having cannot buy also the pelleted feed, he or she can prepare its farm-made food for small-scale aquaculture. Then a lot of things are coming up, like development of fish feed standards for BIS. New Delhi, this is again some feed formulations, farm-made feed, there are all coming up. And proactive health management. This is again a CFAX. It's is a medicine. It's a formulation against epigenetic ulcerative syndrome, which often causes large-scale fish mortality and crop loss in commercial aquaculture. So this is again gaining popularity throughout India. Of course, it has gone to abroad also. In abroad also, the farmers, they require CFAX. So the vendor is supplying everywhere. And Immunoboost C, again another achievement we have, which enhances the immune system of the fish. Then SIFA Cure, one of the again medicine, a formula for ornamental fish for fungal and bacterial diseases. And then our strategies, there are about 9 strategies we have. First of all, I must say that existing the fish production, we have to enhance from 3.3 ton per hectare to 5 ton per hectare, and emphasis on seed, which is a major input, seed production and supply, and incorporation of 50 to 20 diversified species in the aquaculture system, cost-effective feed technology, quality diagnostic and vaccines, affordable farm mechanization, and climate resilient aquaculture technology. This is what we require nowadays. Aquaculture is a major option for livelihood and nutritional security and digital technology. I said SIFA research strategies are major 5 categories. That's aquaculture diversification and climate resilient aquaculture, then breed improvement for growth and disease resistance, and then feed development and feed technology, then aquaculture farm mechanization, automation and gadgets. That's what we are trying to develop through smart aquaculture, that precision agriculture, under the precision agriculture, under the ICR, and we are trying to develop the smart gadgets, which is sensor-based gadgets, and fish health management through different, through molecular diagnostics. And one of the important subject, I mean, it's Android app, Matsya Setu. This is a virtual learning app we have developed here. During COVID, the farmers have 30,000, stakeholders. They have downloaded this Matsya Setu. It's an Android app through which the virtual training program is going on. I mean, they can learn the modules, they can learn the different lectures and they can go through different lectures. They can get a lot of questions. They can get a feedback also to the scientists. And a lot of things have been done through Matsya Setu. And we are also trying to develop Matsya Setu 2, where we are giving all emphasis for different stakeholders to join there, because there are seed growers, there are feed manufacturers, there are other stakeholders. They can come together and they can discuss themselves, and they can also improve their business and all this. So Matsya Setu is coming, it is already working, and I request everyone to download this Matsya Setu if you want to know aquaculture. So I will not speak more about this because time is also short. Thank you very much. Our slogan is grow fish, grow with fish. Let us do it together. Thank you and Jai Hind. Any questions are there? I can. No. Okay. Thank you very much. Thank you so much. Okay. Okay. Thank you so much. Can I remove it? Okay.
So thank you so much, sir, for a very brief presentation of yours. And thank you for finishing this on time. So now I would request Dr. Ashok Kumar, Director ICAR NBPGR New Delhi, to come on the podium and talk about the topic unlocking potential of Genebank collections for climate resilient agriculture. I hope this would be a fruitful one for everybody here. Let's clap for the gentleman. Thank you.
Hello, may I start now? Good afternoon. I am Ashok Kumar. I am from NBPGR, working for the PGR since last 35 years. So I would like to share my view about the unlocking of the gene bank potential for climate resilient agriculture, and I will also cover the nutrition security. So yes. So first of all, agriculture. So what are the paradox of the today's agriculture? Yes. So what are the today's agriculture paradox? As you know, in Indian this center, we have around 3 lakh species, plant species all over the world. And out of that, only 1 lakh is being used for the humankind. And out of that, only 30,000 species are edible. And of that, 7,000 species are being utilized for local food purposes. And out of 7,000, 120 species are the importance for the commercial purpose or the national scale. And 30 crop species that provide the 90% population calorie for their needs. And out of 30, only 3, that is the wheat, rice, maize, that fed the calorie of approximate 60% of the population of the world. So you can see out of the 3 lakh species, only 3 species, that too rice, wheat, maize, are important. And so you can see here the narrow downing of this genetic resources from 3 lakh to 30. We are dependent only the 3 species. So as per our different targets, biodiversity conservation, sustainability. So we have got the sustainable development goals, national biodiversity targets, that is Delhi Declaration, and IT targets. How to fulfill all the targets? These are the parameters: the food for all by 2020, doubling farmers' income, and the climate change. My talk is related to climate change. That is the main specific to deal with this genetic resources and climate change, that too under the biotic stresses, and how to develop the resilient cultivar through utilization of the broad sense genetic resources, and to broaden the genetic base to deal with this vagaries. So as you know, there are 17 sustainable development goals and 169 targets. So we are the signatory. We have to follow and we have to fulfill the agenda by 2030, the sustainable development, by using our own system. Then out of these, the 3 or 4 are very relevant to the plant genetic resources, that is the PGR, that is the no poverty, the zero hunger, the good health for well-being, the rationalization of consumption, and the climate action, the life on land. So the mainstreaming of the biodiversity for increasing farm productivity and reduction in input, that will deal with the zero hunger and no poverty. Similarly, the improvement of the nutrient content in plant genetic resources, and the development about the varieties, or you can see the fortified varieties. Recently released 35 fortified varieties to deal with the hidden hunger and also deal with the good health. Similarly, for the issue of sustainable production, we are responsible for the consumption, and we have to deal with the climatic change. There are so many climatic changes. There are new pests emergence are coming, and abiotic stresses are also coming. So we have to deal with all these climatic vagaries. And then how can the targets can be dealt by the 2020, that the national biodiversity targets, that the Delhi Declaration has been first ISC was organized, and to deal with the national biodiversity targets. So we have developed the Delhi Declaration. And what is Delhi Declaration? The complementary conservation strategies, the awareness of the capacity building, the exchange of the biosecurity, the frontier technology, and the traditional knowledge, the participation of private organizations for the society. So how to deal with the conservation, sustainable utilization of the agrobiodiversity for the humankind, and also for the nutrition security of this system. As you all know, because you all have the botany background, so India is one of the major center for the center of origin, having 12 mega biodiversity centers. Out of 34 hotspot biodiversity, the 3 or 4 are located in our Indian subcontinent. These are the Western Ghat, Eastern region, and Northwestern region. Similarly, the Western Ghat is the hotspot not only for the plants, that is only for the animals, fish species that are endemic to that region. Similarly, the 162 breeds, when we come to the animal genetic resources, the 162 breeds have been domesticated in our country, particularly in the Northwestern region. Then we come to the center of origin. Out of the 8 center of origin, Hindustani center, Indian genome center is one of the center for the origin of the so many major crops like rice, mango, banana, so many gourds like cucumber, Then this chickpea, the secondary center, similarly the eggplant, sugarcane, citrus. In any region, citrus is having the major diversity for the citrus genetic resources. Then we come to the PGR, plant genetic resources. What is PGR? The sum of diversity available in the form of individuals, in the form of genomic resources. So PGR is the backbone of the agriculture. NVPGR is highly involved for the serving of the nation, achieving the sustainable development goal, sustainable food security since 1976. So NVPGR has the 5 major divisions, that is the germplasm exchange, there is the conservation, that is gene bank. So I'm going to come to the gene bank. Then the development of genomic resources. Then the characterization evaluation is one of the major division for the handling the evaluation of the germplasm, for identification of the traits for the future breeding program. And we're also dealing with the services like exchange services and the quarantine. No insect pest can enter our research system, because we are there to help the research system in the import of the germplasm, that is the pest-free germplasm from different countries. So India or NVPGR had got the collaboration with 80 countries for the exchange of the germplasm, and we are very, very thankful to the quarantine division for handling the quarantine facility in a nice manner to enter the pest-free germplasm in the country. This is the mandate of germplasm, the acquisition and management of the indigenous and exotic genetic resources for agriculture. These are the major 5 objectives. The first one is the exploration collection. So my 2 scientists are attending here. They belong to the exploration collection. So we are collecting germplasm from all over the country, from north to south, east to west, and for the purpose not only NVPGR headquarters, the regional stations located in different parts of the country, they are involved for the collection, characterization, evaluation of the germplasm. NVPGR regional station Cuttack or NVPGR West Center Cuttack is one of the center is identified for to collect the diversity from the eastern region, that is from Bihar, Bengal, Odisha, and to also help in the evaluation and characterization of the germplasm. Then NVPGR is working in a network mode with the help of the regional stations. These 10 regional stations are located in different agroecological conditions like Srinagar in temperate conditions, Simla is also in the temperate conditions, Bhuali the mid hills region, then Shillong in the NEH region, Jodhpur in the arid conditions, Ranchi in the subtropical conditions, then Cuttack eastern region and subtropical humid. Similarly, Hyderabad for the semi-arid tropic conditions and subtropical conditions. Some of the these 10 regional stations are involved for all the PGR management activities, that is germplasm collection, that is exploration, collection, characterization, evaluation, and also for the conservation in the form of the field gene bank, or if there are seeds, then in the form of MTS and the working collections. Then the evaluation of NVPGR, as you know, the NVPGR was established in 1976 with the visionary Dr. H.B. Singh. He is known as the Indian Babylon. He was the explorer. He made the exploration missions for all over the country. Then the second decade, the 86 to 95, that was the infrastructure development. The Gene Bank was established with the help of the Indo-US program and also for the Indo-UK program. Similarly, the new facility was created in that one. That is a tissue culture repository, the cryo facility for the non-orthodox species. From 96 to 2005, the modern gene bank was inaugurated with having the 12 modules, and a major emphasis was giving for the collection involving the NGOs, the State Department, the Forest Department, the Central Government Department, the National Agriculture Technology Project for Agrobiodiversity at the NATP. So the program was launched in the mission mode approach, and with the help of the around 120 partners from different backgrounds, we have taken the mission of collection in different crop, including wild genetic resources, particularly from the remote areas, involving the NGOs. Then we have also identified during that period the national germplasm active sites and we have provided the MTS facility to all the centers or all the crop institute. They are dealing with the germplasm. Then during the last 10 years, 2006 to 2016, that is the unlocking of the gene bank potential. That is my title. And here we have taken the large scale characterization, the characterization of the major crop to grow the material one goal and identify the grouping the material, categorize the material, then development of different sets. So through the characterization data. Then we have taken the multi-location evaluation, because if you want to unlock the gene bank potential, collection of the potential of the gene bank material, then you have to identify the value of the germplasm. That is not possible in a single way. That is using the multi-location data and multi-location evaluation. Then we have also taken the basic research through NAIP, and we have developed the database using the PGI informatics. Then during last 5 years, we have developed the gene bank, which was established in 2006, but modernized in 2017, and we have also developed the high throughput genomics for the development of genomic resources and the identification of the trait-specific germplasm and utilization of the land races and crop biorelative for the pre-breeding, because we want to utilize the potential of the crop biorelative. It is not possible directly. That is possible only through the pre-breeding program, because we have to develop the pre-breed material, and as NVP are unable to develop the variety, but the material is being developed by the NVPR, being shared to the any national partner, even private industry, for the sustainable utilization of the germplasm and utilization of the gene bank material. So these are the activity of the NVPGR and the activity of the breeders. So the collection, characterization, and the identification of potential donors. These are the activity of the NVPGR, or you can say the NVPGR curators. And our clientele are the stakeholders, are the breeders. They want the material, and we provide the material to the breeders. And this is my purpose, to see the gene bank material and see the potential of the gene bank material, to evaluate them and provide to the breeders for the utilization or the better development of their needs, maybe the biotic stresses, or for the different nutritional parameters. Then we come to gene bank. So we have the three kind of gene bank, like NVPGR, the seed gene bank, that two modules are there, the medium-term storage and long-term storage. So national gene bank constitute the long-term storage for the seed gene bank. Then the cryopreservation, minus 180 degrees for recalcitrant seed, and the in vitro, there is tissue culture. That is for the in vitro for the tissue culture gene bank. It is for the vegetatively propagated species like taro, banana, or similar kind of the plant which cannot produce the seed, but they are important to conserve. Then we have also identified some of the national germplasm sites, because NVPGR cannot deal with all the species. So we have identified 87 national germplasm active sites for different crop plants, particularly for the horticulture plants. Then now we have taken initiative for on-farm conservation with the help of our partners. So we come to the national gene bank. In national gene bank, we have more than 4.6 lakh accessions belonging to different species. These are the cereals, millets, forages, pseudo-cereals, legumes, oil seeds, fiber, vegetables, fruits. There are so many plants dealing with this. Around 120 species are being conserved in national gene bank. So you can see the number of accessions. More than 1.6 lakh accessions belong to the cereals. Similarly, 5,59,000 accessions to the millets. So the millets and legumes are the major group after the cereals. Then other than this crop germplasm, we also have this the germplasm in the form of crop-wise relatives, because now the crop-wise relatives are getting very, very important, because these are the species or the wise relatives. They have the potential genes for biotic stresses, abiotic stresses, but they are very difficult to utilize due to their breeding behavior or their biological behavior. Then we come to the in vitro gene bank, as I told, the different kinds of plants like tropical fruits, temperate minor fruits, tuber crops, bulbous crops, medicinal aromatic plants, spices, and these kind of plants which do not produce the seed, but they can be propagated by different means, propagules or vegetable propagated means that can be conserved in the in vitro gene bank. So far, we have conserved around 2,000 accessions belonging to the different species. Then we have also got a facility of the cryogene bank. In cryogene bank, we have around 12,000 accessions belonging to different the recalcitrant seed, because the recalcitrant seeds are those seeds which cannot be dried like orthodox seed. They have the moisture. They cannot be conserved in the -20, but they can be conserved in the cryo conditions through their different means like pollens and other seeds material. Then we come to the large scale characterization or the genotypic characterization and phenotypic characterization of the gene bank, because in gene bank what we are having, that is not the potential of gene bank is not known unless until that have been characterized or that have been evaluated thoroughly. To deal with this problem, to unlock the potential of the gene bank, we have taken initiative to develop a mega project for the characterization evaluation of different crops. So we have got a funding of around 300 crore from the Department of Biotechnology for the minor pulses, the CCM, the linseed under the minor mission project, safflower, Niger, and rice, wheat, chickpea, and grain amaranth. So through this program, we are developing the genomic resources, the characterization data, and also the developing the reference set and the trait specification plot. So we, in a nutshell, we want to tell about last 3 years. We have characterized around 50,000 accessions during the last 5 years. Then we have also characterized, evaluated for different biotic and abiotic stresses. Then we have taken large scale characterization in case of wheat, and 20,000 accessions have been characterized. The core sets have been developed, the mini core sets have been developed, and these core set and mini core set have been evaluated, and very good material in the form of reference sets have been developed through this program. Similarly, in case of chickpea, we have characterized around 14,000 accessions, and we have developed the core set. Core set means capturing the diversity, whole diversity, having the 10% accessions. Then similarly, during the last 3 years, we have characterized and core set developing different crops, wheat, linseed, rice, bean, cowpea, and we have also taken this characterization of sesame germplasm, and we have characterized 5,000 accessions of sesame. We are in position to developing some of the core set in these also. Then we come to the minor oilseeds, the sesame, Niger, safflower, and four crops have been thoroughly characterized, and we are also going to evaluate the accesses of the land races of the major crop. Then we come to the through characterization through the institute program. We have characterized the pea germplasm, the maize germplasm through CRP, and we also under the evaluation of the bale, bear, amla germplasm, so many germplasm. Then we also initiated the utilization of the crop by relative, because these are the some of the species, they have the genes for example okra for YVMB, then lentil for rust powdery mildew, vigna for YVMB, and similarly chickpea for fusion build. So we are going to utilize the wild species through this means. Then we've also characterized our germplasm for the biochemical parameter, for the phytochemicals, and we have identified some of the potential germplasm having the nutritional parameter. Then we come to the biochemical parameter characterization. We evaluated rice having good amylose content and the protein content. Similarly, in maize, we have identified some of the grain maize having more than 16% line. Similarly, the linseed, black gram. So we are in the position of the evaluation of the material, developing the trait specific germplasm, and the utilization of the trait specific germplasm through through the process of the registration, NVPGR and ICR has also got a mechanism to register the trait-specific germplasm. So I request to the young breeders and to the information of the our scientist faculty. We have a mechanism to register the germplasm. If you have any unique material, you can register the germplasm with the ICR mechanism system. We have registered so many accessions having the good rates that have been utilized by different means. We have also developed the mapping populations, then developed the genomic resources through the genotypic characterization. Because nowadays, now the genome resources are very, very important for the utilization. And after phenotypic characterization, we are characterizing germplasm through genotypic means, and we have developed the molecular code using the genotypic data, and we are also in the position to handle with this genomic resources and to conserve the genome resources at NVPGR. Then we come to the utilization in a real manner. We have identified some of the germplasm and have been utilized in the varietal development. Then some of the varieties have been released using the germplasm like cluster bean, coriander, and this. Then we have got the mechanism of the knowledge management unit. We have developed the passport database, the import database, gene bank database. So by searching the website, you can download this databases and you can search the material, what kind of material you want, that will be made available from the NVPGR. So we have developed 6 databases, 10 applications. You can use the app and also the 4 mobile apps to disseminate our knowledge of characterization data, export data, evaluation data, and this is the passport data for the utilization of by the breeders, by the stakeholders. With this, I am thankful to the organizer to giving me a chance to be here to share my views about the PGR, NVPGR, and also I am thankful to our colleagues from different centers and from our own institute for helping me to giving this talk. Thank you.
Thank you so much, sir. So now I'm calling for Dr. M. Nedu Cheziang, Head ICR CT CRI Regional Center. He'll be talking on the topic entrepreneurship, development in tuber crops, value addition. Is he here?
Okay, 15 minutes, I will finish, madam. Don't worry. It's not coming. It's not coming. Yes. Good afternoon, everybody. I am from Central Tuber Crop Research Institute. I am talking about the entrepreneurship development in tuber crops, value addition. We know that we are at present having whatever value addition from fruit crops and some of cereals, but we don't have any product from root crops. But it has ample scope for development of value addition through which going for entrepreneurship development. And tuber crops are, we generally refer tropical tuber crops, and other one is a temperate tuber crop. Potato is considered as a temperate tuber crop. Other than potato, all are tropical tuber crops. Among tropical tuber crops, cassava is said to be the main crop. It is widely, extensively cultivated in South India, part of Odisha. In Odisha, especially in Koraput, Rayagada district, it is cultivated. The second most important crop is sweet potato. Sweet potato is a very popular crop in eastern India, especially Odisha. In Odisha, around 40,000 hectares it is cultivated with the production of around 4 lakh tons tubers. The third group is yams. Yams, different species are cultivated in Odisha and elsewhere in India and world. Yam, Dioscorea alata, Dioscorea esculenta are both the species are very popular in India. Apart from that, in tribal areas of Odisha, Dioscorea bulbifera is a very popular tuber crop. Then a group of arid crops also cultivated in Odisha and India, especially elephant putium, very popular marketed, and fried product snacks item also we can make. These are the fried items. These things are available. I have kept in my tuber crop stall is there, CTCRI stall. You can go and purchase. All these products are available here. I have kept a stall here, CTCRI stall. These are some of the things, fried snacks and flours, fried items. Bread, you can make bread and cakes also from this cassava flour. We have in our center, we have all the facilities for making bread and cookies, even pasta, noodles, whatever you say, that all those things we can make with flours of any flour, whether it is a wheat flour or ragi flour. But we advise entrepreneurs to come and develop their own product by using tuber crop flours. And you can use starch also. So these are cookies we can make from tuber cassava rava. Then industrial products is a very popular product. Already it is there in the market, sago. Sago, people will be thinking sago is prepared from sago farm. It is not like that. It is coming from starch. And especially cassava starch is used for sago preparations. And the sweeteners also, we have developed glucose and fructose syrup can be developed from the starch of cassava. It's a modified starch. And biodegradable plastic, we have the technology developing, producing biodegradable plastics. And the starch-based adhesives, it is coming up a large extent, whatever you are finding in the market. Fevicol, all other things are coming from starches of cassava only. And coming to this is a board, particle board. This board is coming from cassava stem. It is a cassava stem particle board. And biopesticide also we are developing from the cassava plant, especially cassava is having biopesticide properties. And we have developed 3 products from cassava leaf. Cassava leaf is a byproduct and which is not used for consumption purpose. That can be used for biopesticide production. And these biopesticides are very effective against aphids, mealybug and borer pests. And we have developed Shreya, Nanma, Menma. These are the 3 products we have developed. We have developed and we have tested in coconut farm and other banana and others for weevils. And papaya mealybug, it is a very big problem in many areas. For that also we have used this biopesticide. It's a completely, this is a biopesticide unit from us. From this unit, we have used to develop biopesticides. Coming to sweet potato, just I am going quickly. These are 2 varieties I want to mention. One is ST14. Now we call it as Bhuswana and Bhukrishna. It's a very rich in nutrients. Anthocyanin very rich, 95 milligrams in case of Bhukrishna. And the other one is beta carotene rich Bhuswana. These 2 varieties I have kept in my stall also. You can take. This is very good for health. And you can develop, as I said, that sweet potato is in India as are now consumed only boiling and taking, and you can develop it into powder. And this powder, like wheat powder, you can use it in making leaven bread, unleavened bread, like making chapati or gruels, whatever you want to make, you can make. And it is very rich in nutrients. See, this tuber sweet potato you can consume in 1 month only. But if you make it a value-added product like flour and other form, you can keep it for a year and you can consume the benefit of, you can get the benefit of sweet potato. So this is the way, some of the technology we have demonstrated to tribal farmers making chapati and roti. These are some of the value-added product. These are chips prepared from different. This is a pasta prepared from sweet potato flour. This is a functional pasta. This is using beta carotene rich yam and sweet potato. So this is pasta just added to the flavors. That's all. And I want to tell it is a low glycemic. As I will discuss with you, if some information is required regarding glycemic index and dietary fibers, gulab jamun and other things also you can prepare. And try to consume at least tuber crop once in a week. It will give a strength to human health, especially colocasia and elephant putium, if you take once in a week, your body, your joints pain will not be there, arthritic problem will be solved. And try to consume sweet potato and try to take arrowroot powder, palua powder. It's good for health. Thank you very much. Thank you for giving me this opportunity. Just a bit. Thank you.
Thank you so much, sir. Proceeding further, I would request Dr. Jitendra Kumar Sundaray, Principal Scientist, ICAR CIFA, to join me on stage and talk about the topic genetics and biotechnology to augment aquaculture production and food security. I hope you are there in the room, so we can definitely clap for the gentleman, all of us together.
Sir. Hello. Anyone? Eh. Very good afternoon. Sorry. I have given only 15 minutes time. I'll see that like I'll cover within that time actually. So now coming from yam, let's move to a little bit about the fish and prawn. So this is my topic is given to genetics and biotechnology to augment aquaculture production and food security. If you look into food security, we have been like listening since morning, and we had a very good presentation from NRRA Katak. But it's not at the regional level or local level. Globally, people have realized what is the importance of the food security and how genetics and biotechnology is playing a role. Even mornings, like Professor Tripathi talk about the photosynthesis. That was clear indication of manipulating photosynthesis. How can you give a better plant health? So this is about like how global is looking into that. So my presentation will be aquaculture status and constraints. I'll be taking less because my director has covered most of the things. Hunger and food security, fish genetics and fish biotechnology. What is to be done to achieve maximum from the aquaculture? We have 4 regional center because what the director has missed, I am going to talk about that. So we have a center in Bangalore, which is talking about mostly peninsular carps. And we have center in West Bengal, we're talking about integrated aquaculture. And Andhra Pradesh, Bijwada, we talk about the feed-based aquaculture. And we have a newly opened center that is Bhatinda, Punjab. We talked about intensive carp-based aquaculture. Of course, a lot has been talked about, director mentioned about PMSSY. Even our Honorable Prime Minister has talked about how blue economy or even the product from aquaculture can play a major role in Indian economy. And some of the review paper has been already published. Again, coming about Director NBPJ talked about MSDG. As far as our institute is concerned, we are concerned about 4, mostly no poverty, zero hunger, climate action, and life below water. Life below water, what talking about, it is fresh water, brackish water, marine ecosystem. And of course, another, this is talking about quality education, number 4 SDG. Even we are not directly involved with education of any system, but we have been imparting a lot of training program, whether it's the students as a thesis or other program. And if you look into National Mission for Sustainable Agriculture, this is government has initiated, and if you look at everywhere, improved crop seeds, livestock and fish culture, water use efficiency, adaptive agriculture, improved farm practices, many of the things has been covered, and it's going to be covered within another 2 days. Again, agriculture productivity and farmers' income. If you remember, it was decided to have a doubling farmers by 2022. The morning, Dr. Mukherjee was talking about how far we have realized, we don't know, and we don't want to make comment on that. The point is, but you should have a continuous improvement of your technology by which farmer gets more productivity, and the entrepreneurs get more benefit out of that. Poverty and food security. Sorry. So the definition, I'm not going to talk about that, but food security is a condition when all people at all times have physical and economic access to sufficient, safe, nutritious food to meet their dietary needs and food preference for an active, healthy life. It's a very simple. If you look into the food security in Bangladesh, everywhere the farmer, entrepreneur, anybody, if you are having a 3 staple diet per day, they take it as a food security. So this is very clearly mentioned. Again, now MB Gupta, who is one of the most renowned fisheries expert across the globe, he made a statement about, like, this is the greatest disease of all humankind is hunger. And if you look into this Indian hunger index, we are not in a better position, 27.5. If you look into this yellow, the cover, this yellow color of this Indian map, actually, we are not even compared to Bangladesh, we are not that good actually. And if you look at Global Index India 2020, and this is the latest one, you can see that what are the percentage of the child undernourishment? What are the like pregnant women are under undernourishment? So there is a need, there is a presence has been felt, there is a hunger, there is a malnutrition. So we need to look into that. Of course, agriculture, animal health, everybody is working on that. So I will be mostly focusing on the aquaculture and fisheries. Okay, how to overcome this malnutrition and how aquaculture can improve the livelihood. So you mean to disclose? Okay. And okay, I don't know how for another 10 minutes, 5 minutes, I'll finish it up. Okay, so again, coming back to nutritional value of different meat product, look into salmon, carp, tilapia, chicken, beef. If you look into this, among fish, 350, you have a omega-3 fatty acids coming from the carbs only. Not looking into salmon, I am taking about Indian context. And why fish is globally important? Again, no need to tell much, but only thing, fish can give a lot of minerals and the feed materials for the populations. Again, if you look into the all proteins, whether pulses, mutton, cereal, meat, fish, fresh fish, the growth rate is quite substantial. 5% growth, if you're not comparing with the egg and poultry. So what aquaculture is giving, if you look into this value chain, aquaculture can give you every value chain, and you can have maximize your profit actually. So if you look into this particular graph, India map, how India eats, the red circles are more about non-vegetarians and the green circles are more about vegetarians. So if you look into that, almost all eastern part, even you look into J and K, other parts. So mostly prefer into the non-vegetarian food. Okay, I'm not going to reveal this one. What I'm going to talk about, Asia rules in the aquaculture. You have like maximum number of the species. And 89% of the world production in Asia, look into China, Indonesia, Thailand, Vietnam, and Bangladesh. Norway hardly 2%. Okay. If you look into the yield gap production as far as the India is concerned, you can look into the yield gap is the brown color and the blue color is yield actually. So every zone, whether it's a central, northeastern or western zone, you have a chance of to enhance your production to a different level. Okay, what are the constraints actually? First of all is like, disease outbreak, feed cost, inbreeding of the populations, management practices, climate change, slow growth, and the outdated farming system. I mean to say you need to move forward regarding Dr. Yashaswi Naik was talking about aquaponics and RS. So there is a need to go for the new farming system actually. So what are the issues Okay, director has talked about that. Our focus species are like this. Left side, if you look, these are the species where we have taken some of the selection program. Again, the point what I'm going to talk is how we have harnessing our research to make the difference. We have a genetically improved Rohu, we have genetically improved Katra, then we have like in a lot of whole genome sequence and biotechnological resources. Then another is hip-hop root, which director talked about. I'm not going to talk on that. So what I'm going to, you will look into genetic improvement program in India. First is Jayanti Rohu, which we are doing, then Scampi, which we are doing. We have taken the only two traits, growth as well as disease resistance. Okay. Scampi, we have taken harvest weight, carapace length, standard. If you look into the, at the end of generation, everywhere you have genetic gain, 17%, 18%. It means from normal Rohu, it gives a better growth rate actually. And if you look into this genetic improvement program in the world, actually, look into the salmon and the trout and common carp, tilapia and catfishes. If you look into every genetic improvement program, they have taken a different trait, which is growth, disease, fillet yield, everything. And this is what director was talking about. Already, we have a 33% higher genetic gain compared to the normal Rohu. And the benefit is the reduction in the rearing period, increase in the yield almost 24%, and the relative reduction of the cost 20%. You can know it can be easily farmer friendly. Katla at the second generation, we have a 15% genetic gain. And if you look into the comparison of this Rohu Katla in a polyculture pond, you can see the BC ratio. It's 1.7 when it is with improved Rohu. It's 1.4 when it is without improved Rohu. So this is what Director talked about. Just let me talk about a global scenario. This is a species called tilapia, which is almost ruling the world as far as the world is concerned. And the challenges are there: domestication, tackling genotype and genomic resources. Again, gene editing, scientists from NRI talked a lot. What I'm going to talk about, this is the first commercial product, genome edited tilapia. So what the benefit is, 70% significantly improved the fillet yield. Wherever go to hotel, you see the basa. This is all about the fillet. So because of genetically modification, we can have a 70% genetical yield, 16% growth rate is improved compared to the normal, 14% feed conversion efficiency, and shortening the feeding time. That is more important. If you can get a lesser feed, more weight, that is much more beneficial. Again, with me and my other colleagues, we have started working on the CRISPR-Cas9. Since morning, she talked a lot. I'm not going to talk. So we are having a program within Norway and Australia. We are working on this CRISPR-Cas9 mechanism. Again, you have a lot of beautiful fishes which you can have a color customization. So by applying CRISPR-Cas9, we are already working on to giving new fluorescence to the different ornamental fish. And this is again, we have created or we have established artificial sperm from the magur by which you can go for a fertilization. Again, this is one of the best thing I just want to tell you. Just look into the large fish and the small fish. The small fish attained the same size weight in a large period, but the larger fish by gene edited, it has attained a, if you look into that, standard salmon gets around 4 kilo at the time of around 850, where genetically edited, genetically modified, it gets around 6 kilo in less than 750 days. So this is what the power of technology is. And they almost, of course, genomics is played a major role in the health management. If you look into the precision technology, we have been working on few diseases, any virus or this Aeromonas hydrophila. And again, this is, this particular tilapia was infected with the tilapia leg virus. So by application of genomic technology or genomic science, they could locate which is the chromosome is responsible, gene is responsible for this tilapia leg virus. So they could knock out. So now they could get it a disease resistant to tilapia leg virus. This fish can brought it. And again, genomic selection, I'm not going to talk much about that. Again, one new things you all might be knowing about this surrogacy in case of human being, right? So we also have been quite successful in the surrogacy technology. It means a larger fish can be brought through a small fish. So my last slide only. So if you just plan it, let me talk about this Rohu. Actually, if you look into, we have applied like genetics, genomics, epigenetics, metagenomics, transcriptome, transgenomics So this all we can combine, and this is what the team is working at SIFA to maximize a production from a species like improved Rohu. And again, just few things. We need to increase the breeding capacity of individual species and add number of species to can be bred. Again, doubling fish production through diversification, global food security through genomic approach, Cas9, what morning NRRA talk, now we also talk. Optimizing water use and increasing water productivity, science communication for enhanced information. This is what we are lacking actually. The science need to reach to the clientele actually. And integration of fish countries nutrition program, mid-day meal scheme. So that is a requirement, we need to do it. And finally, like, future of the fish, this was somewhere in the nature. Aquaculture has the potential to sustainably meet the gap between supply and demand for aquatic foods. Same day, it has the potential to lift the substantial number of poor people out of the poverty. Needs clarity, clearly articulated research agenda, police support to the program. So like director mentioned, so thank you. And we go with a slogan like grow fish, grow with fish. Thank you very much.
Huh?
Check. So sorry for keeping you waited. We are all set to start the beginning of the inaugural day of our entire session. So we have had some fruitful sessions throughout the day. I hope you enjoyed this. और अब फिलहाल हम पहुंच चुके हैं उस सेगमेंट में जब हम आज के दिन का इनॉग्रेशन करेंगे, पूरे इवेंट का इनॉग्रेशन करेंगे. So we have our special guest over here. So one by one, I'll be requesting them to please join us on the podium and do the honors of lamp lighting, because as I say, any beautiful thing should be started by seeking the blessings of the Lord sitting above. I would really want everybody to smile a little. It's a good thing for us. We are inaugurating something. I would want everybody to clap. First and foremost, our chief guest who will be joining us virtually, Dr. Trilochan Mahapatraji, Honorable Director General ICAR, Secretary DARE. Let's have a wonderful round of applause for the gentleman for making some time for us. Our guest of honor, for today, Dr. Jai Krishna Jainaji, Deputy Director General Fishery Science, ICAR. Please have a wonderful round of applause for the gentleman. Dr. Ajay Kumar Parida, Director, Institute of Life Sciences. Professor Sanjay Kumar Nayak, Vice Chancellor, Ravensai University. Professor Pawan Kumar Agrawal, Vice Chancellor, OUAT. Dr. Padmini Swai, ICAR Director and NRRI. Dr. Ashok Kumar, Director, ICAR and BPGR. And Mr. Divyanshu Prasad Das, Program Director, Agri. We also have a gentleman representing Ruchi Food Line. I would request all of you to please step forward and do the honors of lamp lighting first. And all of you, let's have a wonderful round of applause. And also to the students, to the people watching this, I would request to please stand up for a while. Jai Jagannath kariba tapare amaro inauguration kariba because amaro inauguration to emti hi hue, right? Yes. Thank you so much, everybody. Let's say this together. Jai Jagannath. Thank you so much. So since we have already taken the name of the Lord sitting above, and I believe he would be blessing each and everybody, whoever is sitting under this roof, and everybody around the world, across each and every corner. लेकिन जब उनकी बात होती है तो उनके यहाँ से कुछ हम चाहेंगे कि आपके सामने प्रस्तुत हो. I heard that a lot of people are here from different state. It's an international conference, so definitely people are from different corners of this country. So since all of us are here, we would love to start this evening off with a beautiful presentation, which somehow the art from somehow has started from this state itself. People know this state because of this art form first, and then the language. It's said Odissi. It started from the temples of this state, and now it is critically acclaimed across the country. All the people, just not just in this country, rather internationally also love this art form. So let's start it with an Odissi number by Nrityanjali Dance Troupe. Let's have a wonderful round of applause for all the performers here. Thank you.
विनाशक
So that was a beautiful performance to start the session with. So now I'd be requesting all our guests of honor to please take the seat over the dais, and then let's begin the evening off. First and foremost, Dr. Joy Krishna Jaina ji, Deputy Director General Fishery Science, ICAR. Dr. Ajay Kumar Parida, Director Institute of Life Sciences. And I'll request my lovely ladies to please felicitate them with the flower bouquet. Thank you for taking time for us. Professor Sanjay Kumar Nayak, Vice Chancellor, Revensa University. Professor Pawan Kumar Agrawal, Vice Chancellor, OUAT. Dr. Padmini Swai, Director, ICAR, NRRI. Dr. Ashok Kumar, Director, ICAR, NBPGR. And of course, we from the entire team behind Agri Vision 2022 thank each and everybody over the stage who could give their time out from their busy schedule and be here for us. Request. Okay. I would also request Professor Vaishnav Charan Tripathi sir to join us on the stage and be here. As always, he's always a guiding angel for us. Sir, please join us on stage. Okay, since the evening is very special, I would definitely love to hear from everybody sitting over here, at least for 2 minutes. But before that, let's start from our chief guest of the day, Dr. Trilochan Mahapatra, Honorable Director General, ICAR and Secretary, DAR. Let's have him on screen. No lectures now, sir. Let's just speak. Do we have lectures? Chair मत उतारना. Then no need of. Yes. No, it's okay, sir. If you want to watch from there, then it's okay. Otherwise, it's okay. Yes, let's. Please start.
Who is going to speak?
Sir, first and foremost, we would love to hear from you.
Okay. What is the time? What is the time available?
Yes.
How much time is available?
10 to 15 minutes, sir.
Okay. Yeah. Thank you very much. All the important dignitaries on the dais who are present. Dr. P.C. Tripathi, former Vice Chancellor of Revensa University. The current Vice Chancellor of Utkal University, Vice Chancellor of OUAT, my colleague Dr. Jana, Dr. Ashok Kumar, Dr. Padmin Swai, and others who are present today. I apologize for not being there physically, and but it's a very happy occasion that many stalwarts are present there, and time is limiting, and there are so many to actually speak about the agriculture for sustainable future. Agriculture is the mainstay of our civilizational needs. Civilizations perish if agriculture perishes. It's because food is the essential need. And without food, certainly we can't exist. More so in India. India with 1.3 billion plus per population needs agriculture more than any other country. 70% to 80% people living in the rural areas. So it is not just the food needs of the rural or urban population. It's also the livelihood security of 70 to 75% of people in the country. That depends on agriculture. Rural development, when you talk about, it's primarily agriculture that matters. More importantly, engaging such a huge population, almost 50%, directly or indirectly, has been a challenge and remains a challenge. I do not have any formal presentation. I'll speak for 10 minutes and then finish it up. When we talk of a sustainable future, we have to keep these facts in mind, that sustainable future of India would depend upon how best we deal with this huge population, large chunk of it being rural, their livelihoods, the food security of the country in totality, and through food, the nutrition security, which has been very, very tough, and that impacts our standing in the Committee of the Nations at the global level. The future also depends on how best we are able to address the challenges which are here today that we are facing. Challenges of climate change impacting agriculture in a big way. Challenges of diverse, complex socioeconomy that is prevailing in this country. The challenges of small and marginal farming situations. Close to 80 to 85% farmers being of this category. The land being fragmented, less than 2 hectares and fragmented. The natural resource base degrading over years due to various reasons. So sustainability and sustainable agricultural future for all of us is a matter of concern. Agriculture has done exceedingly well. A recent CII report says that during past 8 years, there has been growth of 11%. And that is something which is remarkable. Year-to-year growth, if you see, during COVID times when all other sectors were severely impacted negatively. Agriculture sector grew more than 3%. India is not only feeding 1.3 billion plus population, it is exporting. 41 billion US dollar exported last year, which is $19 billion more than what we imported with regard to agricultural commodities. So we are agriculturally surplus nation. The begging and borrowing of 1960s and 70s has been adequately addressed. The import substitutions are being emphasized again and again by Honorable Prime Minister. And we still are struggling to address the nutritional component. Despite the enormous growth that we have in agricultural production, we are still importing edible oil. Last year, it was more than 1 lakh crore, which was spent on edible oil import. It's a big drain on India's economy. The soils are degraded. More than 15 nutrients for which soils are deficient at this point in time. The water availability is declining, per capita availability is declining. Unevenness of rainfall is causing far more damages to agricultural production in certain regions. Though we take great deal of satisfaction in the production gains that we have over 1950-51, more than 6 times production of food grain, more than 3 times production of pulses, more than 6 times production of oilseeds, more than 12 times production of cotton, more than 65 times production of eggs, more than 18 times production of fish, more than 11 times production of our milk. So all these reflect that the progress has been quite satisfying. But despite these satisfying situations in many sectors and export happening, our concerns are there. Sustainable future demands that our development needs of the present day are met without compromising the ability of the future generations to meet their needs. This is the most important underlying factor, underlying point that we have to remember and work. How do we really do this? If we talk of soil system, there is a need to address the soil needs and soil health and maintain soil health for future generations so that the productive potential of soils are maintained sustainably. The productivity is not sacrificed in a mad rush to produce more and more. And this calls for attention of everyone concerned. Honorable Prime Minister has given the call to reduce use of chemicals in agriculture. The chemicals, certainly the unbalanced use of chemical fertilizers, pesticides, pollute environment, degrade soils, pollute water, and also impact human health negatively. How do we really reduce this? We are promoting organic agriculture. More than 60 organic production practices are developed and promoted. Government has brought schemes to promote organic agriculture. The farmers are encouraged to go for organic agriculture, and currently about 3 million hectares are grown organically in the country. Market linkage, higher returns for organic produce, the quality control and certification process, all these are put in place. But there's a long way to go to promote, to strengthen organic cultivation in this country. There's a need to integrate organic agriculture with modern day tools and technologies which are formed part of precision agriculture, where the artificial intelligence, blockchain technology, and so on and so forth are being, in fact, experimented and already employed in certain situations. Large-scale use of such technologies would bring in greater precision to agriculture. Use of drones and also remote sensing technologies, various imaging platforms would in fact reduce great deal of input use, particularly chemical use. Application of those in appropriate quantity at the right time would enable sustainable agriculture. Use of biopesticides, use of biofertilizers, biocontrol agents, a large number of technologies on which are developed by ICAR can be deployed, and everyone concerned can actually jump into it and support this so that they are mainstream in our agricultural production systems, thereby making agriculture sustainable for future generations. Water is another concern. As I said, our per capita availability has come down below 1,500 cubic meter, which was 5,000 cubic meter in 1950-51. How do we really sustain agriculture production with less amount of water? The central government has given the call, catch the rain where it falls and where it falls. Catching rain is so important. Water flows into the ocean. And we can't really have that water utilized for our purpose in agriculture and for sustainable production systems. We have to catch this rainwater, which is fresh water, store them and utilize very efficiently, develop crop varieties which can effectively utilize this water very efficiently, and also develop micro irrigation system. Micro irrigation, currently the government of India is emphasizing, and it has gone up to 12 million hectares. A long way to go. Water is wasted in many agricultural practices, whether it is rice cultivation or sugarcane cultivation, potato cultivation, many other crops which are called water guzzling crops. Crop plants are essential to plan out and grow crops in areas where water is available in case of water guzzling crops, and leave those areas where water is limited. Not to grow sugarcane in water limited situations. If at all one has to grow, opt for micro irrigation practices. So sustainability of production systems with regard to water by way of reducing water footprint in the environment is so crucial for our future generations. If there will be fight between nations, fight between states, fight between regions within state, and fight between people in a state, within a district, might happen in future because of water shortage. And that's a serious concern. So sustainability of our production systems depends on that. Similarly, we have to think of reducing greenhouse gas emissions from agricultural systems. Reducing greenhouse gas emissions would provide positive, in fact, factors to grow crops, rear animals, and without compromising the greenhouse gas impacts, which are so huge on agriculture. How do we do this? Growing rice on standing water situations should be actually dispensed with to the extent possible. Direct seeded rice and other systems of rice cultivation are to be promoted. Technology is available. Eastern India needs such technologies where water is managed appropriately in crops like rice, and rice fields where water stagnates. The methane emission primarily takes place, and rice becomes #1 polluter among crops. Animal system also do pollute by way of emitting methane. And I believe very strongly that the technology available with us that reduce methanogen populations in the population, in their digestive tract, that would help in reducing methane emission from animal system. That needs elaborate application and adoption. Similarly, there are various other situations where we can manage, greenhouse gas emissions from agricultural fields. Agroforestry system needs promotion and integration very strongly, as a result of which we can manage our ecosystems better, sequester carbon better, harvest carbon dioxide by way of photosynthesis, and store them for very long periods in tree species, and as a result, reduce greenhouse gases which are there in the environment. And sequestration of carbon in soils, and also carbon storage in plants, they would be going a long way in making agriculture sustainable. Sustainability cannot be isolated and thought about in isolation without, in fact, bothering for farmers' income. So farmers' income are to be thought about simultaneously. So sustainable future not only requires a very responsible behavior, whether it is responsible agriculture, responsible animal husbandry, responsible fishery, and responsible agroforestry, responsible water management, everything, everywhere, we have to be responsible. And that needs behavioral change. That needs attitude change. That is the toughest task. And that is what is requiring creation of awareness. Programs like this, where even educated people and scientists who have gathered there. And in fact, among us, we also need creation of awareness. We, in our mad rush to evolve ourselves, to develop ourselves for our own progress, we often sacrifice and we don't care for those smaller aspects of life where the environmental aspects are forgotten and the sustainability is sacrificed to a great extent. And I strongly believe that in our food system, in our water use patterns, in our daily life, there's a lot more to be done so that our future remains sustainable. And I'm sure the programs like this will bring sustainability of future agriculture, future food system, and future development in entirety, would be brought to focus. And we would be deliberating and bringing out recommendations which would be of great help in actually, first of all, looking at those critically and deliberating further to implement them through government programs, through participation of the private industry, and also non-government organizations. All have to come together. Even civil societies all have to come together to ensure sustainable future, sustainable development in the future. so that our future generations, without any botheration, are able to address their needs. So with these words, I profusely thank you for inviting me to be part of this important program. And many respected delegates are there, and they would be giving more insight and throwing more light into the topic. And I wish the program a great success. It has been already initiated in the morning, as I learned, and I'm sure till tomorrow it will continue, and will bring out more facts and figures and more data and information for us to work on. And thank you very much for inviting me, and wish you all the very best.
Technology. My colleague who has put a lot of effort, Professor Pradeep, was very much in this seminar, the organizer. People are talking about sustainability on agriculture. I am basically a man of plastics. So I will just cite what are the sustainability in agriculture, followed by earlier harvest, less reliance on herbicides and pesticides, frost protections, and water conservations. Plastics in horticulture and floriculture, we all know soft plastic films which are mulching. Today we are talking about biodegradable mulchings, which is a huge applications for the whole plastic cultures. Then you have agriculture and farm equipments, which dominates use of pipes and fittings in the agriculture sector. These are the materials which are extensively used across the globe in terms of supporting the farmers and plus agriculture in particular. Then you have a real components called sprinklers, drip irrigation, sprinklers, greenhouse, all these materials which are being basically a material which is being used for plastics applications. Let me tell you, when you say eco-friendly material, then it's basically the plastics used in place of other materials and which are recyclable. We can recycle, reuse, and that is how it can be economically balanced. Now, the material which are used are polyethylene, polycarbonate, polypropylene, polyvinyl chloride, ethylene vinyl acetate, polymethyl methacrylate. These are the material extensively used in plastics in agriculture known as plastic culture. So whether it is silage, whether it is a plastics reservoirs and irrigation systems, whether it is mulching, whether it is low tunnels, whether it is recovery and opportunities. So dear friends, dear scientists, I know I don't have much to talk about the nitty-gritty of the agriculture, but I was working for a ministry which was dealing with petrochemicals as well as fertilizers. In my tenure, we had a fertilizer called neem coated fertilizer that was developed by the ministry, and the materials which were injected through micro irrigation system, sprinklers, all these materials are extensively used. You will be requiring any of that support, probably we can always help you. I sincerely thank the organizer. I would be grateful to this organizer if they have started this conference with the support of Ravensa University and the Department of Botany in particular. I will be highly obliged if they sponsor some fellowships to our research scholars. That is how we can motivate researchers, and they can talk to my colleague, Dr. Pradeep, Professor Pradeep, encourage our young bird for research. Please take their care. We are here to support them. We are also trying, but these agencies should come forward, and the sponsor who are coming here to support this conference must encourage research, and Ravensa will be always waiting for that. Thank you all for calling me.
Thank you so much, sir, for your wonderful address. And now, before sir leaves, because as we learned just now that he is busy and he has to go somewhere, I would definitely want to call on stage Mr. Divyanshu Prasad Das, Program Director, Agri Vision, to carry forward the next special segment, which I won't disclose. First, let him disclose that.
Very good afternoon. Like in AgriVision 2021 was really very tragic for the AgriVision family, because we lost two of our gems. Dr. Sabus Sahu was the Dean and Reader of Department of Biotechnology, Utkal University, and Professor M. Mahadevappa, who is popularly known as the father of rice. They both have been associated with the AgriVision family since its inception as the organizing committee members. They used to be our candle bearers. They used to guide us. They used to advise us. But it's at last it's the will of the destiny, and we lost both of them to COVID. So since last year, the AgriVision family has been honoring with Dr. Sabus Sahu Memorial Lifetime Achievement Award. So for this year, I would like to invite Sanjay Naik sir to do the honor. So please come ahead. So for this year, Dr. Sabus Sahu Memorial Lifetime Achievement Award. So I would request Professor B.C. Tripathi sir to come on stage. Please give a big round of applause for Professor B.C. Tripathi sir. Yes. Please put a big round of applause.
So since we started this last year, Dr. Sabut Sahu Memorial Lifetime Achievement Award, the first one here goes to Professor B.C. Tripathi, our former Vice Chancellor, Ravensa University, and Professor JNU. Thank you so much, sir, for always being generous as ever, and always being a guiding angel. And thank you for teaching us. Thank you for making us what we are today. A big thank you to you. Let's have a wonderful round of applause for the gentleman.
Next, I would request Dr. Joy Krishna Jena sir. Sir, VC sir. Sir, you have to award 3 people. Joy Krishna Jena sir, please put a big round of applause.
The next Dr. Sabut Sahu Memorial Lifetime Achievement Award goes to Dr. Joy Krishna Jena, our Deputy Director General Fisheries Science, ICAR. Thank you so much, sir, and congratulations.
And now I would request Ashok Kumar sir to please join us. Sir, please come. Dr. Ashok Kumar, Director NBPGR. being felicitated with Dr. Saboo Sahu Memorial Lifetime Achievement Award.
So this year we had 3 winners for Dr. Saboo Sahu Memorial Lifetime Achievement Award. Professor B.C. Tripathi, Dr. Jai Krishna Jaina ji, Dr. Ashok Kumar. Let's have a wonderful round of applause for all the gentlemen again. Carrying forward, we have a few more awards. This one is Krushak Bandhu Award, which I would request Professor Pawan Kumar Agrawal ji, Vice Chancellor OUAT, to come forward and give away these awards, along with Dr. Jai Krishna Jaina ji, Deputy DG Fisheries Science ICAR. Also requesting if Divyanshu Prasad Das ji could also join us to give these awards. First and foremost, Krushak Bandhu Award, the first award for the same category goes to Mr. Dhirendra Kumar Bhanubhai Desai from Gujarat.
I would like to introduce Mr. Bhanubhai Desai. He is is a renowned farmer from Gujarat, Bharuch. So he has been farming banana in 40 acres of land. So his farming techniques are really inspiring. Please put a big round of applause. Thank you, sir. Thank you for joining us.
No, sir. There's a couple of more awards to this category. The next one goes to Mr. Jyoti Prakash Mahanti from Cuttack, our own city. Okay, I guess he's not present. So the third and the final one for this category goes to Mr. Manoj Kumar Bisoi from Samantrapur, Cuttack.
Mr. Bishoy is a leading farmer of Cuttack district. So he has set up an pearl farming institute in its small farmhouse.
Congratulations and thank you so much, gentlemen, for doing the honor. The third category of award which we are to give now has to be presented by Dr. Ajay Parida, Director, Institute of Life Sciences. Sir, can I please request you to come forward? And there's just one name to this category. The category is the Business Innovator Award, which goes to one of our consistent pillar, because any big or any great show cannot be done without our supporters, without our sponsors. So one of the biggest pillars of our fraternity, Dr. Sarat Kumar Sahu, Managing Director, Ruchi Food Line Limited. Let's have a wonderful round of applause for the gentleman. Okay. Congratulations, sir. Thank you so much. Thank you so much, sir, for doing the honor. And the special one to give away this, this is not an award. This is rather a felicitation. This is a token of love from the fraternity of Agri Vision 2022, 2022, to give this small and big and whatever you want to say, this token of love. Let me call on stage Divyanshu Prasad Das ji, Program Director, Agri Vision. And guess who's getting this? Can we give a blind guess to this? This is a special token of love a special person should get. So this special person is Professor Sanjay Kumar Nayak, our Vice Chancellor, Revensa University. सर जैसी बातें आपने यहाँ कर दी ना और जितने विजन्स खोल के रख दिए हैं सामने हमारे, स्पेशल तो आप बन ही गए हैं कि नहीं? एक जबरदस्त तालियां होनी चाहिए सर के लिए. थैंक यू सो मच सर. I'll request all the dignitaries over the stage to please take the seat down, and one by one, I'll be requesting each and everybody to come on the stage and guide us on this wonderful occasion. So thank you so much, everybody, for joining us. So the first address, I would request Dr. Jay Krishna Jainaji, Deputy Director General Fishery Sciences, to join us on stage and address the gathering.
How much time I will get?
5 minutes, 7 minutes, whatever you want to take.
Okay, whatever is, okay. Good afternoon, friends. Most important dignitaries who are there, some of them have already left. Honorable Vice Chancellor of this esteemed university, Dr. Nayak ji. I could see here Dr. Tripathi ji, Dr. Agrawal, Honorable Vice Chancellor of OUAT, Dr. Ajay Parida ji, Director of ILS, colleague directors of some of our ICR Institute, Dr. Ashok Kumar, Director NBPGR, Madam Shwai, Director NRI. We have Director of SIFA, Bhubaneswar, Dr. Saroj Kumar Shwai, some of our colleagues, Dr. Rautrai, and I find many of our esteemed friends who are assembled here. Student friends, faculty, delegates, ladies and gentlemen. It is indeed a great honor and privilege for me to be here. I consider it a rare opportunity. I consider it also a coincidence because this program I was not knowing. About 3 months back, I told I will be there. Then in between it got postponed. Then suddenly some other program came. So I am here. Friends, I do not know how much time I would get. I asked Madam how much time you can take. She told first 5 minutes, then she told whatever time you can take. There is a long difference between 5 minutes and whatever time I can take. So I do not know how much time I will be having. Friends, our chief guest of this today afternoon, Dr. Mahapatra Shah, Honorable Secretary DARE and DG ICR has spoken about the sustainable agriculture, where we are at present, and where we want to reach. All of us know, if we are able to smile, if we are able to talk a bit loudly, when I say we as an individual, we as a country person, we as anybody in a country, if we are speaking proudly and loudly, it is only because of our stomach is full. That is why we are able to speak loudly in the international front also. I would not be speaking much of agriculture because I have very rudimentary knowledge on agriculture. I come from a fisheries discipline, a small area. People many may not like to listen, also those who are vegetarian. But in Orissa, I find most of us are non-vegetarian. So I don't have much problem definitely here to speak. DG sir was speaking about the growth where we have been able to achieve in last 75 years. From 50s to today, 1950 today, where we are, he was giving a figure, 10-fold increase, 20-fold increase, 60-fold increase, and so forth. And we know the challenges before us. Yes, our stomach is full, but by what? Carbohydrate. Still we talk of protein. We talk of protein security still is an issue. We all the time speak about nutritional security is an issue at this point of time. When we are talking, that is the reason why everybody is speaking today only biofortified crops. All of us have been working on over these years, whether paddy, wheat or vegetable, we are trying to bring in fortification of several things, whether it is nutrient, vitamins, minerals, and whatnot. Madam Shwai is here. She must be speaking on how we are going ahead with the biofortification. That is why golden rice we talk of all those time. When we speak about the fish, I feel bit proudly. I say I work with a commodity which not only provide food security, but protein security, nutritional security, whatnot. And at this point of time, we say fish is not only a protein-rich food, not only a nutrient-rich food, but it is a health food. That is what we try to emphasize at this point of time. You can, we know all of us are speaking at this point of time, sustainable development goals. Today, all of us speak. Whether you speak about goal number 2, 3, and all, you can go on counting. But to start with, zero hunger, that is what is the first priority before us, or the well-being, the goal number 3. That is what is most important at this point of time. In this context, I consider fisheries is one area where we can contribute significantly to this. As because this is being organized by botany department, I sincerely believe many botany students should be there, but you may have zoology as a separate other subject, I'm sure. So you may have some interest with a fish. That is why we say today, if you see the literature published by FAO, Food and Agriculture Organizations, you find 10 superfoods. Out of 10 superfoods, fish is considered one of the nature's superfood. Not only FAO says, but also several organizations, including Harvard Medical School, report, if you see, they consider fish as a superfood. But when we look at the future, we do not know whatever we talk of superfood, whether it will be available for us, we do not know. When you go back to our history, we know that people were eating fish even 5,000 years back, Mahabharata time, you know, Matsya Gandha and all, you must have heard. That time I do not know, this Mahabharata war was fought somewhere around 7,000 years back or 8,000 years back. But some of the study, if you see literature, that is what example I give many times. Recently, not recently, about 7-8 years back, one paper was published in PNAS. The study was an isotopic study. They got a skeleton in the Tibet region. It was buried inside the snow. When they got that skeleton of a human being, they tried to analyze that human being. They found that skeleton was a skeleton of a young man of about 40 years age. And at that 40 years age, and that skeleton was about 40,000 years back skeleton. 40,000 years back skeleton of a 40 years young man who had eaten a lot of freshwater fish. That means 40,000 years back also people knew fish is an important food item. Now the question before us, whether the same fish will be available before us another 40,000 years ahead, we do not know. There are big question marks. Maybe 4,000 years ahead we can get, we do not know. The biodiversity loss that you see at this point of time. And even 2100 Mahapatra Sahib was mentioning the challenges before us, the climate change challenges. 2 to 3 degree temperature increase in another 80 years, the big thing. We are not able to predict what is going to happen another 500 years ahead. We are talking only about 80 years today, 2100, 3 degree increase in temperature. That is what we do speak. Consider if another 5 degree increase in temperature happens, what will happen to the biodiversity? We do not know at this point of time. He spoke about the water challenges of water, fresh water. We do not know whether the rivers will remain live. We do not know whether water will be available. Even we do not know the very availability of water for the agriculture. We have seen how water table is going down and down. There are big concern whether the fish will be available at all. But we should be optimistic that we should be able to, a human being should be able to meet all those challenges through different whatever. That's why we are here. When there is a issue, we are here to counter those challenges. I'm sure we should be optimistic that fish will be available. Consider fish will be available, but what quality? That is the challenge. The safety and quality issues are remaining major issues. Even today we talk of antibiotic residues, pesticide residues, and so forth. So what quality of fish will be available? That is the bigger challenges before us. But we have been able to do well over last 70 years. from less than 1 million ton. Today we are talking of 14 million ton, second largest aquaculture producer, third largest fish producer in the globe. We like to retain this position, probably elevate our position, if not to the first, but at least up to second position, we should be able to go from third to second. Definitely we should be able to reach in another 5 years. But the task before us is quite Herculean. The 14 million ton, the government of India has set a target of producing somewhere around 22 million ton. And recently again by the government of India, they have given us to directive to formulate a strategy or roadmap for what will be the scenario in next 25 years. That is why Vision 2047 roadmap we are trying to prepare. What will the fish, how much fish will be available? What would we demand? Whether we will be able to meet those demand. If we are able to meet the demand, what are those fishes which we can provide them? So there are challenges are many. The challenges which was there earlier, our expectation was to increase the production, productivity. Not only I am talking about the fish, milk, DG Sahab was mentioning more than 20-fold increase in milk production from 17 million ton. He told about 200 million ton or 198 million tons, and cereal crops 300 plus. All those thing we used to say. But the challenge before us is how do we have the diversity what we want to give. All those challenges what we are talking, diversity when the challenges remains. For example, the climate change is one of the challenge. Water will be second challenge. And a third important challenge, what we called it, third generation problem, is the AMR, antimicrobial resistance. It is the challenges which probably all of us are not able to see. Maybe another 20 years, 25 years, we will be able to know the difficulties what we will be having, the amount of our investment we need to do in the AMR issue challenge. This is a challenge not only for the country but the whole globe. So on this, in this scenario, how do we ensure the quality and safety of our food products are so important at this point of time. We have been talking of sustainability. We do talk of maybe just sustainability is not good enough for us. If we want to fill the stomach of all our increasing population, 160 billion, 1.6 billion people in the next few years, just sustainable production is not good enough. We need to have sustainable growth in production, or some acceleration is necessary. Just growth is not enough probably. We need to provide diversity that we have been able to achieve to good extent, but with the quality safety issues. At one hand, we say that, okay, we'll be able to increase production productivity. Definitely, yes. That is what everybody says. India does not need 130 million hectare area for crop production. If India has to grow, probably we need to reduce the area. That is what all of us speak. Then only sustainable intensification and all we do speak, or diversity we should be able to bring. But definitely the challenges are many. We used to say that, okay, the target given before us, I'm sure those, if you ask, if somebody is producing maybe 100 ton, if you say that you keep a target 200 ton, probably he will be able to achieve. But where he will sell, even the talk of fish. For example, the target given before us is another addition of 6 million ton in next 5 years. 14 million ton production we have been able to achieve in 70 years, and 6 million ton additional in 5 years. Yes, we will be able to produce, but who will buy? Yes, everybody want to eat fish, but do they have money enough to purchase those fish? Whenever we talk about the fish production, people say this is a very cheap fish, Katla, Rohumurgal, cheap fish. You should go for diversified fish, high value fish. Yes, high value fish will produce, but who will purchase? That is what is the problem. People say, okay, we take pride all the time that our export is 47,000 crores, 18% of the India's agriculture export. We take pride in the sector who are working in fisheries. And the target given us that we should be able to export 100 crores, 1 lakh crores. 1 lakh crores export by another 5 years. Yes, we should be able to achieve. maybe from 8 lakh ton to 14 lakh ton, then 14 lakh ton to 30 lakh ton, and by 2050, then we say that, okay, but you consider who is the market for us? USA, Europe, China, Vietnam are the market. How many people will, for example, that means we will be increasing threefold or fourfold production, but population increase will be there only 15% or 20% during this time. With 15% increase, 400% increase in production, whether the global market will be able to absorb those. And it is not only India. It is not only India who is sitting here to produce. Whole globe will be competing with us, whether Ecuador, Vietnam, or Taiwan, Thailand, Korea, and so on and so forth. Everybody will be competing with us. They will not be just simply sitting with, just looking at us that India is growing. If everybody goes on production increase and everybody looks at USA or Europe, then what is that status? So our only objective should be, this is what how do we tap the Indian market? How do we tap the domestic market? Our market is quite huge. Not only we should be giving emphasis on export, but how do we increase our domestic market consumption? That is what is the primary requirement ours should be. But as I told, production productivity will remain mainstay, but at the same time, yes, safety, quality we speak about the equity, we speak about gender, everything is important for us. So goal is not one before us, goal is multiple goals, multiple challenges, but we should be optimistic that I do not know, I think I have taken time, but we have geared up to work on several areas, whether diversification, water use efficiency, water productivity, increasing the water productivity, or all the aspects of nutrition, health, genetics, and so forth. Dr. Sundarai must have given up the work being done at SIFA on the genetic improvement program of Rohu, Katla, and so forth. He must have given the expectations before us also he must have given. But another major challenges before us is the health problem. All of us globally, people have realized how health is important, whether it is animal health, livestock health, or fish health. Whole globe is working last 2 years. Today, every nook corner of the country, globe you go, even a small children of 5 years, he will be able to say what is called quarantine. What is called vaccine, what is called diagnostic, probably everybody knows that. So I'm sure in coming days we will be working all those aspects, and unless there is a issue, probably people will not realize our importance. So now everybody realized the importance of health sector only when Corona was there. So only when there will be scarcity of food, there will be, people will have more purchasing power. That is what we envisage, that another 10 years or 20 years down the line, people will have enough money to purchase so many things. That is what we give example of France and all, how much food they purchase. So probably Indian people will have so much of money to purchase. So then only people will realize the importance of not only scientists, but also everyone associated in the food chain, starting from farmers to producer to market to industry to everyone. Thank you very much for the opportunity, Dr. Dash, that you have given us this platform to be here. But madam, I have not taken more time. I would have loved to be, because whenever we speak, after 2 years, people are hungry to speak. Because last 2 years, everybody started speaking only virtually. So when they see people physically, they start talking. So it is very difficult to refrain. Thank you so much.
Such a jolly human being, sir, you are. Thank you so much. And now I'll request Dr. Ajay Kumar Parida, Director, Institute of Life Sciences, to please join me on the podium and speak to the gathering. Let's have a wonderful round of applause for the gentleman.
No, 5 minutes, not more. Thank you, Dibyansu, for calling me for this. And this is, I don't know whether it is 4th or 5th of your event which I am participating. Third The last one was, again, was held in ILS itself. Last two was held in ILS itself. So it's a pleasure to come out of ILS and speak here in front of you. I think my job has been made much simpler by the remarks made by Dr. Mahapatra as well as Dr. Jena. They have outlined, I think there is no denying to the fact that India has made significant progress in the field of agriculture. So that I don't want to go to the statistics, 10 times increase in the food grain production, and all those things are there for the textbook. So what I thought that I will just point out of the focus on three basic points so that we can take it forward. Despite the growth in agriculture, despite our resolve to make farmers' income more attractive, still there are challenges. I think both Dr. Mahapatra and JK, both of them has highlighted the problems we are facing. I think while going, toward the end, Dr. Jena mentioned about the COVID. In COVID, I think I have been actively engaged in the COVID per se. So we have brought into the stage where if you tell somebody is positive, you test the sample and tell that you are a positive, irrespective of the educational qualification or whatever it is, they will simply ask you, what is my city value? I think we have made science so much of popular during the COVID period. So they have even know a villager in the village knows what is the city value, what is the type of virus load it has. Then when there was opposition to COVID, the government, entire bureaucracy, the government, everybody started talking about why have a faith in the science. I think that has driven the COVID pandemic challenge. And I'm sure Dr. Jena will agree with me. We need a similar situation where policymakers or decision makers say that believe in science, believe in scientists. And as it was said that the land is going to decrease, the population is increasing. So what is our priority is that we have to produce more food per unit area. I think that is the challenge which we have. I think that is what Dr. Jena said. And that can be only achieved when you use technology. For example, a country like Bangladesh and Philippines has accepted GM technology. They have accepted a GM technology in brinjal, which was developed in India, but we are not able to accept it. And believe me, if you accept genetically modified brinjal, your pesticide application will go down by 80%. The farmers will save money, and the productivity will increase by another 40%, which will get more money. So don't you think that technologies are required for now? The agriculture has to be technology incentive. There is absolutely no denying to the fact we are delaying the process. The more early we accept technology, I'm not talking, I just gave you an example in the crop sector, but I think even in fishery sector, in every animal husbandry sector, technology application is a must. I think all of us should start arguing for or pressurizing the government or advising the government to accept the technology per se, so as to increase the productivity per unit area and also increase the farmers' income. I think that is the only way out. My second point comes is that the integration. I think integration is the key, where if you have to produce more from a limited area, integration of agriculture, aquaculture, animal husbandry, fisheries, everything has to be integrated so that you try to multiply the impact which you are coming. There are, I think, many of you, the students, you can read about the integrated farming system models which they're talking about in various agro ecosystems, where what happens is that for 1 acre land, your income generation goes off. And that also requires a huge partnership. I was just sitting there, I was looking at the number of logos Divyanshu has been putting it forward. So I think this is what the type of collaborations and cooperations you require if you want to bring in agriculture. I think he had partnership with state government, central government, business establishments, I mean, all those people. So I think that is what we are looking at. The third thing which I would focus on is I think already Dr. Jena and Mahapatra has mentioned about it. How do you integrate agriculture with nutrition? I think there is a study which has been conducted by FAO which says that the more the people depend on agriculture, the more malnourishment they have. For example, India, with a country of about 65% of the people dependent on agriculture, there are 35% or more malnourished people. For example, take the case of our Kalahandi, Koraput, Malkangiri, I mean the Western Odisha. The mother and children who are anemic, that is a staggering 71%. And their food, I mean their regular food comes from the locally available resources. So can you do something for it? I think, again, Dr. Jena has mentioned about the biofortified crops, but there are also naturally biofortified crops. Take, for example, moringa. I mean, if you go back to your, what you call the saying that take 1 carrot a day and that your vitamin A deficiency is completely gone. So there are a lot of things which has to be done with the food system. There are traditional knowledges, associated traditional knowledge with associated with the practices, food practices of the tribal populations of Odisha or anywhere else. So how do you, I mean, we have to go beyond, like in last 50 years or last century, we have only 3 food crops which has given us more than 80% of the calorie: rice, wheat and maize. Can you think about something else which the tribal people are cultivating, consuming for their betterment? So I think we need to go back to our, like the government's focus, or there was a popular mandate. I think Pawan's group is working on it, the millet mission. How come the millet become popular? Millet, which was forgotten completely, that also become popular, and those are being cultivated in the tribal areas. So maybe we should look into some of the type of traditional crops, add value to that, and then make much more agriculture relevant. My last point would be, how do you attract and retain youth in agriculture? I will not, I mean, none of us, I think all those people are sitting in the front row. Have any of you thought that to put your son or daughter in agriculture? No. A doctor's son wants you a doctor. A advocate's son wants you an advocate. A scientist's son wants to be either a doctor or a education, whatever it is. But we want the farmer's son has to be a farmer. Why? I think that's a real question you have to ask. The question is that, I mean, I'll just give a small example. Why, I mean, we have made so much of progress in our, I mean, what do you call, automobiles and all the services sector. Why cannot we have a tractor which has air conditioned chamber? If you have that, then you can attract the educated people and all these things to do that. So I think the way we are thinking, there are tremendous opportunity. How, if you want to attract the people to agriculture, then I think the new word which is going all around is what you call the startup ecosystem. I think that is where we require, whether it is value-added food or the service sector, service-oriented things, marketing, and all those things, we can have a startup, people who can engage startups, so they are not job seekers rather than they are a job provider. Just to give you, I mean, last 2 minutes, I think, just to give you a small example, ILS we have just initiated because of the COVID, it got delayed. I think last year we have initiated a startup, I mean, a bio incubator. And I can proudly say, out of 20 startups we have initiated there, 5 are working on agriculture sector. And 3 of them are under the value-added services. For example, Dr. Jena, you will be happy to know that somebody is preparing manures and fertilizers from the fish waste. So that, and he has now, last week, he has signed an agreement with the state horticultural department to supply something like about 25 tons per month of biofertilizer, fish-based biofertilizer to the horticultural things. Similarly, all of you know, how many know, when all of you, those who are from Odisha, you will be knowing the Torani or Peja and Torani. Now one of the entrepreneurs are making Tetra packs. It's all only Torani based Tetra packs, but these are what you call value added, in the sense some are with cardamom flavor, some are with cinnamon flavor. And believe me, each of this packet is being sold at 15 rupees. And that particular person has a huge market. So I think there are great opportunity. So as I said, embrace technology, look for the traditional knowledge, work in partnership, and look for employment opportunities in agriculture. And that is how we can really transform the agriculture for the betterment of the society. As our honorable, the first Prime Minister of the country has made this statement that everything can wait, but not agriculture. And he has made that statement in 1952, and we are in 2022, but that statement which he has made in 1952 is still relevant and still need to be addressed. Thank you very much.
There cannot be anything which we cannot agree upon. Each and every statement said holds true. And of course, we need a wonderful round of applause for the gentleman for saying all the things boldly on this platform. And now can we request Dr. Padmini Swai, Director ICAR NRRI, to join us on stage and speak to the gathering. Let's have a wonderful round of applause for the lady.
Okay. Very good evening to all of you. Most Honorable Director General of ICR and Secretary there, Dr. Trilochan Mahapatra sir, our most respected Deputy Director General Fisheries Science, Dr. Jena sir, most respected Vice Chancellor, Dr. P.K. Agrawal of Orissa Agriculture University and Technology, OUT Bhubaneswar, the most respected Professor, Dr. Sanjay Naik sir. He has already left anyway, but he was in the first speaker. And most respected Dr. Ajay Parida sir, Director, Institute of Life Science, my colleague, friend, Dr. Ashok Kumar, Director, NBPGR, ICR, Dr. Swaim from CIFA, Director CIFA, my colleagues from NRRI, Cuttack, my friends from Ravenshaw universities, and the most important, the organizer, Dr. Dibyansu, who has organized. I thank profusely to invite me for this forum. Of course, this is first time I have come here, but I could see it is a very vast program he has organized, and the today's theme of this sustainable agriculture for the future. The sustainable agriculture from the beginning, Director General has told, Dr. Jena has told, Dr. Parida has told, this sustainable agriculture, the sustainable goal, it was fixed by United Nations in 2015 with 17 themes. And what Professor Jaina told, that first 2 points are very important, that poverty alleviation and zero hunger. That is, we will only smile when our stomach is full. So we need food first to survive. And before Green Revolution, we are also not having sufficient food. And our National Rice Research Institute, which is built in 1946 in the backdrop of most important Bengal famine in 1943 due to shortage of food because of the brown spot disease, as well as the food shortage, and a lot of people, we had a lot of casualty of around 3 million people in that particular time. And after that, the institute is established, and by the help of starting from center to state, many people are involved there to establish the institute to develop rice research. Rice is our staple food, and we need rice. We are 85% people, we are eating rice. So this Rice Research Institute developed, and by this time we have developed more than 154 varieties since last 75 years. And what Professor Jaina told, that we have also developed 3 biofortified rice varieties, CR than 310. That is for the first time we have developed the high protein rice. It is having more than 10% protein in the polished rice. Then 311 and 315, that it is also biofortified with zinc, having more than 25 ppm zinc in the grain. So these biofortified things, why should we have? Because as Dr. Parida told, Dr. Jaina told, the whole population, now the 1.6 billion in India, and we are also going, our population is going to increase by 10 billion whole globally. So for this purpose, we are having the challenges like climate change, and that soil erosion, soil is getting degraded, also soil quality is getting degraded, water scarcity is the most important challenge we are going to face. So many things getting spoiled because of the climate change, and all these things, everything is getting reduced, and from these reduced very less resources, we will be definitely trying our best to get more food production. And in that food production, the quality is also important, and for that purpose we are trying biofortify the rice varieties. And we have biofortified 3 rice varieties, and we have also developed hybrid rice on just to what Dr. Parida told, before Green Revolution, we are not able to have our food sufficiently. And for that purpose, we tried our best. Many programs has developed, and we got the food production now, the bumper food production of 300, not only rice, the whole food production 316 million ton in our country. And not only in food, in horticulture also, and fisheries, animal science, everywhere. We don't we need food along with all these components. Dr. Jaina has already elaborately told how we need the nutrient also from other sources like fish, animals and vegetables, not only from rice. So all these things we have to get with a challenging mode to get from these all reduced resources, how to get maximum yield, maximum production. So to face all these challenges, we have developed, our scientists and our friends have developed many technologies, many machineries, many IT-based technologies like artificial intelligence and drone-based technology, how to take images of crop fields and appropriate application of insecticide, pesticide. Of course, nowadays our prime ministers demand is that we have to go for natural farming because we have already spoiled utilizing many chemical fertilizers, insecticides, pesticides to spoil our environment. So we should go for natural farming. In this natural farming, whatever our resources are there, natural resources are there, we have to utilize that one, and we have to produce more from that. So to sustain our food requirement, we have to go for, of course, advanced tools, machineries, technologies developed by different scientific organizations, and not only the quality, quantity of food, quality also, nutrition, and in every aspects we have to go. So for sustaining our demand from this agricultural sector, mostly we have to take all the challenges in a holistic manner to utilize our resources. And today's this seminar on sustainable agriculture for future is a very befitting one, and I think our 3 days program will definitely give some throw some light to the students, researchers, scientists, those who are attending this one, and they will gain maximum of this and utilize in their future to sustain our demand, food demand. Thank you, I think.
Thank you so much, ma'am. Now requesting Dr. Ashok Kumar, Director ICAR NBPGR, to join us on stage and address the gathering. Can I request for a wonderful round of applause for the gentleman? Thank you.
Good afternoon. The most respected Dr. Mahapatra, DG ICAR, Secretary there, and Chief Guest of this function, Dr. J.K. Jaina, Chairman of this function, Dr. P.K. Agrawal ji, Vice Chancellor OUT, Dr. Ajay Parida, Dr. Padmini Swai, Dr. Nayak, Dr. Tripathi, the Vice Chancellor of this university, and the colleagues from the Eastern-based centers, ICAR center, that is Bhubaneswar or Cuttack, NBPGR, CIFA, and CHES. So, and the colleagues from NBPGR and NRRI and the delegates of this function. I am highly thankful to Divyanshu Das for giving me opportunity to be here. When we talk about natural farming, organic farming, sustainable agriculture, or climate resilient agriculture, we have to be dependent on the biodiversity, agrobiodiversity, plant genetic resources, crop genetic resources. All are the biodiversity. The agrobiodiversity is the component of the biodiversity. Agrobiodiversity means it depends on the plant, animal, fish, microbes, even the insects, the beneficial insects. So we are totally dependent on these component of the for the agrobiodiversity, for the sustainability of the agriculture. Then we come to the sustainable development goal. I think out of these sustainable development goals, 3 or 4 are very much relevant to the PGR or the agrobiodiversity. So now you can think here how these are important, how these component of biodiversity or agrobiodiversity are important for the sustainability of the agriculture in the form of different farming system. When we come to the products of agriculture, Dr. Parida timely told that out of 3 lakh species are available in the world. We are utilizing 1 lakh species in different ways. But for our calories, 60% population are totally dependent on the 3 species, 3 crop species, that is wheat, rice, and maize. So you can see the downstreaming of this, the biodiversity. So, and the quality of the Now, these are the main challenges. And if go for the genetic background of these crop varieties, the most of they have the very similar parents. So the diversification of parents are not there. So they may be any time they may be susceptible to any kind of the biotic or abiotic stress. So I have given the talk, the unlocking gene bank material for the broadening the genetic base. So I have taken the initiative to characterize whole germplasm and to develop a different kind of reference set for the utilization of different breeding programs. It may be wheat, rice, or in minor pulses with the help of ILS, and a network mode in different crops, even in the mission mode project of the DBT and oilseed mission. So when we come to this, if you will come in this, the challenges. So how PGR can help in this to deal with the challenges? First, more Dr. Paida told, we should broaden our food basket. We have a lot of indigenous plants. The moringa is one of the examples. So many medicinal plants, they are very good for health. Similarly, some of the plants, they are very highly nutritious. Some of the resources or plant genetic resources, they are very much higher value having for the nutritional and health security. So first of all, we have to have our broadened food basket. Then the other one is the mainstreaming of the plant genetic resources. Because we have the land races, we have the farmers varieties, there are a lot of material available in the system, and we are collecting of these material, but the potential of these material is to be recognized in the form of climate resilient agriculture, or their values and the resistance to biotic abiotic stresses, or for the nutrition security. So if these material are having very good quality in the form of nutritional value, maybe the protein, the minerals, or anything, they can be go again in the farmer's field, and that not all they will not will help to the farmers, but they will also be helpful to the our population, which are with the urban population, which are suffering from hunger, hidden hunger. So that can be find place. They will increase the farmers' income, and also they will be beneficial to the health benefit of the urban population. So some of the genetic resources that can find close. So these are some of the points, like broadening of the food basket, the germplasm for the climate change, resilient agriculture, and the germplasm for the nutrition security, that can be broadened, and that can be find place in the different kinds of farming, maybe the natural farming, agriculture farming, or then some of the genetic society can also find place in the breeding of the varieties. So you can say the biofortified varieties. You can blend some of the incorporation breeding, some of the unique trait like high protein, like high content in our high yielding varieties. So they also be used in this way, and this kind of the technology through technological innovations or through breeding programs. So the utilization of the plant genetic material or the plant genetic resources can be helpful to to deal the challenges of our national system, and the problems of the farmer and the problems of the agriculture. With this, I am highly thankful to the organizer for giving me a chance here. Thank you.
Thank you so much, sir. And last but not the least, I will request Professor Pawan Kumar Agrawalji, Vice Chancellor, OUAT, to join us on this podium and address the gathering. Let's have a wonderful round of applause for the gentleman.
Thank you, the organizers and all the honored dignitaries, Honorable DG ICR and Secretary Dear, my esteemed colleagues, DDG Fishery Science, Dr. Parida, Director ILS. I'm not naming everybody, and Honorable Vice Chancellor of Ravenshaw and former Vice Chancellor, and all my colleagues. All are my friends. I know everybody's name. I won't name again. So I honor all of you and student friends and all the participants. When you talk agriculture and when you talk sustainability, exactly why we discuss that. So what makes us, what to sustain basically? Why I'm putting a question, what to sustain? So if you know our point here, we stand and we have to sustain, that makes a sense to debate or discuss. All of you know 1947, 32 million population. Today we have more than 1.35 or 36 billion population with almost 35 million tons of food grain and having 318 million tons food grain. We are in a good shape as far calorie intake is concerned. But today for us the challenges are, we need enough food. When we talk of food, we talk of calorie, we talk of nutrition, and lot of people when they talk of doubling farmer income or sustainability, one more point comes to us. We have produced enough, at what cost? We could produce enough, what way approach we followed? The policy of after third 5-year plan and today, how we have moved, whether the movement was judiciously well planned or we have something to rethink again. 1960s was a turning point for us that all of you know, and India became a highly food deficient country. Indian agriculture minister going to US for begging food. To 1972 almost were comfortable as far as calorie intake is concerned. Calorie requirement is concerned, not intake. I may give a wrong message. But then lot of issues were there. Whether we have enough produce the calories that is required by us, all the kind of food we need, have you got it? Have you stabilized the food production? And if it is calorie only food, then how to go for nutrition? Many of you touched. And suppose you want to make enough food in terms of calorie and nutrition, how to sustain it? Sustenance in terms of yield production, sustenance in terms of environment, sustenance in terms of income and livelihood. Somebody may say that we did a wrong thing by not putting the livelihood and income that time. But believe me, given the occasion 1960s, whether you are the Prime Minister of India or I am the Prime Minister of India, there is no other occasion than to think of calorie and food in terms of filling your belly. By 1980s, when we were having enough food, at least marketable required food. I'm sorry, if you say biological calculation, definitely you may say that, okay, we need more and you are having less. But if you talk that whatever is available for filling your belly, living and sustaining, we were comfortable. Then India had a second phase where we're looking for stabilizing the yield and looking for nutrition. And what we talk today, doubling farmer income, doubling per se may be in different meaning for everybody, but one common message that today agriculture, you have to see beyond production. Income and livelihood is equally important as production nutrition is concerned. So today's sustenance, when you are talking, you have to talk that from less land, it may be crop science, horticulture science, fishery science, or animal or poultry, whatever. The agricultural commodities, everything has to have a optimum level of production and productivity. For the population we are having, and if surplus we are having, we will export. So from less land, enough production, enough productivity. With a less cost to environment. Environment has natural environment like water and soil and many other components. And a precise application. Yesterday what we did, yesterday means last 50-60 years. Suppose you are putting nitrogen. We put n number of nitrogen per hectare and we thought the whole land is same.
Suppose I may be a pond, I want make an application to the fish for production. I believe that if all the ponds for particular species is similar, I make a similar application technology. But last 15-20 years, agricultural technology has seen a sea change. And not because finding in agriculture sciences, because recent science developments in other subjects like robotics, like nanoscience, like sensor application, like lot of other things, BRT, IoT, ICT, lot of things has happened last 15-20 years, and that has a potential to be used in agriculture, that is already being used. And in Indian agriculture, I believe another 20 years, those technologies with core knowledge, with diversification within the commodities, diversification between the commodities, and diversification between the varieties of a commodity. Huge potential is there. And that is why today we are talking of sustaining the production product. Sustenance without production productivity, sustenance without livelihood has no meaning of sustenance. So let us understand, because you need it or because you want to have only organic, you may not create one Sri Lanka again in India. Sri Lanka, you can take a risk because people like country like India and other countries are there, they can give surplus food. Tradable agriculture product is not more than 5% in the world. Even if you are having gold and silver with you and you buy agriculture product and a country like India needs extra food, you may not get in the global market because total tradable agriculture produce in the world is not more than 5%. So that is why a country like India, we cannot take a risk of making one opinion by one way to sustain. We have to have a time-tested technology, science-based research, holistic planning, without compromising production and productivity, without compromising the food and nutrition to his people, without sacrificing the economy and export. We have to sustain. We have to use our agriculture land from less land to more, less cost to the environment, soil and water, judicious use of input like fertilizer, pesticides, or any other related technology. Today, India, the developing world, and the world as a whole, we have a lot of scientific knowledge and technology availability and a lot of potential that we can do it. So today it is a challenge for us, and Indian scientific community, Indian governments, whether it is the government of India or state governments or local governments, we are all conscious, we are more than committed, and I'm sure we together, all of us, will bring a sustainable agriculture using technology, using science, having right policy and convergence, synergy between all the partners. I'm sure a modern thinking together, working together, science-based working, technology use, and all other things, not only agriculture sciences, but related science developments will succeed and will be successful. Thank you so much.
So before we commence the day one of Agri Vision 2022, I would request Professor Vaishnav Charan Tripathi ji to come forward and felicitate all our guests of honor one by one. I'll request Dr. Jai Krishna Jaina, Deputy Director General Fishery Science, to join us on the stage and accept the token of gratitude from Professor Vaishnav Charan Tripathi ji. Requesting Requesting Mr. Divyansu Prasad Das, Program Director, Agri Vision, to join us on the stage and also do the honor of all our guests. Guest of honor, please. This one is for Dr. Ajay Kumar Parida, Director, Institute of Life Sciences. So may we please have you on stage? Professor Pawan Kumar Agrawal, Vice Chancellor, OUAT. Dr. Padmini Swine, Director, ICAR, NRRI. Exactly, like right before our Women's Day, that is on March 8th, seeing one lady among 7 males is definitely a special thing. So let's have a wonderful round of applause for the lady. Followed by Dr. Ashok Kumar, Director, ICAR, NBPGR. So. सर वो अवार्ड था, ये फेलिसिटेशन है, प्यार है हमारे तरफ से, एक्सेप्ट कर लीजिए. Of Botany for collaborating with us for putting up such a wonderful show. Pradipta Mahapatra sir, I'll request you to please join us on stage. Absolutely. So it is said a man cannot walk straight until he or she has a straight spine. So they are our spines. Professor Vaishnav Charan Tripathi ji and Pradipta Mahapatra ji. Let's have a wonderful round of applause for both of them. This is just the day one that we will be closing off now with, of course, I'll request Divyansu ji to propose vote of thanks, but also requesting the 2 lectures which we could not hear today is going to happen tomorrow sharp at 9 in the same hall.
So with this, we're coming to end the sessions of the day 1 of Agri Vision 2022. Before closing for the day, Please accept my sincere apologies for the deviation in the program. It's because since it's the inaugural day, so we had a lot of hectic schedule for which we missed 2 lectures, which will be accommodating tomorrow. I'm really thankful to my respected directors from different ICAR institutes who accepted my request and they blessed me with their gracious presence. My heartfelt thanks to DDG sir, who among his busy schedule, he planned this trip specially to join us for AgriVision 2022. I'm indebted for your visit, sir. Thank you so much, sir. I'm thankful to Ashok sir, who was badly occupied yesterday because DG was having direct program at NBBG New Delhi, but still he made this possible. Today early morning he traveled down to Bhubaneswar to join us for AgriVision 22. Thank you so much, sir. I thank Professor Tripathi, Vaishnav Tripathi sir. At this age, sir was repeatedly requesting, no, Mr. Das, I'll be joining online. Still I requested, sir, with your presence, not only Revensavians, but other students of Odisha are going to be motivated with your speech, with your presence. So I humbly request, please join us physically. And at last, he gave up and he came and joined us physically. I'm really thankful to you, sir. Thank you so much, sir. I'm thankful to Honorable Vice Chancellor of Revensa University, Professor Sanjay Naik, sir, who have arranged each and everything for the smooth functioning of AgriVision. Whatever, whenever, wherever we are in need of any support from Revensa University, is just a call away. Once the call is taken up, the issue gets resolved immediately in a click. I'm really thankful to you, sir, for kind support. I'm thankful to Honorable Vice Chancellor, OUAT, Pawan Agarwal, sir. Sir has been associated with us since our inception and he has been joining us since last year with the AgriVision family. Thank you so much, sir, for joining us. Last but not the least, Ajay Parida, sir, is my pillar of support. He is our rock for the AgriVision family. Whenever we need any support, let it be venue support or any sort of guidance, Ajay sir never let us down. He always guide us and support us with any possible way. Thank you so much, sir, for joining us over here. So before closing the day, I'd like to thank all the participants who have traveled down from different places of India and joined us physically and virtually for this conference. I'm thankful to the universities of Odisha like Centurion University, Ramadevi University, National Rice Research Institute, Revensa University, and all the other ICR institutes whose scientists, research scholars have joined us in huge numbers for making this conference a great success. I am thankful to the print and electronic media for publicizing this event. I'm thankful to all our exhibitors who joined us from different institutes of Government of India for exhibiting and showcasing their research works and new technologies to the students, to the farmers and the visitors. So before closing for the day, not in order, I'm going to express thanks to all our sponsors and exhibitors, not in order. Larsen and Toubro, Central Bank of India, Odisha Tourism, Bank of India, APEDA, NABARD, Tea Board of India, National Medicinal Plant Board, Department of Biotechnology, NALCO, TriFed, City CRI, Coconut Development Board, COIR Board, Botanical Survey of India, Central Institute of Women in Agriculture, Central Institute of Brackish Aquaculture, Central Institute of Freshwater Aquaculture, Indian Institute of Horticulture Research, MCL, Canara Bank, Coffee Board, ICADA, CMFRI, Coconut Development Board, NRRI, OSWC, Bharat Petroleum, OAIC, NBPGR, National Seeds Corporation, CRIPCO, PCRA, Ruchi, HomeFed, Paradip Phosphate Limited, Ormas, Odisha Language, Literature and Culture Development Department, Government of Odisha. Lastly, before closing, I thank all our awardees who were recipient of the Krishak Bandhu Award. Thank you so much, dear farmers. It's you who not only feed us, but you motivate the upcoming generations. Thank you so much all. So as requested, so the last two sessions that we're going to continue it tomorrow morning at 9 a.m. sharp. Thank you so much. Thank you for joining AgriVision. Thank you.